Hello. Hello.
Hello. Hello.
May I request you all to please settle down? We're about to begin shortly. Cellular devices on silent mode. And for those joining us online, to please put yourself on mute as we will be starting shortly. I request all to please settle down in their seats. We're about to begin. Good morning, everyone. I, Sneh Bajpai,
And I, Lavneesh Thakur,
welcome you all to the second day of International Meet on Preparedness for Future Epidemics. Every great institution needs an innovative leader who can help direct the effort of the entire team into a fructious outcome. Thus, to begin with today's event, we would request our torchbearer, Dr. Pramod Garg, Executive Director, THSTI, to kindly begin with today's proceedings.
Uh, Good morning, ladies and gentlemen. A very warm welcome to this second day of our international meet on epidemic preparedness. And it's my real pleasure to welcome and introduce Dr. Rajesh Gokhale, who is the Secretary of Department of Biotechnology and Chairman of the governing body of THSTI. Dr. Gokhale, as you know, has been an eminent scientist working for the last 25 years or so in India on tuberculosis and has actually published excellent papers and contributed immensely to science. And now in his role as the Secretary, Department of Biotechnology, he is spearheading science as an administrator to facilitate all-round development of science. Science, as you know, has shown the way during the last 3 years how it can triumph over cynicism and come out on tops. So without further delay, I would request Dr. Rajesh Gokhale to give his opening inaugural address. Over to Dr. Gokhale, please.
Yeah, namaskar and greetings to all of you. Thank you very much, Dr. Pramod Garg, for this introduction and also for inviting me to this very important meeting on pandemic preparedness. I must say I'm delighted to be part of this really wonderful group that involves people who are researchers, clinicians, people from manufacturing companies, vaccine companies, and also people who are actually in the innovation setup of entrepreneurship of different kinds, actually. It's also important that folks like us who are part of regulatory ecosystem understand the really what is really required of us to be able to make this entire process amazingly gratifying. And as you already said, the COVID showed us the way that how science can triumph over all sorts of adversaries in life. In fact, from a government point of view, I can just say that the government of India is very considers science and technology as one of the key attribute of the way future of 21st century will emerge for Indian subcontinent, actually. If you really look at the real challenge right now for all of us lies is how we can be the serious competitor in the foundation technologies of 21st century. And what Department of Biotechnology is trying to do is to foster biotech innovations and enterprises across the country in various forums and activities that do. Also, it is strategically trying to bring together and nurture and strengthen the bioeconomy in the country, which obviously means supporting various endeavors of manufacturing companies or various folks, researchers, and be able to catalyze these activities. Some of the things that what we should really understand is in present environment, the technological solutions, in fact, have become a kind of it's also become a kind of weapon now for how countries' preparedness is kept together as well. I mean, as the meeting says itself, actually, pandemic preparedness, you can see how complex this whole pandemic preparedness is also issue is about, actually. In fact, a very recent science paper that some of you would have read about the origin of Omicron, you would have realized that even with the travel ban itself, absence of travel ban, the Omicron already was spreading around all the African nations, and therefore, one cannot actually stall the progress by actually taking actions that are that are limiting the travel and other aspects of the growth of humans, actually. I think what is important for us to be able to, therefore, have scientific policies in place, have scientific preparedness in place, and have capacity and ability to be able to make impact as and when required at a pace that is unprecedented and would be actually of tremendous value to it. Towards this, you would realize the Department of Biotechnology, in collaboration with its Section 8 company, BIRAC, and various stakeholders in institutions like wonderful institutions like THSTI, NII, NCCS, and various institutions of Department of Biotechnology, has done some fantastic stuff over a period of time. In fact, THSTI has done, and under the leadership of the wonderful leadership that Dr. Garg has provided, actually have done tremendous role to play in COVID mitigation strategies for this country, what it has been given to us actually. I mean, the kind of cohorts that have been created, the various immunological assays, testing, genomic testing, and various activities of development process has been remarkably tuned to utilize and make the best out of it in this given environment. I think we should all be proud about what we have done first, and my most important point to all of you. But obviously, we cannot be, complacent about our successes, because there are challenges, and not everything appears the way COVID times will appear. Actually, these are war times, and at war time we all gel together, we come together, we very quickly forget our very important other ideas and practice what we do. What we really need to embark upon is a very coordinated efforts of how careful analysis and systematic gap challenges can be identified and be taken up ahead, so that we actually create a synchronized system and mechanism that will make all this really worthwhile to do. So from my own perspective, from Department of Biotechnology, please be assured that we are all with all of you actually in anything that you're required to do, and anything that we can do to facilitate your process and be able to guide you to activities. There may be delays, and which is part systemic, but we are trying to correct those issues as well, actually. And I must agree that RCGM and various other regulatory bodies, in fact, worked remarkably well during this period to be able to catalyze what once used to be once in 6 months and 1 to 3 months meeting to 15-day meetings and 4-day clearances is absolutely unprecedented in the regulatory network phenomena of life, actually. I must also congratulate all of you for contributing to $80 billion economy that now bioeconomy that exists in the country. Actually, it's because of all your efforts is where we are today. Government of India, for example, I say in 1986 was when Department of Biotechnology was set up. And it's a very lean department, one of the smallest department in Science and Technology Ministry. And in fact, it's a gangbuster, I think I would call it, with about 400 scientists spread across the country, with whom all we work together in doing this fantastic stuff, actually. I think what we have done is what we really need to, converge on a mechanism that will actually make it more impactful in every aspect of disease biology, either in terms of, dengue vaccine, which is obviously a very imminent vaccine that we need to look at and develop, or maybe chikungunya, or maybe some of the other viral or bacterial infections, or for the matter of fact, the most dearer heart, the study that is close to my heart, which is of tuberculosis, actually, the biggest challenge that India has been facing as a problem for years to come now, actually. And I think it's all need to be solved through some really innovative solutions, because what we have been able to deal with, if you look at every single vaccine manufacturer that came to India, in fact, had a very, very unique innovation to their own kind, whether it is to do with starting to do with the recent success of nasal vaccines, or to do with mRNA vaccine, which is really the stable mRNA vaccine platforms, or the vector-based platforms that could be, enhanced and grown to such a high level, or for that matter, even the, heat inactivated or inactivated viruses. So I think we have done fantastically well. And of course, the DNA vaccine, which is the first of its kind in the country, and a wonderful work that was done, which was published in Lancet as well, is really something to be proud of how trials and activities can be done in India at a scale that makes us all feel wonderfully happy about it, actually. So I think I'll be happy to listen to, most of the talks rather than me only talking about it. But what I really want to convey to you is that THSTI is our flagship bearer of technology development, actually, partners. And I think We are really grateful for whatever the leadership they have shown over a period of time. And I think I'm very happy that Dr. Shailja Gupta, who was part of Department of Biotechnology now, is actually leading CEPI's program there at THSTI. She's been a colleague of mine for a brief period of mine, and I know how much she can make an impact to various programs and activities. So very happy to see the CEPI program that is going on there. And I again congratulate all of you for all the wonderful things you have done, but the path is complex. And I think the times are ripe for us to make an impact one more time to make huge difference to what this country is all about. I think let me just wish you all the best for the meeting. And, if you really look at very quickly, if you really look at the healthcare systems itself is in transition period, actually. From a reactive health, we are going to move to a proactive health, actually. And vaccination is an important component of proactive health, where you actually take care of vaccinating yourself much before you, in fact, get sick yourself into it. And HPV launch of the HPV vaccine by Department of Biotechnology was another wonderful example of activities that we're going to do. We're also hoping to make big breakthroughs in the gene therapy and stem cell therapy programs and activities that we are going ahead with right now in Department of Biotechnology. And many of these curative therapies are going to be the future therapeutic programs of how, we will be able to solve the problems of people and make healthcare a much more gratifying system, actually. So I thank you, all of you. Again, thank you, Pramod, for getting me over here. And I wish you all the very best and all my best greetings to all of you. Thank you. Namaskar.
Thank you very much for your kind words and tremendous support that we at THSTI have been receiving. And I'm sure we'll continue to receive all the support and help and guidance from DBT and under your leadership, which has actually transformed, all the ecosystem that we are experiencing. So thank you very much for joining, and we hope that you will be able to join for some more time during the course of the day and proceedings. I hand over the mic to Dr. Shailja Gupta for carrying on the proceedings. Thank you. Namaskar.
We now begin with our final session of the international meet, regulatory aspects, clinical trials, and human challenge model to accelerate vaccine development. Each talk will be of 10 minutes, followed by 5 minutes of question and answers. After concluding the third session, there'll be a 20-minute long question and answer session. To chair this session, we have with us Dr. Rubina Bose. Ma'am, can we please have you on the stage?
Dr. Rubina Bose has a regulatory experience of more than 23 years, working in CDSCO, the National Regulatory Authority of India, in various capacities. She has started her career in CDSCO as drug inspector, conducting GMP, GCP, GLP inspections. She has 5 years of experience in working in production of injectable drug prior to CDSCO. Presently, she is heading Biological and International Cooperation Division. Prior to this, she was also working as Zonal Head, Deputy Drug Controller in CDSCO West Zone. She is also working as WHO expert in various national and international meetings and has also represented India in ICH. Ma'am.
Okay. So I invite the people one after now or after the presentation. Right. Okay. So thank you for this invitation and giving me this opportunity to be here. I'm really honored to be in this session, chairing this esteemed panelist. I don't know that it is like a great privilege to me. And I would also in this context of all the experts, researchers, scientists, and the academias and industries experts being present here. Would like to say a few words on the topic which has been for the panel discussion, the regulatory aspects, clinical trials, and human challenge model to accelerate vaccine development. So as we have seen these unprecedented and the past 2 years, and we have seen also come across through these regulatory resilience. Actually, we have learned this in this pandemic period to how to respond. So I'd like to highlight some of the things which is a basic requirements for any regulatory aspects for a vaccine development. So we'd like to say that there are 4 steps in the vaccine development. So you start with the manufacturing of the batches for examination, test analysis, for development of the manufacturing process, and also initiate the stability studies, and also for the purpose of the preclinical studies. In the preclinical studies, when we start for the preclinical studies, what is needed is the vaccine is tested in the animal studies for efficacy, safety, including the challenge models, challenge studies, and also for the human dose determination. And there are also need of it is being tested for local and systemic toxicity studies. So this is just the basic requirements I'm just highlighting in this. Then going to the clinical development phase. So there are pre-approval clinical development phase, which includes the phase 1 in a very small groups of healthy adult volunteers, which also receive the vaccine to test for safety and immunogenicity. Then going to the next phase after a successful completion of the phase 1, establishing the safety with the dose determination, goes to the phase 2 clinical trial, where the vaccine is being tested in few hundreds of people who have the characteristics which is similar to the intended population, and also for those whom the vaccines is intended to be given is preferably chosen in the phase 2 trial, and it is tested to assess the safety and the immunogenicity. Phase 3 clinical trial is where the vaccine is given to large number of people and tested for efficacy and safety. Now, then comes the post-approval clinical assessment, which includes the assessment of the adverse event following immunization and the adverse events of special interest, and or the phase 4 clinical trial is also maybe needed subsequent to the phase 3 or a post-marketing surveillance under the post-marketing scenario. So coming to the topic in the accelerated development, what we need. So where the normal development of the vaccine, which is performed in each step, in case of a normal development, it is normally a sequential. So we start with the preclinical, with going to the phase 1, phase 2, and then to the phase 3. Whereas to accelerate the vaccines development, it can be done in parallel. Regarding the design, the concept of the adaptive clinical trials, which we have very much we have actually understood and used in this situation of the COVID-19 pandemic. So the concept of this adaptive clinical trials and authorization in emergency situation, which has been actually done, but the regulatory tools were already in place. Would like to see on the regulatory enablers of the new drugs and the clinical trial rules. We had all the provisions of the accelerated approval to undertake expedited review process, and regarding this decision, it was always based on the interim analysis of the data which was adapted, and finally the various vaccines was given into approval for emergency use authorization. Regarding the adequacy of the data for approval for restricted use under emergency situation, it was decided such data was examined in consultation with the subject expert committee. In case of also the accelerated development, the most important which is important part is that the assessment of the adverse event following immunization or AESI and the phase 4 post-marketing surveillance becomes most important and are very critical because the vaccines is studied and under the emergency and based, and it is being permitted. It has to be permitted to make it available on the basis of the interim analysis data of safety and immunogenicity. So post-emergency approval, a complete follow-up of the data on the safety and the efficacy, along with the post-marketing surveillance data, was mandatory to consider the continued emergency use of the vaccine, and finally for the full approval. So such application for all conduct of clinical trial or approval are given by CDSCO in consultation with the subject expert committee. And finally, all the vaccines are tested. Also, another mandatory part of it is the inspection of the GMP inspection of the facilities to meet with all the requirements for manufacturing of such vaccines. And finally, all the lots are tested by Central Drugs Laboratory, Kasoli. So each lot of vaccines are tested by the Central Drugs Laboratory, Kasoli, to release. So coming to the last, the point regarding the views on this human challenge model. While considering this importance of this human challenge model, would like to say that as it is understood that it is one of the important model in accelerated research program, the concept of conducting research in which healthy volunteers are intentionally exposed to these pathogens which can cause infection needs a careful ethical considerations, where right, safety and well-being of the research participants are of paramount importance in considering the regulatory aspects. Though WHO has come out with a guidance in January 2022, and its importance in the recent context is already recognized, it is still an unfamiliar research area, and there is a need to develop the ethical guidelines for this. Other areas which also need the regulatory considerations and development includes, in case of such human challenge model study, is the development of the strain, the manufacturing conditions under which, so there are discussions which are required for such strain development, the consent process, the volunteers, the site selection, and the other, the disease, the real risk benefit and the disease burden, the various research questions. So these are to be answered. So thank you very much for this, giving me this opportunity. And I would like to now, yeah. Right. Yeah, please. Okay.
To co-chair this session, it is an honor to have Professor Dr. Narendra Kumar Arora with us here today. Dr. Arora is currently the Executive Director at England Trust International and the Chair of NTAGI, National Technical Advisory Group on Immunization. Professor Arora was a faculty member of AIIMS New Delhi and retired as a professor of pediatric gastroenterology, hepatology, and nutrition in 2007. Professor Arora is a well-known public health expert in India and is extensively involved in policy-making program planning, implementation, and evaluation of public health, both at national and international levels. Sir, it's an absolute delight to have you here with us today. We request your presence on the stage, please.
I'm already there. Thank you. Sorry for being late. Right. So Rubina is asking me to make some opening. I think I just heard the last part. I would say that during last 3 years, regulatory system has transformed. I think it has really made huge attempts to come up to the expectations and requirements of the pandemic. And as I understand, right in April 2020, the regulatory system changes were brought in, and then a discussion took place in May and June. And that is why a lot of things happened at an accelerated pace. Having said that, over the time, when we look at the global regulatory environment, the pace with which the development took place concurrently, how regulators not only provided the clearances, but I think certain kind of guidance and clear pathways were also given to the manufacturers. And as a result, time which becomes an essence, both from a perspective of availability of the product for the community, simultaneously on the global platform, how we perform vis-a-vis other products on the same issue also becomes important. And I think that as we move forward to be labeled as a credible regulatory authority, I think that's the word which is used. We need to bring about, move forward, and I think take important lessons from last 3 years, so that we can then be recognized as one of the credible regulators, because India being called the pharmacy of the world on one side and the largest vaccine manufacturer, I think it is imperative on us, both from a health point of view, public health point of view, and the commercial point of view, that a regulatory system needs to be further fine-tuned in a manner that helps everybody concerned. So with these few remarks, we look forward to hear the presenters, and then like yesterday, we will try to have a brief 15 minutes presentation followed by 5 minutes of question answer, and towards the end we'll try to have half an hour for some discussion. So over to Rubina ji, please. Yeah.
Thank you. So I take this opportunity, and it's an honor for me to invite Dr. Vadhu Gupta, Technical Officer from WHO office, to present on the topic regulatory aspects and innovative approaches in vaccine development and the collaboration of vaccine stakeholders through the product life cycle. So I think it's no more, I think she doesn't need an introduction, and everybody is familiar with her. And she has worked along with all the regulatory agencies like the Indian regulatory agencies, and was very systematic in developing the institutional development plan, and has a lot of contributions to this. Thank you, please. Yeah.
Thank you, Dr. Rubina and Dr. Arora, for setting the stage with very pertinent opening remarks. And the theme of the session today, the first session of the day in the overall international meet, preparing for future pandemics and epidemics, is a very, very forward-looking theme on how, we have to consolidate our learnings through the last 3 years in India, how various science agencies have worked in unison along with the regulatory authority of India and achieved what they have achieved. So looking at the entire product life cycle and the vaccine development, WHO has been working hand in hand in collaboration with member states and very closely with the Indian regulatory authorities and the Indian stakeholders, the science agencies, etc. The COVID pandemic actually has tested the health system at large. The entire innovation landscape, the research and development, how we can cut short and accelerate timelines to for the regulatory system to be agile. And I'm really happy to share that not only at WHO pre-qualification, the agility part, the flexibility part to introduce the accelerated procedures, but also at the Indian Regulatory Authority, CDSCO, a lot of agility, a lot of flexibility has been seen. And, to tighten timelines, to shorten timelines, and to accelerate the approvals, look at those methods which could be looked at to shorten the timelines. So just a global bird's eye view on where WHO, World Health Organization, has been in the research and development efforts. Some of the critical elements which started coming in as soon as the pandemic began in 2020. A broad global coalition had to come together very quickly to evaluate candidate vaccines as quickly and as safely as possible. And this whole coordination role, WHO stepped in and created this global coalition, started mapping these candidate vaccines, where they were at different points in their life cycle. We all are familiar with the life during the pandemic on Zoom meetings and team meetings and WebEx meetings, and our life was limited on that laptop box, the screen of laptop, and the flurry of meetings one after the other. So that is how all of us have lived, in vis-a-vis meetings, global meetings, regional meetings, etc. So defining the correct target product profile characteristics was one of the key roles WHO took. Desired characteristics of safe and effective vaccines, and coordinating clinical trials, global clinical trials or across the world, for safe and effective vaccines as soon as possible. For this purpose, there was a COVID-19 vaccine tracker, which was developed by WHO, which compiles detailed information on each of these COVID-19 vaccine candidates in development. And these reports were periodically updated, published on the safety data, immunogenicity data, and efficacy data of the vaccine candidates, very periodically and updated. Every almost a week, the updates were going up through the pandemic periods on the WHO global websites. The key attributes of each of these vaccines, the information required, what is the vaccine platform, schedule of vaccination, clinical endpoints, basic information, as it was emerging dynamically, it started getting updated. We all have heard about the Solidarity Trial, the global Solidarity Trial in India for therapeutics, which ran in India very successfully at NARI, ICMR NARI, but the Solidarity Trial for vaccines was also run by WHO. It is a global trial which was run by WHO for promising new vaccines for COVID-19, along with the ministries of health in countries. And it was delivered on a global scale, a global rolling testing platform with adaptive clinical trial design was formulated for this trial. Since April 2020, this whole access to COVID-19 tools accelerator, this ACT accelerator was led by Dr. Soumya, actually as a chief scientist in the World Health Organization headquarters Geneva office. And it is with her leadership that this ACT accelerator partnership was launched by WHO with partners. And from research to rollout, the ACT accelerator remains the world's only end-to-end solution, for the not only the research and development, the manufacture, the production, the supply chain, coordinating the financing solution at the countries, how the drugs or vaccines will reach the country, what are the challenges it will face, and across the three products, vaccines, therapeutics, and diagnostics. It's called COVAX, with the cross-cutting health systems and a response connector to actually address the challenges which are faced at the health system level. So vaccines pillar of the ACT accelerator is called COVAX. And COVAX is co-led by CEPI, the Coalition for Epidemic Preparedness Innovations, Gavi, and the World Health Organization, along with our key delivery partner, UNICEF. And WHO had multiple roles in COVAX. not only the normative role on vaccine policy, regulatory role, looking at safety aspects of the vaccines, research and development, country readiness and delivery, and this whole stream actually occupied a lot of time and energy since the pandemic began across the 3 levels of WHO, headquarters, region, and the country office of WHO. SAGE, as we know, is advising WHO on the overall global policies, emergency use listing procedure, pre-qualification program. The entire emergency use listing came forward with the advice of SAGE, and the countries then adapted this to their own country settings for assessing unlicensed vaccines, therapeutics, and in vitro diagnostics during the public health emergencies such as COVID-19. WHO target product profiles were developed, and these target product profiles were continuing to get updated on the WHO website as and when the updates were coming in. WHO's current priorities for vaccine development were also, reflected in these TPPs for the COVID-19 vaccine. The landscape of observational studies on effectiveness of COVID-19 vaccination. This landscape lists 1,887 observational studies on vaccine effectiveness. So this, is getting, again, it's a large exercise which WHO has undertaken in the pandemic months, in the years now, pandemic years. We can say we are in the 3rd year of the pandemic. And it provides an overview of different observational studies at different time points where they are prospective, retrospective. You can see, cohort studies, cross-sectional at a current point in time, and various endpoints data is getting collected in this as well. So broadly, I think the solidarity trial, to just sum it up, the broad key takeaways from the pandemic period is in the vaccines context is the global WHO solidarity trial, the creation of all these normative target product profiles, the observational studies mapping, the COVID-19 living map, the COVID-19 candidate vaccine tracker, etc. And the first mRNA vaccine coming out, the Pfizer vaccine, to receive emergency use authorization from WHO since the outbreak. And it was done at an unprecedented speed with the close support of the regulatory authorities in the countries they were coming from for all vaccines, which were developed during that time. Coming to the aspect of newer and innovative global approaches, which is a theme, one of the critical themes of this session. A lot of the innovative global approaches currently underway. I will touch upon just 2 or 3 approaches for the sake of time. What you see on the screen is a Eureka digital platform for clinical trials developed by NIH USA to facilitate mobile and internet-based the medical or health research for any interested investigator. Platforms like these, I think, need to be leveraged and stitched, for large multi-centric studies in our country, not only industry-led, but also academic research studies for using technology, digital technology, and in creating that enabling data collection and the entire life cycle of the clinical trial using technology for that. And this was developed as a national resource in the States for digitizing and mobilizing health research. It's being used by many universities and many industry partners also. Now, so I just wanted to flag this here and as a food for thought that maybe we could look at such approaches in the future. Dr. Rubina very aptly touched upon the human challenge trials, and the concept indeed is very promising. It is a new concept, newer concept in the recent years which has come, and CDSCO has participated and shared their thoughts in several of the meetings over the last 3 to 4 years on this subject, which were coordinated by WHO and THSTI jointly. These human challenge trials are indeed very promising and an approach which is being looked at by both developed stringent regulatory authorities as well as the developing country regulatory authorities as another model, which needs to be looked at as a tool to enable licensure, quicker licensure or accelerated licensure of vaccines. Vaxchora is the example from US FDA, where they have licensed this vaccine. And our typhoid vaccine, which is our indigenous vaccine, which has actually got the human challenge trials done in Oxford in UK with the help of the UK Oxford University. And now that is a WHO pre-qualified vaccine, which has leveraged this human challenge trials approach. And the US FDA actually based their approval, their licensure, based large part of that was based on the human challenge study results. So WHO had been very curiously looking at how this whole area of human challenge studies was developing. And over a deliberation of almost 1 year, 9 months, this ethical guidelines of WHO has come out in May 2020. The key criteria for ethical acceptability, just at the time when pandemic was beginning, the guideline was released. And what are the ethical criteria, ethical acceptability, and the WHO's view on that is. So I would urge all of you to definitely refer to this guideline. In India now, as Dr. Rubina also highlighted, there is a need to develop ethical guidelines for these human challenge studies with collaboration of all the stakeholders, with ICMR, with THSTI, and CDSCO, and the others. Vaccine safety initiatives, few of the very innovative initiatives in India. One I would touch upon with close collaboration with CDSCO, WHO country office has brought the smart safety surveillance approach in the for the monitoring of the vaccines, more like a data triangulation from different sources of the adverse events following immunization, in collaboration with AEFI Secretariat, Immunization Division, Ministry of Health and Family Welfare, the CDSCO, and the WHO, along with other vaccine stakeholders in the country. So institutions in the country which were collecting data on Rotavac vaccine, the first oral Rotavac vaccine developed, it was selected as a vaccine pathfinder for this approach to introduce and test the smart safety surveillance approach. And a white paper was developed in quite a short time. Over 1,500 cases of intussusception were analyzed, and through a self-controlled case series, this white paper actually provides assurance on the safety of this vaccine, in the country and for global use. And this white paper was released in the World Conference on Access to Medical Products in 2019. And it provides a better characterized safety profile of the new vaccine. There is another concept of signal management and safety monitoring, which is now taking picking speed in India. This is a panel, an independent body at the national level, which is continuously looking at potential signals of importance from databases of our own national databases, adverse events following immunization with COVID vaccines and the other vaccines. So this panel is already up and about and is very dynamically working. Professor Arora is contributing, and Dr. Rakesh Agarwal is chairing the meetings. And just a glimpse, this is my last slide. This whole approach of signal detection and assessment for safety monitoring of newer vaccines. India is the first country in the region which has developed this system in the region. So this is indeed a very, very big achievement that we have this system up and running, and it's looking at Covaxin, Covishield, and Corbevax in India for COVID-19 vaccines, but it's going to move very soon to the other UIP-based vaccines and look at their safety signals, etc. These are some of the meetings which have happened and in the past almost 6 months, 8 months. So I will end with this. Thank you. Thank you very much.
We can take. Yes, sir. please.
Yeah, good morning, doctor. This question is regarding, I'm Mihir Chai from Bharat Biotech. This question is regarding the Eureka platform. And here,
Yes, we can. I can see you now.
yeah, this is regarding the Eureka platform. There was a particular statement that caught my eye, which says remote recruitment opportunities. Now, how does this in Eureka tie in well with NRA and with complete informed consent of patient? Because in India, I don't think remote recruitment would be something which is pragmatic at this current juncture.
Thank you. I think that's a wonderful question. In fact, there are many things in this platform. I have myself been wondering about how will it work on the ground, actually. You know, you have this phone, mobile app. How will it recruit patients? How will regulators validate? How will the site staff validate? So for this, you know, we have asked them to provide a demo to some of our academic institutions in the country, some of the ICMR institutions, some of the other academic institutions, and some industry partners. So I think that is planned soon, sometime in January or February by the NIH. And I think it will be useful, Dr. Veena, also for the AFI Secretariat and maybe the CDSCO regulators also to see what the demo is about. But I think if a lot of institutions and developed country industry partners are using this for large trials, which are located in different countries, not only in US, they are located in different countries with both developing and developed country settings. It is worth to look at, you know, similar solutions, not only this one, but similar looking solutions as, you know, a digital enabling tool for clinical trials. Because, you know, the artificial intelligence, the digital technologies, as we know, permeating every sphere of our life, whether it's education, whether it is finance. So I guess it is this one for clinical trials, how digital technology can be leveraged or make the timeline shorter or make it easier for patients, for doctors at the site to monitor the patients better. So it would be worth to take a look. I don't have a ready answer for that. I'm also myself preparing a lot of questions for that demo as well. Thank you.
Yeah. Okay. Thank you very much, Madhur. Is there somebody else? Yeah. You can go ahead. One more question if you want. Okay. Thank you very much, Madhur, for that very overarching presentation and making us understand the various initiatives. And maybe when Eureka demonstration takes place, let the stakeholders know. It looks very attractive and exciting to understand that. Thank you very much.
Thank you.
Thank you, Dr. Madhur. So I would like now to invite Dr. Neeraj Jain, from PATH Country Director, to give his presentation on the regulatory in the accelerated vaccine development. So as everybody knows that he's having over 30 years of extensive leadership experience in strengthening organizations, and his strengths lie in business development, setting up new ventures, ecosystem development, strategic leadership. So thank you, Dr. Neeraj. Please, over to you.
Thank you. Thank you so much. Just give me 1 second. So firstly, I'd like to say I'm really honored to be here. I'm probably the only non-scientist in the room. And when Dr. Shelja invited me, I was wondering whether she sent the invitation to the wrong person. So thank you for having me here. And full disclosure, I'm not even a medical doctor. So I'm going to stay away from the science and really talk about the regulatory aspects and what we've learned from our work in India that PATH has done, plus globally. And hopefully that'll give us some sort of a direction on possible pathways. Just to talk about a little bit about the vaccine landscape, and especially in low and middle income countries, and that's going to be my focus today. The WHO came up with the Global Vaccine Market Report, which shows inequitable distribution, not only unique to COVID, but actually all other vaccines. And developing countries really struggling in terms of being able to access vaccines. I think one example is the HPV vaccine, where only 41% of the countries around the world, low middle income countries, have got access to it. Whereas the wealthier countries, of course, do have full access. In terms of value, in the last 2 years, of course, COVID has seen the landscape has really changed over the last 2 years. In 2021, we had 16 billion vaccine doses in total, worth $141 billion, which is almost 3 times of 2019, where it was 5.8 billion vaccines with a market value of 38 billion. So from $38 billion in 2019, the market has gone to $141 billion in 2021. So you've seen this huge change in those numbers over the last few years. And although manufacturing capacity has increased substantially, it still remains very concentrated amongst the top 10 manufacturers. Not including COVID, they still provide about 70% of all the vaccines globally. And some vaccines like PCV, HPV, measles, rubella, rely on actually just 2 suppliers, mostly from the global north. This also leads to risk of shortages and regional supply insecurity. And the reason I'm touching on all of this is because the regulatory environment in low and middle income countries actually has a huge impact on how this inequity actually is playing out and how it will probably increase going forward. Another interesting point I'd just like to make is in 2021, the African and Eastern Mediterranean regions were dependent on manufacturers headquartered elsewhere for 90% of their procured vaccines. So again, huge amount of inequity in terms of where the vaccines are produced and where they're actually used. Now I'll come to the topic that was given to me, and that's on enabling regulatory conditions. See, we've seen, PATH has been operational for 45 years. We worked on, at any given time, we work on about 20 plus vaccine candidates. And some of the vaccine candidates that we've seen and we've gone through the whole process have seen, so for example, Japanese encephalitis from China. Started in 2003, and we've just got the WHO pre-qualifications just a year ago. The MEN-Afrivac started, it was between 2001 and 2014, and that WHO, DBT, a lot of us were involved. Serum Institute, of course, was the main partner. The RTSS malaria vaccine, 1987 till current, now it's finally starting to roll out in some of the African countries. So there is a long process, it's a lengthy process, and rightly so because of the risk factors involved and regulations that are required. The only challenge is this very well coordinated, and probably we could do better. We've seen also during the COVID vaccine development, a lot of this has played out. Of course, it has also played out because of the pressure of the disease itself. But how are we going to somewhere come in between these 12, 13 years and where we were with the COVID vaccine? Clearly, we realized that the approaches that we had were not suited for an epidemic. And I think CEPI had already started thinking about this even before COVID hit us. So I remember the first time we read about CEPI and we talked about CEPI, there was a big question mark as to, do we really need a CEPI and what are they going to do? But we clearly saw their role playing out during the COVID pandemic. I think the slowest part, as already mentioned by Rubina ji also, is the testing piece. And, how do we test them from animals to humans, etc. And we've talked about human challenge models, so I'm not going to go there. But it also, so the challenge is that these processes vary in time frame very much from region to region, country to country. And that is where I would actually look at my colleagues from WHO to lead on some of this, this regulatory process becoming more systematic, more equitable, more sort of a global process rather than just country by country, both for chemistry, manufacturing controls, and non-clinical, clinical. I think it's very important that the difference between different regions is very, very big, and we need to try and bridge that gap. The COVID vaccine, we saw accomplished, we managed to actually push through a COVID vaccine in less than 300 days. I know CEPI wants to take it to 100 days, and it's definitely possible, I guess, with all of you in the room. But we need to think about what we need to do in the future to possibly get closer to that. Couple of thoughts from our experiences across the globe, and I'd just like to share them with you. We need, firstly, in terms of the regulatory process, we need it to be, of course, very, very strong. Risks are high, needs to be very efficient. How are we going to become extremely efficient? And maybe also increase transparency of the regulatory process. Maybe some way it's, the visibility, the transparency is actually something which would really help. And especially, again, I will focus on manufacturers in the low and middle income countries. The smaller manufacturers, the manufacturers who are waiting in the wings, the manufacturers who have just started, they are the ones who are mostly challenged. We could reduce multiplicity of approvals. Lots of approval processes. Can we reduce them? Can we actually try and harmonize clinical trial applications? Look at something which is probably global. I think that would really help to just faster clinical development. I think that's an important need. I think also global bodies like WHO need to partner with the national regulatory authorities, the NRAs in multiple countries, to establish and implement some of this. So a stronger partnership between these would be very, very useful. We need to have a common global set of requirements for clinical trial applications as per phase development. Can that be set up? A parallel ethical committee and regulatory authority review rather than sequential. Is that possible? These are just ideas, and I'm going to leave them with all of you. You're the scientists working on this day in, day out. But these are some of the thoughts that came out of our system in terms of how we, and I must tell you also, with Africa starting to emerge and having a lot of conversations about becoming a vaccine hub by itself, a lot of these things are actually starting to play out there. A lot of these conversations are also playing out there, which is what I'm also sharing with you. As I said, clear, transparent assessment timelines and commitment to these timelines will be absolutely critical. And are focused on reduced regulatory burden overall for NRAs, ethic committees, and vaccine developers. Again, at no point letting go of managing the risk, but really reduced regulatory burden. If we could look at the current regulatory process and see how that could be tightened. The emergency use listing, absolutely great, very, very useful. We couldn't have managed COVID without that. And can we actually go somewhere between emergency use and full clearance in terms of timeframes, or we could have a stepwise approach, could be a possibility. I think India has really taken the lead, in the EUA guidelines at the height of the pandemic. I think, India is where we are without, I don't see anybody wearing, I have seen maybe 2 people wearing a mask now. So clearly we've achieved a lot in this process, but I think more to be done. Very important also, we are now, we have the presidency of the G20. And we are talking about South-South cooperation. India is 60% of the world's manufacturing of vaccines. We have shown the world that our regulatory processes are strong, our systems are strong. How can we help other low middle income countries achieve some of this? How can we, share our learnings with them in terms of regulatory is another area which we could look at. Recently in Pune, there was a developing country vaccine manufacturers network meeting. I was also present, and I know a lot of you were there. Lots of work that can be done South-South, which we need to look at. Having said that, a lot of the manufacturers are hamstrung by funding. Definitely regulatory limitations. They also funding the regulatory process is also a challenge. How do you fund navigation of the regulatory process? The bigger ones can manage, the smaller ones struggle. Lack of access to data, technology transfers, property monopolies. So all of that sort of adds up. In terms of the CEPI roadmap, which is 100-day vaccine manufacturing, I think a little note to everybody who's here from CEPI, or if not, if the message could be sent across. We feel that there is a perception of a lack of transparency from CEPI in terms of selection of manufacturing partners, and also a lack of clarity on what the requirement is, making it very difficult for smaller manufacturers to actually come in and participate in some of this work. So maybe something that could be looked at. Especially when we start thinking about pandemic preparedness going forward, vaccine stockpiling. So I think those are areas which CEPI could help. My final point before I close, I think backward integration is another important piece that we need to think about. The essential raw material supply chain systems need to be developed. And again, there is a regulatory aspect to that. How do we make it simpler so that there are better supply chains in the developing countries that could be actually leveraged? Some Indian companies like Serum are working towards this, but I think it'll require a much more well-coordinated approach by the government, by WHO, and by all of us in the ecosystem. Thank you so much. Thank you.
Thank you, Neeraj, for sharing your valuable experience as well as perception and opinions how to move forward. Just want to share that as I understand, Southeast Asia Regional Office is taking initiative for the 11 countries to bring the regulators together and strengthen and harmonize the activities with them. So, but I think that's an extremely important step where the countries come together and there is better kind of standardization of processes. We are open for questions. Yes, Dr. Sanjay Singh.
Hello. I have a small question. How you feel that PATH can play a vital role in this process?
I think what PATH brings to the table is the learning over the many years, the learning across geographies. Just to let the audience know, we do a lot of work with Chinese manufacturers of vaccines, Vietnamese manufacturers of vaccines, in Brazil, I mean, all over the place, Bangladesh. So if we really think of an organization who has actually visibility of a lot of that, PATH is probably very well suited. So we are advisors. We're happy to provide the information. We're happy to provide our thoughts. I think somebody needs to lead it, and probably it could be the government of India, could be WHO for sure, bring it all together. But in terms of the information and the visibility of the globe, I think that's where we could bring value to this conversation. Thanks. I thought you were going to ask me a technical question. I got a little worried there.
Any other questions? Yes, Shailja.
So do you just advise, or do you also help manufacturers with the process, regulatory process? I mean, advise and then work with them to through the regulatory process, because there are a lot of companies, both startups and big vaccine industry here, and maybe that would be useful then.
Thank you, Shailja. We do. So firstly, we are a not-for-profit, so we need to get funded for whatever we do. That always becomes a sort of a challenge. Unless we have the funding, we are not able to actually provide support. But yes, we work with pretty much all the manufacturers. We work with Serum, we work with Bharat, we work with BioE, we're now working with Genova. So we work with all manufacturers across the world in supporting them. But our only challenge is that we also have to have the funding to be able to do that. But yes, happy to work with anybody who needs help. Advice, we're happy to provide free advice, but if we have to start really working, WHO pre-qualification, for example, I think PATH has been leading on that, on many of the vaccines. And Dr. Rao can probably answer that better than me.
That's what I was taking. Yeah, just thank you. PATH has really helped us in the Rotavac development, entire WHO pre-qualification. In the beginning, we had a collaboration with WHO, PATH, and Bill and Melinda Gates, where the PATH was the leader in the corporate. John Bosclago and the entire group, we were put together. Thanks to PATH, really accelerated our Rotavac development. Thank you, Dr. Jain. Thank you.
Thank you, sir. Thank you. As I said, Dr. Rao would be able to answer that better than me. I must say, I'm also really honored to be here because we did a lot of work with Dr. Bhan. And, I've never been to this auditorium, so it's just a really nice feeling in remembering him today. Yeah.
Thank you. Any other? Thank you very much. And a very important point I think you is made, 2 points. That one is transparency of the processes, and the second is level playing field for all the manufacturers. I think these are 2 very important points, along with the point you're making, navigating through the regulatory processes, where PATH has been playing a very important role. Thank you very much.
Thank you, sir.
So I have the honor to invite Dr. Sanjay Singh, Chief Executive Officer and Director, Geneva Biopharmaceuticals Limited. So just to not to elaborate, but to mention that he has led Geneva to develop a messenger RNA vaccine and establish India's first mRNA-based vaccine platform. So I would like to request him to deliver his presentation on regulatory framework and innovative vaccines, path forward for pandemic and non-pandemic situations. Over to you, Dr.
Thank you, Rubina ji. And thank you very much, Sailja ji, for inviting me. And invitation came from her, so I said I must go. So this is more for the students and scientists, because I will go into the hard data. So Neeraj ji was worried. So here, the talk is India ready to meet the CEPI 100 days vaccine challenge? Yes, we are ready. The only thing, what are the ways into it, what are the means into it, how you want to do it, those are the some part I'm going to discuss. But the most important things comes into it, that as a science ecosystem and as the Department of Biotechnology, our science side is much stronger than most of the nations. And we can say we are as good as developed countries. And that shows from our biotechnology, our product in the market, how we are competing, and similar kind of vaccines was there. But where the regulation was 10 to 20 years to bring a vaccine, and CEPI is saying to bring a vaccine in 100 days, it needs transformation. You cannot use bullock cart to go to moon. You have to develop some advanced sort of rockets to do that. So this is one point I wanted to say. The second point I wanted to say that we must develop our stringency and systems so we don't have to look towards others. And this capacity we have in-house. So for every time, if you have to look towards WHO, you have to look towards FDA, you have to look towards EU. Why we have to look towards? Because they don't look towards you. And I'm very practical on this. They don't look towards you. So we have to do it. So this is one of the examples I'm walking through. So what are the vaccine platforms available? It is generation 1 vaccine. And it has given a lot of vaccines, and it has helped a lot to work around for generations. This works. It goes. And then the second generation came when this DNA sequence and protein sequence came. This is around 40, 45 years ago. It came where few of the vaccine came, like hepatitis B, HPV, seasonal influenza. They have also made a great role. But what I'm saying that this is the way the transformation started happening. And Then next generation came adenovirus non-replicating DNA or plasmid DNA vaccine, and one of you see that Zydus developed this DNA vaccine. And the last one came, which is we used to say vaccine 4. This came as a COVID vaccine, and before COVID, none of the product was approved from this platform for the vaccine. For therapeutic was approved, for gene therapy, but for vaccine was approved. We at Genova were developing something which is in the oncology, personalized cancer vaccine space with neoantigens. But when it came, then we decided, can we work towards it? And very thankful to the secretary at that time, Dr. Renu Swarup, and still the same support is available. She was keen to listen the talk and said what we can do. I said, this is going to be a huge program, which may cost 500 to 600 crores. We as a small company cannot take the first load, but we'll make sure the vaccine is delivered. And the first Indo-CEPI funding came, Dr. Selja, the first funding was Indo-CEPI, 25 crores came into it, and we started the program. Why I'm saying that if you have to say these 100 days, then you have to produce in 100 days. Because the slogans are for politicians, not slogans are for scientists. If we say it, we have to produce it. Here's the vaccine in the vial. So one advantage with this platform is that you can start in the morning, by the evening you will have the vaccine available. Because it is whole synthetic platform. It's just a test tube with synthetic, only you can increase it. But yes, there are many factors comes into it. So what we did, we developed it. Finally, we gone through CIRISCO phase 1, phase 2, phase 3 trial. And also we have ensured backward integration of raw material, because this was a huge problem for us when we were developing the vaccine. And here we have all kind of criticism for regulatory system. But if my regulator, which is directly Dr. Somani, would have not been there, we would have not developed the thermostable vaccine, which we are alone in the world. Alone in the world who has the vaccine available at 228, and now our 6-month data stability coming, they are stable at even 30 degrees centigrade. He gave the opportunity, we came face-to-face presentation, and we said these are the possibilities. And because vaccine equitability, democratization of vaccine of most advanced platform was not possible if this vaccine is not available at 2 to 8 degrees centigrade, because our whole distribution system is based on 2 to 8. We don't have -70. If we have to send to Arunachal Pradesh, it is not feasible. So this is very important for youngsters. We have to innovate for our needs. If we are not innovating for our needs, yes, papers in science cell, they are very important. But equally, it is important till your science is not translated into human impact, that science is less regarded. So we had it. And why we did it? Because at that time, why we chose the mRNA platform, because these viral diseases, when it transmits very fast, they have a very poor proofreading system. And then the mutation starts. And we were knowing that mutation will come. And that was one of the reasons to choose the mRNA platform. And here what it gives the glimpse, if you see that if you have a new variant, you start day 0, and you can make the human injectable vaccine within 50 days, once your platform is established. This is the uniqueness this was there. And here I would like to take the credit, but I got a call from the top government official that when Omicron came, and it was, I remember, on the December 30th around 9 p.m. And Professor Arora is aware of that fact, that is Omicron is coming, what you want to do it. So here I want to say, this is for the students, we have broken down this whole process. So from day 1 to 2, day 3 to 4, day 5 to 22, day 23, 28, day 29 to 31, day 30 to 33. We have broken out this whole system, and here we have made a huge investment. Everything is under one roof to do it. Everything is under one roof to do it, and we started this. And on 27th of February, we were having, December 30th was 27th of February, we were having CGMP 3 batches made vaccine into this. But not from this point. We have tried to make a transformation. But the problem happened, and this is the Omicron variant was there. You can see how many mutations we have gone through and worked around and designed it. But here I would like to say, and this is not from point of criticism, it is point of how we can do, Rubinaji, hand to hand to work it around. Because it is very important for the regulator to see the safety of these vaccines, because we are giving to healthy volunteers or healthy people, and the vaccines are going to millions of people. So if we have not evaluated well, there will be a problem. So this is the day we started. Within 44 days, we were having the CGMP grade vaccine available, but you have to go through such a rigorous process. You have to go to RCGM and they have to ask some question, and then RCGM will come and this will go and this will go. And what is the problem is, and I'm sitting from the open platform, we have to build capacity of knowledge, capacity of resources under one roof, which is CDSCO. Because we cannot go for local FDA license to local FDA, then local FDA will send to CDSCO, and then it has to come. The RCGM has to do, RCGM has to send to CDSCO, it has to come. There are people temporarily sitting on the RCGM committee. I have nothing against them. But this is a very knowledgeable subject, which earned the knowledge. At present, the person who's Professor Arora knows, David Caslow, to whom I replaced at NIH because he decided to leave the tenure and I was the person who joined at his place. He has now chief of vaccine research at US FDA. Minimum 25 years of hardcore experience in the vaccine biology. And why I'm naming him, because he was also at PATH for so many years. So this is the knowledge which is getting accumulated at our regulator place. And this is continuous knowledge should be accumulated. Not somebody has been transferred in 3 years, somebody has been done some, no. If this fellow, which was the first one, which was retired and after that, this came out, Norman Baylor, he was head of the vaccines for 28 years at US FDA. And he's the one who has approved the maximum vaccines in the world. So my request as a scientist, I may be wrong, but my request as a scientist, this should be under one roof, under one door, where I can go and talk to. On the other hand, I'm saying that resources, DCGI help us to bring the 2 to 8 degrees centigrade vaccine, but also on the other hand, and if you see my pain, we have made in 44 days, but RCGM taken 174 days. 20 times they have had the meetings and even out. There may be some reason, there may be some weaknesses, and then 61 days gone, and we have done 3,400 phase 2 and phase 3 trial within 37 days. So industry is going to bring transformation. Industry is going to bring innovation, and we as industry must know that nobody has taken Fiat Ambassador out of the market. If the technology is not there, you will be out. So you have to come with the best possible technology, you have to come best possible regulations, and you have to come beat these kind of possibilities. Now I will go for the last slide. This is why important, because this is mRNA is a disease agnostic platform. And again, for the young students, here means everything is same. Your facility is same, your quality assurance is same, your quality tests are same, everything is same. Your raw material is same, these are the things. Only what you have to change the plasmid. Once you change the plasmid, mix these things, and finally the vaccine is available. And that is why you can do this in 54 days. So I'm saying there is a role of technology. If we have to transform for 100 CEPI days, we also have to somehow work hand to hand and make sure how it is. And there is one suggestion coming from me for this. The mRNA platform, you can see it. As Rubina ji said very clearly, preclinical toxicology is needed because Moderna and Pfizer were not asked to do preclinical toxicology, but Genoa was asked to do 2 preclinical toxicology back to back. Then phase 1 trial, and here animal challenge trial can be done, because once the phase 1 has in limited people has been demonstrated, we should immediately jump for phase 2 clinical trial and human challenge model. And once it is through, then we should start doing limited phase 3 trials and see the results of that. This is the thought process which is emerging at global level, but I wanted to say it. And because Selja ji asked me to talk about this, today 17 human challenge model is available. And we may going to use 2 of them. One is for leishmaniasis and one for dengue at NIH. So these are the human challenge model. This is report is just came in Clinical Microbiology Review in September. 17 things are available. We have to bring these tools if we are dreaming about having a vaccine in 100 days, and we can do it. Only thing, concern of safety has to be addressed, collaboration has to be addressed, hand to hand has to be done in this way. Thank you very much. And again, I would like to say, without Indo-CEPI support and without DBT support, mRNA platform would not have seen the day. So thank you very much. Thank you.
Thank you very much, Sanjay ji. We are open for, you have raised some very important points. So let us have some questions.
No questions, sir. I
Yeah,
think.
no, there are, yeah.
Yes, it's a very good presentation. But I would like to add one more thing over here for fast development of vaccine. Another thing is, once the sequence of a virus is known, then its epitopes can be mapped. So the immunodominant epitopes, they can be selected. Epitopes which are immunosuppressive or have auto-react with human proteins, they can be eliminated. So taking into then consideration all these epitopes, immunodominant epitopes, a gene can be synthesized. So mRNA-based vaccine or DNA vaccine can also be made based on immunodominant epitopes. So this may also be a very fast process using bioinformatics tools.
Yeah, thank you. This is an important point. So when COVID vaccine was going on, there was a possibility that antibody, disease-based antibody enhancement can happen. So at that time, some work was done by at NIH, and if you go back, you can see certain publication from those groups came. So at present, somehow this concern has been, we have seen in dengue. Because of the patent confidentiality, we cannot explain it, but this is an important point and we are putting into consideration already. Thank you.
Yes, sir. Please.
Hello, sir. Good morning. It's a wonderful presentation. Thank you for that. We'd love to know, since you have discussed about your mRNA and so on, so how it will counter when there is so many mutations happened, first. And second, what is the efficacy of the proposed mRNA that you have talked about across the Omicron variant or Delta variant or whatever we have?
So at present, you may see a lot of publication and everything about making the universal corona vaccine. I think Sweetie is sitting here from THSTI. We have a program making universal flu vaccine with European Union. We have not succeeded in making the universal flu vaccine yet, because this is not a defined process by which these mutations comes. And we have 2 systems. One is pseudovirus system by which we immediately test, just like B.1 we have tested in B.1, B.4, B.5, whatever new are coming. So we are testing in vitro immediately, immunizing the animal, getting the sera, and we are testing it. So this is test model, but if you say I have one slide later on where we have explained. So there are 3 generations we feel for coronavirus. One was first generation, which was the base vaccine. This is mutant specific vaccine has come, and we might have to make with time few more, just like in flu every year, we have to make few more. But the third is possibly that we might have to enhance the T-cell response, memory response very heavily, which hopefully some data will come from our side, because we have changed the route of administration. We have used intradermal in doing our booster, and let's see, because memory is very important. If memory is not there, then you virus cannot identify. So this is hardcore basic biology. So we are into that direction, and hopefully we are waiting to get some good result. Priya ji.
I loved you. I loved your presentation, and I just wanted to ask you, your platform that you use for COVID-19, would it lend itself to organisms as diverse as tuberculosis, where, you know, antibody response, we don't know how good that is in actually eradicating the infection in likewise for leishmaniasis or fungal infections or diverse viruses like HIV.
So you may knowing the name San Crotty, there's the immunologist San Crotty. Certainly, so what is happening? All these foreign organisms make the proteins. Once these foreign organisms make the proteins, sometimes like malaria, like tuberculosis, their expression pattern, because we are using body as a factory, we are analyzing it that whether those post-translational glycylation are making an impact or not. But we are working in tuberculosis with the program at AIMS. We have a huge list of programs where we are working, because for us, for Genova, COVID was one of the byproduct from our mRNA platform. It was not the byproduct. And now you must be very pleased to hear this, that WHO invited last month for its mRNA hub program. And I hope that we can work closely with WHO and together we can make mRNA hub as a success. So let's see how it goes.
Yes, Ramaram.
Yeah. First of all, I'd like to congratulate you for setting up such a very important technology. The question is that I know you're very frustrated. It took a long time for you to get through the regulatory, but once now you have human data, do you think it will take the same amount of time for the next vaccine?
So I decided to leave my NIH tenure and come here knowingly all things very well. We are working with regulator and every day we are improving. So it is question of difficulties must be explained. The son has to explain the mom, it is hungry, it is having a problem, that is the nature. But it is going to, again, the way we have transformed the manufacturing, I am very confident in next 5 years, our regulatory is going to transform. I have full faith into that.
Yeah, sure.
And this is a general question. Sanjay, you talked about carrying out human trials using human challenge models for dengue and chikungunya, you said?
Leishmania.
Leishmaniasis. Two of these diseases, which are of great importance and almost epidemic in India. How then how will it work in India when we are not allowing human challenge studies in India? Because CDSO is not going to approve any vaccine which has not carried out human trials in India.
So,
how does it work?
Yeah. So nobody approved the vaccine based on human challenge model.
Okay.
In US also, it is not based on challenge model only you can do. Because challenge model is just giving an indication that it is effective vaccine. So what happens, we have done the phase 1, we have done the phase 2 and human challenge model. We go back to US FDA and present our data. US FDA gives a fast track process. It is a voucher process. So once you get the voucher process, it means CDSCO has decided that this program has met its guideline. What are the ways by which we can approve the product very fast? Maybe 3,000 trial or 5,000 trial or 6,000 trial, but that whole machinery is on the expressway, the fast, that. So they are looking, but for that doing, they have to have that you have qualified that thing. So only based on human challenge, no vaccine has been approved. You have to go for limited phase, but it gives a fast track voucher. And that I think once the data is there, our regulator will take it positively.
So the human challenges studies being done abroad will be acceptable to Indian regulators?
Absolutely.
Okay.
Absolutely. They have to, they ask question and we have to answer the question. That's simple.
They are acceptable.
No, I know. This is one of the data, yeah. So I can discuss at the end of this session. But this is one of the research questions. So there are many other aspects which have to be taken together. Thank you. Yeah.
Thank you. Thank you, sir.
Thank you very much.
So now I have the honor to invite Dr. Veena Vaswani, Director for Ethics and Professor in Epidemics University, to give her presentation on the elements of preparedness for future pandemic, a toolkit for formulation of strategy. She's also a professor of forensic medicine and toxicology with additional qualifications in bioethics. So over to you, Dr. Veena.
Good morning. Can you see me? Like the virtual time has come back. So I'll be standing here and speaking. Okay. When I asked for water, I guess you must have thought, is she going to sing a song or something? But I have a dry voice and every time I start with drinking water. So thank you. Now, this is the preparedness for future epidemics. It's very challenging to be standing here before you all because this is almost everything that has been said. And the opportunity before me is I can always say this was covered in these lectures, but I will also be trying to focus from the other end, from the downstream, from what it looks from the ethics committee members and from what it looks from the perspective of the people who may be taking part, and then how do they respond to it. So elements of preparedness, it's good that we have taken such a big step, a moonshot, because whenever things go tough, the tough get going. So I'm sure that we are about to see something that's very good that's going to happen for in the preparedness for next pandemics. So this is something that we've already discussed, the 100-day challenge, and also with the 3.5 billion that's going to be invested, because 14 million to 24 million is a big number. And death actually brings in a lot of economic loss, leave alone the emotional and the break of family and all other things. In fact, from death itself, people say, is that a harm? And death has got a distinctive harm, because when you disrupt a person from the time when he's not expecting to die, and it causes a chaos in the family. And along with that comes the burdens of economy. So now I'm going to speak about the CHIM model for pandemic preparedness. And before that, I would like to say, and I have no conflict of interest. So one, can CHIM be effective model to accelerate vaccine development? And what are the challenges? And what an example of engaging communities, you could say engaging the ethics committee members, if you look at them as a community, and also we work with transgender people. So how have we engaged them and how have they responded? And a bit on the ethic and the epidemic perspectives when you look at vaccines. So up to very basics, ethics, ethos, which comes from also the word mores, models, from the Latin word custom or habit. So both are result of society's evolution towards a standard behavior. So once we come to a standard of behavior, we look at others and look at it like these are non-reformed people. And we at the other end of the spectrum in front of the computers are more refined and the others may not be. So that's one of the biases we need to keep in mind. Do we have this? And ethics consists of fundamental issues of practical decision-making, and the major concerns include how to improve life, how to do right from wrong, good from bad, and virtue from vice. So in bioethics, when we talk of, it is what is the study of ethics when it's applied on human being in every field that exists. And it is not just doctor and patient, nor science and the recipient. But how does any technology, how does any change in life, politics, society, social change impacts a particular problem? It investigates problems arising from medical and biological practices. It was born in USA as a result of an alarm, especially following the Second World War. And then there were ventilators, the iron lungs that were called. And then who would get it? Would only the person with money get it, or would everybody get it? So the question of, are doctors playing gods, deciding who are only going to be a part of it and who don't? So this resulted in one of the formation of the clinical ethics committees, which came slightly before. And then the research ethics committees also came as a result of the Nuremberg trials that happened. How can human beings use a fellow human beings for such experiments that resulted in their death and destruction? So now when we talk of the first in human studies, usually the initial way of looking at first in human from the perspective of ethics committees is a totally kind of, is it really required? But sometimes we need to take a step back to look at the bigger picture. What about the diseases which are newly emerging? And for those diseases, can we not have a method that is first in human, that needs to be studied? Can you just overlook it? Because once the risks are predictable, the subsequent trials, the risks can be much lower, but somebody has to start that process. But the precautionary principles should prevail in first in human studies. That means whenever you are having such a study, the most important thing is, does the participant know that this is an area of uncertainty? Does the participant know it may have some risks, could be substantial risks? And is the hospital prepared to give the rescue treatment or available necessary treatment that should be given? So ethical principles in research is to balance the risk and the benefit and make sure that the benefits outweigh the risk. But this is a very tricky question in a study that you are doing for the first time. What if the animal models, you cannot replicate such a disease in animals? You have a limited knowledge from the animal studies, but you're now trying to bring it to first in human. But still you could when you're taking it, but in which case, Christine Grady from NIH, in the article on research risks, if the participant is aware of the risk that they're going to face, or is told about the risk and is able to take the risk, then that may be there, you can enter them in the first in human trial. But all over the world, there is a lot of limitation in developing countries, that whether they really understand what they seem to understand. It's easy for us to say, okay, I understood when we are signing a bank statement, but we really don't read everything. We sign it because that's required. So to understand the understanding itself is the tall order that rests with the ethics committees. How did you gain their understanding? Because voluntariness is something so difficult to gain, because even I'm pressurized by somebody to do something, I'll say, no, I'm most willing to do it. But then of course, that's a voluntariness, but understanding is something we need to know that the participant has understood. And we need a more regulatory guidance in how to assess anticipated social value, because we think if there is a pandemic, in a situation where there is no other go, if we have to do it and the participants are ready for it, can we treat it as it's a social, there is a social value in the study. Secondly, the fact of participants thinking that it's going to be beneficial to them. In these days where CARDAs are supposed to have immunity, it's so difficult for participants or anybody to think they can take something, but it will not have a beneficial effect. It is just for the sake of understanding how the drug responds and how they respond to the body. So this is where a lot of engagement is required. Control human infection model studies, also the control human infection studies, because not all of them are models, or human challenge studies, they involve an intentional infection on a consenting human volunteer with a well-characterized attenuated strain of an organism under controlled condition. The organism is controlled, it's manufactured under good clinical practice, good manufacturing practices, and it is in a dose that is controlled, it is injected. So it's also called a human challenge study. So the traditional way is to wait for the patient to develop the condition, whereas in CHIM, you don't know how it responds, so you're trying to inject it. Could be to study the process, how the disease develops, or to study what can go well with it and what can be given as a treatment. Even a simple antibiotic resistance at a national level, like I come from Mangalore. Mangalore is pandemic for malaria. And in fact, we have done autopsies on children of 8-9 years, cerebral malaria, not resistant. So if there is such a thing, can it be said that the social value is exceeding and for which it should be permitted? And so CHIM reduces the time to bring the vaccine to market and reduces the mortality. So comparing the first in human with the CHIM studies, from the definition, the act of deliberately injecting a healthy human participant in order to study the disease or find a cure or a vaccine. It is the context that is driving the need. So here, I would like to say that in 2018, I was one of the totally no for CHIM study. I was also a part of the ethics committees, and I used to look at it with shocked value. How can you inject on somebody and then bring about a disease? Is it that non-maleficent? I mean, isn't that maleficence? That doing something that directly harms? But over a period of time, when I got the Global Forum on Bioethics Research Fellowship to Johns Hopkins, and I've interviewed people in order to bring out a white paper, I realized that when there is no other thing existing, isn't that opportunity cost lost for research? Should the research not be in that direction so that you at least make most of it if you can try to help people with that? So if the research addresses this disease and develops a cure for a preventive strategy, the opportunity to treat a patient or unmet need is the opportunity cost. And this would be lost not doing a CHIM study. But it requires to be communicating and to engaging with the stakeholders. Now, when I say there is a need, but I'm not very sure that the need for community engagement is done. We have yet to do it. Now, THSTI took a leadership in 2019 onwards, because I attended a meeting with WHO consultancy, this thing in Chicago, and then I knew that there was India was trying to have a meeting with the CHIM study and the consultants. So I was called upon it in 2019, before COVID, and even during COVID and later. There was the discussion on, can we understand the research ecosystem in India for a CHIM study? So the 6 medical college hospitals were selected. And then of these 6 colleges, the ethics committees, it was a protocol that we wrote. We made little changes in the local setting, and it went through ethical clearance. And then the study involved, the study was understanding perception about controlled human infection model studies among members of ethics committees of Indian medical institution. This was a qualitative exploration. I would like to say any amount of science does not complete itself into the deliverable form unless you have a qualitative exploration as to what makes it acceptable to them. Please go back, if you remember, the Alma Ata Declaration in 1978. It was meant for accessing, for a better access of primary health care for everybody. It was a wonderful WHO initiative. It was very well thought of, but it didn't work much in India. Why? Because the aspirations of the people were not taken into consideration. Just making 4-5 rooms and calling it PHC, PHU without an access, without the way, the care that was only modern medicine, and their own aspirations of other systems were not included into it. So having a plan is very good, but a deliverable plan should be slightly different from just having a plan of production. Otherwise, you would have countries holding up, you would have people holding up, you'll still have Africa getting 25% of vaccination, you would still have women in different countries not accessing vaccines or a partial vaccine. So I'd like to say that this is where we look at the study. And from the 6 institutions, 11 focus group discussions were held and 31 in-depth interviews, and total ethics committee members from different institutions were 72. And then, it's published in the Wellcome Trust open this thing. So the engagement with the ethics committee members brought out a huge concern about the safety. Initially, they were anxious, but then as the COVID was progressing, when they had we changed from in-depth interview to focus group to in-depth, they were saying if this is the only way, we have to go for it. Uncertainty and risk associated with study, risk of spread of infection to the third party. Here again, this is one risk that may not be seen in every first in human studies. Whenever you have injected a person, he could be infecting others. So if the person wants to go against medical advice, it becomes a difficult thing. And the monetary inducement, because they're going to be with you for some days, this has always been a kind of equilibrium that is disturbed when we talk of compensation or monetary, the exploitation, what causes what. So the exploitation of the vulnerable population was in the picture. Whereas the engagement also brought about the worry of the ethics committee members on stigma discrimination. Is it going to, because is the family going to separate them, saying that you're going to infect even beyond the source when they've been infecting? Lack of rescue treatment, because in those days when there wasn't much of a treatment available, if you start doing a human challenge study and should adverse event occur, you don't have a rescue treatment. So can you do that? So engagement with ethics committee members brought about a lot of values that they held closer. What about insurance? How long does it pay for? Should it also pay for the family members if they infect the others? And these were the other questions that came with it. This is where I'd like to talk about the ethic and the emic perspective. If there is a tree out here, a huge, nice tree, it's coming in the way of the traffic. We would like to cut it because then you can make a super highway. But then if there are a community staying there, they're worshiping this tree. For them, the tree means everything, including giving fertility. How do they look at the action? So ethic perspective is when we look at from an outsider's perspective into people, into community, into lives. Emic perspective, staying within the community, how I look at inventions, how I look at vaccines, how I look at whether government is tied to them, whether I think it's only an election mimic or whatever. So now who will talk to the community? And it is always without this community engagement that we may not see the purpose what we are doing, because it's like the doctor may be a gold medalist or whatever. It's not worth it unless he's able to hold the hand of the patient and tell him, Baba, I'm here for you. Let's do what is best for you. Because that connect between the community should not be lost. So our experience with transgender people told us that no, we didn't get vaccines because we didn't have Aadhaar card. We changed our gender. The new Aadhaar card showed us as females. They were saying that I'm not the same person. So no card. And so some of them who got were only taking it because they were saying, if you're not, you'll have to evict this place. But because they had comorbid conditions, diabetes, HIV, hypertension, this was never addressed. So many of them went underground and didn't take the vaccine. So there wasn't a level of consultation that was going on with them, or nobody even told them why, what, when, how. So that is one of the reasons. So besides vaccine, then what else should be the priority? If we make a lot of, vaccines up to whatever the challenge is required, does that translate into actually what happens? What about the racial gender inequity? Even in India, the consumption of vaccine by the females lagged very much at the end of the first vaccination, because the ladies didn't know how to access, didn't go, and they did not have the opportunity because they felt not so important. They were disproportionately hit by the pandemic because all the men had mortality, the ladies were burdened.
Conclude fast. Yeah.
And also about the dead body management, because I'm from forensic, and we knew the way the dead bodies piled up and nobody was claiming them. And we didn't have guidelines what to do with them in the first few days. There was the first case which dropped the dead body for 30 meters down, and with a cement concrete written, this is positive for COVID. So this is how there was a lack. What we need to, I can say in short, this is a lesson learned from Ebola, but if we have no time, I will skip through. And I'd like to go to the concluding slide, that strengthening our defense against COVID-19 and reducing risk of future pandemic is right. It's true, but we have to learn lessons from Nipah, Lassa, and other Lassa fever, because we haven't yet learned. The first, managing information and not letting the WhatsApp university take over is the biggest challenge, because they break groups, they break communities, and they break the scientific ethos. So towards the end, I would like to say that engagement is a long drawn process. Media hype and undue publicity, it may bring therapeutic misconception, and immediate lines of communication have to be established, and equal risk benefit should be spoken when we are a part of the patient participant into it. And in the end, a good science is science in which knowledge and ethics are held to the highest standards. For this, ethics must live as much as innovation. So thank you very much.
Thank you very much, Veena-ji. You have brought out the issues, but also showing how to negotiate behind the concerns. And it's so heartening to know that you have been part of that group, which is trying to establish human challenge facilities in the country. Question, please. Yes, sir.
That was a very thought-provoking presentation. Thank you for that. This is not a question as such, it's a suggestion. And a suggestion for all members, including the managing members and all government organizations. During such important platforms in future, I think biomedical engineering and a lot of neural and other technologies that are emerging in the simulation aspect. We have very detailed simulation models for manufacturing. Lab on chip models are at a nascent stage, but if on such science platforms of biologicals and vaccines, and we also start inviting the IITs and the technology institutes who are working on simulations, lab on chips, which is the future. 20 years down the line, you'd be able to do in vivo in certain, which you're already starting to do. They are going to address a significant aspect of the ethics that have been brought out. It's a suggestion that such platforms we have, even the technology and engineering and the biomedical research, man-machine interaction people also available on such platforms for regular interactions.
Yeah. Very valuable information that this collaboration with cross sectoral collaboration is very important. And Dr. Rao is witness to that, that unless different sectors are brought in, things either don't move or move very slowly. So thank you very much, Veena. Thank you very much.
Thank you, sir.
Thank you, Dr. Veena. So I would now invite Dr. Nivedita Gupta, Senior Scientist and in charge of the Epidemic Communication Division of ICMR. So she needs no introduction, like she is the key person to formulate the validation protocols for the COVID-19 diagnostics, conduct validations, and commission multiple testing platforms like RT-PCR, TrueNat, and CBNat for increasing the outreach of testing till district level. Welcome, Dr. Nivedita, for your deliberation in the topic, facilities for clinical trials in India, need for integration. Ma'am, over to you.
Thank you, chairs, and a very good morning to all the esteemed experts and all the colleagues in the audience. So the purpose of my talk today is to walk you through the facilities for clinical trials and various supporting activities that are available in India But as per me, they are fragmented right now. So if we bring them together, I think we can deliver quite a bit. So I'll just give you a glimpse of what we have and what we need to integrate. So if we look at what we did in the COVID-19 vaccine development, sitting in the government sector, I think it was an exemplary effort both from government as well as industry and all our stakeholders and academicians. So India moved on 2 tracks as far as COVID-19 vaccine development was concerned. One was leveraging on our own indigenous R&D, right from product development to large clinical trials, and then taking care of our in-country supplies and even vaccine diplomacy. And Covaxin and Zycov-D are burning examples to that. The second was leveraging on international science and technology, because India being a huge country, we always need a mix and match of several approaches. And this approach also yielded a very fruitful outcome in terms of Covishield, Covovax, Corbevax, and Sputnik, and definitely many more in line now with the mRNA vaccine also. If we look at how the government changed their strategy in terms of providing critical and timely support to the industry, it started right from funding, assisting in preclinical studies, bringing in facilities to work on preclinical trials, then clinical trials, then a lot of laboratories chipped in, both from ICMR, DBT, and many other laboratories in the country who could help. Then epidemiological studies were conducted by all the relevant SNT institutions. DCGI also came up with a rolling regulatory review, which was quite innovative. And the manufacturers were allowed for advanced purchase orders, at-risk manufacturing. Then also government aided in raw material import and expedited batch testing and release. Though I understand the woes of the vaccine industry, but still efforts have been taken from the government. But I acknowledge that a lot has to be done from the side of the government as well. Now, moving forward, what we have built during the COVID-19 pandemic, I strongly feel that this is time to integrate and not lose on the capacities which have been developed after so much of hard work and intense efforts of all the stakeholders. Now, in India, if we look at the opportunities for vaccination, our routine immunization program is the strongest.
Your slides are not moving. Is it the same slide you're working?
No, I've actually moved on to.
IT, please help.
Okay, I'll change from this, sir.
Okay.
So I'm on this slide. If we look at the routine immunization program in India, it has been significantly strengthened over the past several years and caters to one of the largest birth cohorts of 25 million children a year. But beyond routine immunization, what are the opportunities for vaccination in India beyond 5 years? Only ANC visits in pregnant women, and then Japanese encephalitis vaccine, which has limited rollout in states like Assam and West Bengal. So what I'm trying to say is we never had a robust adult immunization platform in India. And with COVID-19, we have really been able to create that. And thanks to COVID and all the efforts of the Ministry of Health and Family Welfare and all relevant stakeholders, we are now well positioned to roll out adult vaccines in India. And attempts are underway to deploy COVID for routine immunization. And also we should strongly leverage this platform for introduction of other vaccines of public health priority, be it influenza, HPV, and other vaccines and boosters, as we are seeing re-emergence of diseases like diphtheria, etc. I think this platform should be used and our momentum should be maintained. So in a recent prioritization exercise for diseases of public health importance, which NCDC carried out with all the stakeholders, there are certain pathogens which are prioritized from One Health perspective. And if you look at the list, scrub typhus, Crimean Congo hemorrhagic fever, leptospirosis, anthrax, brucellosis, plague, and dengue are the prioritized pathogens for which we do not have any vaccines available. So we do understand, as per our priority score, that we do have a threat of major epidemics from these pathogens for which we have no vaccines available. We also need to look into vaccines or better vaccines for diseases targeted for elimination, particularly tuberculosis, malaria, and lymphatic filariasis. And definitely, as Dr. Sanjay Singh very beautifully showed, plug and play vaccine platforms needed for preventing unforeseen public health emergencies, and mRNA platform happens to be one of them. So we really need to work on all these areas with a very dynamic vaccine introduction platform now. So what are our existing capacities for aiding in this kind of a vaccine development, expedited vaccine development, 100-day vaccine development? We do have sites to conduct and support epidemiological studies, be them disease transmission, burden, or impact studies, which all of us do understand are very critical for understanding the need for any vaccine in any kind of geographic setting. Then to conduct preclinical studies, to conduct clinical trials, all phases, then to conduct vaccine efficacy studies, model disease burden, and also maintain disease registries to understand the impact of various vaccine programs. So we do have facilities which are funded by various organizations, including Ministry of Health, ICMR, DHR, as well as DBT. Now, this is a glimpse of Health Demographic System Surveillance Network, HDSS network, as we call it, spread across India. This is a very rich resource that we have, 25 sites spread across the country, which are maintained or funded by ICMR, DBT, as well as other global sources. But if you look at this, we have these sites where we can do epidemiological studies, disease burden estimation, disease transmission studies, and also study impact, because we have a very well characterized denominator where we are going to study any effect. But right now, all these sites are fragmented in terms of they are struggling for staff retention. There is a problem with data management, no uniform SOPs for data entry and management, and also issues related to funding. So this is something, even as ICMR and other stakeholders, we need to understand that we really need to pull up this network, support this network, and sustain this rich resource which we have in our country. There are also several GCLP laboratories which have been strengthened by particularly DBT, I would say in this case, which is also a tremendous rich resource that we have. Now, these laboratories, we really need to ensure as government agencies that there is sustained funding. Our staff needs to be continuously trained and undergoes capacity building activities. We need to have access to international standards and various protocols, and here the role of international collaborations really comes through. And then we also need to establish effective linkages with the industry. These are the large animal testing facilities which the country has invested in. As I'm not even mentioning the small animal facilities, which are many, but if we talk about large animal facilities, government has prudently invested in facilities in the city of Hyderabad. Like if you see, ICMR itself has two large animal facilities at National Institute of Nutrition, Hyderabad. Then we have recently come up with an institute known as NARF Hyderabad, where we have an array of animals, right from mice to horses and everything that you can think of. And then DBT facility again at Hyderabad. Then we also have ICMR NIV. Then in Mumbai, we have a facility at our another institute, Reproductive Research Institute. And recently ICMR has established a center of excellence at Vizag. So these are the animal facilities which are available. I think if we have established effective linkages of these facilities with our industry, then they really sometimes spoil a lot of time going outside the country and getting their studies done. So with these linkages and well-oiled machinery and approval processes, and then a very trained and I think, oriented staff, we can really make these facilities available to the industry and fast track the vaccine development process. ICMR has recently launched an Indian clinical trial and education network, which is spread across 47 sites in India. And across all zones, if you can see the representativeness of this network, it is known as INTENT. And the vision is only of this network is to provide evidence-based, cost-effective, scientifically sound, and culturally sensitive solutions to problems of public health importance. And there is a lot of effort going on through this network, wherein we have a seed funding to these 47 sites going through all the year, and they are all the time ready to undertake clinical trials linked with medical colleges, as well as the clinical trial sites, which may be their community sites or HDSS sites. And we are working with various stakeholders, be it CSIR, DBT, Ayush, and many other ministries, as you can see in this diagram, where we are trying to bring out the ideas for promising leads where we can take them for clinical trials. So this is also a rich resource which can be tapped for doing any kinds of clinical trials. Then before COVID, we only had one vaccine testing facility, which was there at CDL Kasoli. But at the time of COVID, the government has invested in bringing up quite a few more, like the National Institute of Biologicals, Noida, now is the National Control Laboratory for 5 vaccines and is aiming to be there for 11 other vaccines in pipeline. Then DBT also had 2 of their laboratories at Hyderabad and Pune coming in as national control laboratories for COVID-19. I think the scope of these laboratories can be further expanded to vaccines of public health importance to expedite the batch testing and release process, because CDL Kasoli has its limitations because of being the only facility as well as its location. And these are our vaccine development capacity over the past years, what we have been able to demonstrate in terms of complete indigenous development. We have 6 kinds of platforms: inactivated, live attenuated, polysaccharide and conjugate vaccines, recombinant vaccines, DNA vaccines, and now RNA vaccines. And we do have an established capacity for developing combination vaccines and also taking in technology and upscaling it. But definitely a lot of capacity enhancement is needed for research and development for viral and non-viral vector-based vaccines. mRNA vaccines, like Genova has taken the lead, but we need many more mRNA vaccines and many more industry partners chipping in to take this promising platform forward. Then protein subunit vaccines, novel adjuvants and fusion proteins are our weaknesses. We need to work to remove these weaknesses as well. These are our identified priority areas on how to achieve more in the area of R&D for the vaccines. I will not read each of them in view of paucity of time. And over the years, ICMR has also systematically invested in developing quite a few platforms which are available for use for disease estimation, as well as modeling and doing clinical trials, of which we have 146 virus research and diagnostic labs, 30 model rural health research units. The beauty of these units is they are located in rural areas and have a defined catchment population, where we can do a lot of studies to understand the rural distribution of diseases. And a rich BSL-3, BSL-4 network of 18 labs, of which 10 are operational and the others are in the process of becoming operational. And thanks to INSA-COG, we had a lot of upscaling in our genomic sequencing capacity with a network of 52 sequencing labs also now available. And also to expedite the testing as well as virus isolation in terms of huge epidemics and pandemics, we recently came up with a mobile BSL-3 laboratory, which was first of its kind in Asia. And with this kind of facility, we can make testing available at the point where the epidemic has happened, and also isolate the virus and also develop assays later on. So quick isolation, quick laboratory-based testing is the objective of this network. Now, this is my last slide where I sincerely urge all the stakeholders who are involved in the activities related to vaccine development and clinical trials that it is very important to leverage the capacities that we have developed during the COVID-19 pandemic, not lose them at all. In fact, synergize all the existing capacities to improve our outputs. And of course, with all this dynamic milieu of vaccine development, we need a very dynamic regulatory milieu as well, because in many instances, as has been discussed since morning, we do have problem in conducting efficacy trials, especially in diseases which do come and go in spurts. For example, Nipah, Zika, we have no idea how to do an efficacy trial in such cases. So we really need regulatory guidelines to look into the challenges which are posed by such pathogens and advise us on how to bring these vaccines to licensure. And definitely the vaccine development for diseases which have been prioritized by India under One Health umbrella and those targeted for elimination, we need to prioritize development of vaccines for those infectious diseases and take them to a logical end. And last but not the least, sustained funding is actually pivotal to everything that we do, and preservation of resources, including trained manpower. So I would stop here and thank you very much for your attention.
Thank you very much, Nivedita, for that is very encouraging and also makes us proud that in last 3 years, a lot of augmentation of facilities have occurred in the country, and ICMR, DVT have taken lead on these issues. We are open to a few questions. Dr. Sanjay Singh.
Thanks, Nivedita. So for phase 3 is a difficult proposition. For example, if we are going to prepare for phase 3 for dengue, we might have to wait for the season. But I think working with ICMR, that before season comes, we can select the sites and integrate these things. I want your views, what you say.
Absolutely. And besides what we have been observing in dengue, definitely there is a spurt in disease as per the season, but a certain baseline of cases we are now seeing throughout the year. So there is never a dengue-free season, but definitely the lull period goes in preparing the sites, and we launch the trial just before the start of the epidemic season so that we are well oiled to capture the cases when the season is there.
Thank you.
Yes, Professor Ramarao.
Are there any networks like clinical trials networks that is something like that is established in the US? Do we have something in India organized by run by the either ICMR or DVT?
Yeah, so that's what I showed in one of my slides, that ICMR has very recently launched a network which is known as Intent. And this is a network, clinical trial network of 47 sites. And we will give them a seed funding which will be available throughout the year, of course, with trained manpower, capacities, and continuous training and hand-holding, and also giving them clinical trials of promising leads which emerge through various deliberations with the ministries and departments that I showed in my slides. So that's been already launched by the Department of Health Research and ICMR.
And then is there a process by which people approach these networks to push forward with their specific products?
So right now we have consulted various ministries and departments, but definitely if there are individuals who are interested, then there is a secretariat at ICMR and anyone can approach. Even we are entering into a lot of international collaborations now. So there is a full-fledged secretariat and a unit which is handling this now.
Okay.
Thank you. Yeah, please go ahead.
Dr. Nivedita, you told about the animal facilities, like 5, 6 of them. Is there any plan by ICMR onto the NHP facilities under containment? Because that has been one of the limitations of the infectious agents.
Yes, so when we talk of NHP facility under containment, then the BSL-4 lab of National Institute of Virology is very well equipped to do that. And that's how we did the Covaxin trials, the challenge studies there. So we do have a facility, and wherever we have an animal facility, we are coming up with a BSL-3 plus facility so that we are able to do primate studies under containment. But BSL-4 option definitely remains there all the time.
Okay, thank you.
Yes, sir.
So these animal facilities and some of the other facilities, what is the model? Do they operate on a fee for service, or do they seek intellectual property when a study is done? How does it work?
So right now, if I talk about ICMR particularly, there is now the guidelines whether we will operate on a fee for service mode or on a IP collaboration mode, they are still under development. But definitely now it is being dealt on a case-to-case basis. Like some of the vaccine manufacturers come to ICMR when they are not only seeking NHP studies. They want a long-term collaboration in terms of doing NHP, then doing clinical trials, doing the laboratory assays, then it becomes a shared IP. But if it is only a one-time thing that they only need a primate study, then definitely fee-for-service model works better.
Thank you.
Hello. For a nice talk, my question is, what are the initiatives by the government for preservation of the trained manpower, and how you are addressing this particular concern?
Yeah, so trained manpower, what we are trying to do is like raise this to various senior authorities and make them understand that we need to have sustained funding. Like we can't create positions for them, permanent positions. That's, you know, how challenging it is. Wherever opportunities are, they can apply and get absorbed. But of course, sustained funding so that there is no attrition, and a little flexible guidelines for specific situations would help, because our project guidelines would say that after 5 years, you can't continue. So those kind of initiatives are underway, under discussion.
Okay. Yes, Shailja.
And Nivedita, thank you for the wonderful talk. ICMR has so much of data, and I think most people would like to know if they can access the data, and how can they use the data. That is one question which everybody would wants to know.
So ICMR data, you are talking generally of any data? Yeah. So the ICMR data, as far as the infectious disease data, particularly testing, if I give you an example of virus research and diagnostic laboratories, we generate data and NCDC also generates data. So far we have been struggling to put that data into the IHIP portal so that it can be viewed by all the policy makers, though we send weekly bulletins to all the relevant stakeholders. But then there is a fear of duplication of data because NCDC also collects it and we also collect it. But very recently we have entered into an understanding with NCDC that wherever our VRDLs are located, they will block the entry into IHIP, and they will take the entire data from ICMR through our central portal. So then there is no duplication of data, and entire data of the VRDLs gets reflected in the in the IHIP portal. And similarly for COVID-19, there was a lot of data cleaning efforts that were required with especially COVID, because we had a very synergized system with the Ministry of Health and Family Welfare in terms of capturing deaths and hospital outcomes. But then we could not match the vaccination records. But recently, with the intervention of our new DG, there is a lot of effort ongoing to match the vaccination data with the testing data and see the effectiveness of vaccines and do all the calculations. So there's a series of meetings going on at ICMR with all these stakeholders, after which definitely this data can be made public. But it's not yet very clean and cannot be matched. That's why the limitation was there and it was not put in public domain, but I think it will soon be there. And also our registries are available in terms of wherever we collect data. There is a cancer registry, diabetes registry, and they are very much accessible. Yeah.
So thank you very much, Nivedita. It is very encouraging. So if we have to achieve this CEPI 100-day challenge, I think a lot of groundwork has been done by the country. Thank you very much.
Thank you. And I would like to thank Dr. Shelja and all the organizers for giving me the opportunity. Thank you.
So thank you, Dr. Nivedita, for the beautiful talks. And then I would invite, we have two more speakers here. Dr. Vijay Kumar Chaudhary, he's a professor, served as a professor in the Department of Biochemistry at University of Delhi, and is currently the NACI Senior Scientist at Center for Innovations in Infectious Disease Research, Education and Training, University of Delhi. So Dr. Vijay, for Dr. Chaudhary, you're requested to deliver the presentation on can utilizing antibodies via stopgap alternate till vaccines. Over to you, sir.
Okay, I think good noon or whatever. In fact, me and Amita are grateful to the organizers for inviting us, and also more thanks that they accommodated our brief discussion, because it is a misfit in this session. I'm not going to talk about regulatory, but just to make it catchy, last night we changed the topic to can neutralizing antibodies be a stopgap arrangement till vaccine. This is what I learned during the COVID, that there is a possibility that if long-lasting with long half-life antibodies could be produced, then till the vaccine is available, they could prevent, if not the general public, at least the medical practitioners. It is like teaching class. Most of us understand the antibodies, but what is important to show here is that it is the Fab small fragments or single chain FV fragments which are used. In fact, in this talk, I'm going to tell you about our efforts in making phage display library, a naive library, which has been a resource of isolating antibodies in a finite time. And that is what is the basis that it could be used for creating antibodies, for isolating antibodies or generating antibodies against any infection which comes across. The antibody science for therapeutics started with targeting the tumors. And as soon as, not as soon as, when the monoclonal antibodies, the mouse monoclonal antibodies became available, people started thinking of therapeutics. However, in the first instance of using mouse antibodies did not succeed. And the reason was the efficacy was much less than the anti-mouse response. So that necessitated the conversion of mouse antibody into human-like antibodies, and that was done through engineering in phases. And finally, today the technology exists where you can get the fully human antibodies. Again, revenues, money play a very important role. We all say that funding has to be available, but we have to create the molecules. So in molecules, as a basic scientist, we think that if we have isolated an antibody, we have achieved breathing. I think listening to the talk today, I mean, I knew it before, and we others, younger people should understand creating a lead molecule is one part of life, but taking it to the people is totally different science, and you have to face a lot of challenges, but we have to be ready for it. So at least if we don't do it, companies will do it, but we should be knowledgeable so that we can appreciate the efforts of those industry people who do that job. We may discover, but they are the who make our discovery successful. And here is a list to encourage you to work in antibody field, that a single molecule, I don't know, I mean, sells for $20 billion. Well, how do we get antibodies? There are at least for human antibodies, I mean, there are at least 3 platforms available. One is that you can take the cells from the convalescent patient. And for this in India, at least several labs have perfected this technology for isolation of antibodies in COVID. Recently, they have published, and also for HIV. Another is the humanized mice, and those are highly proprietary. So they can be used for isolating human antibodies. But then there are technologies which is based on fast display technology, which uses either the synthetic antibody genes, or another thing is naive human antibody library. So here in this lecture for next 8 to 10 minutes, I'll be telling you about our efforts in making of a very, very large human antibody library, which has been successfully used for isolating molecules which are important. Phase display technology was important, and especially for the discovery of the antibodies, and the two scientists who, George Smith, who described for the first time in 1985 what is fast display technology. At that time, I remember I was attending conference, people did not understand what is the technology, but then together many people, and especially the Greg Winter, shows the part that this is important technology and antibodies can be isolated, and they were of course awarded the Nobel Prize. This technology involves creating a library of antibody genes and cloning into the phage, fused to one of the phage protein as shown at the top. And here the antibody, the fragment, the single chain Fe antibody, the scFv, is displayed on the surface while its gene is inside the phage particle. Let me see there. Okay. The gene is here. And if you create a library, so if you have, 1 million VL and 1 million VH, you can make artificial combination to get a very, very large library. And that is what many others have done. And we also were able to make a library of 10 billion clones. And as was listed in the first slide, all this work was initiated many years ago in collaboration with Genova, and Sanjay is here. So then you go through a process of panning of these 10 billion clones, and through a process, you get about few thousand clones which are highly specific to your target. And here is the science and the technology involved, which we perfected and at South Campus. And I'll show you with the examples. So here it is that we took the RNA from the naive people, constructed the library, again by a very high proprietary methods, which yielded a patent. And then we go through the panning. And what you get in the end is individual phages carrying some antibody, say binding to receptor binding domain of the SARS virus. But those antibodies in this form are not useful. Therefore, they have to be converted into this full antibody. So we developed, this slide describes the details of the library that everybody says that our things are better, but we think that it is pretty good. And it is reflected with the collaboration which was successful in isolating antibodies, which worked for Genova, and the clones were transferred. Then, because we belong to the university academic institution, we have to continue doing, people do PhD, but then they do work which is relatively meaningful in the end. So then we were optimizing the processes of panning. And our goal was actually, when we started, the goal was that we should have a platform, we should be able to discover the antibodies for the whole proteome of an organism. Okay. So I'll not go to detail. That's a talk for some other time. What is most important is our antibody library is very, very large, and 10 billion clones, of which 7 billion clones are in frame. That means out of 7 billion clone, you will find 1, 2, 5, 10, 100 antibodies which are to your targets. And again, for example, just to illustrate this, that antigen 85 and antigen 85B of mycobacterial proteins are very similar in sequence. Out of 230 amino acids, only 10 are different. But yet, through this library, we are able to isolate antibodies which bind to A, which bind to B, and which bind to both. That means so much diversity. So I as a biochemist don't see antibodies as immune response. They are already there in the body. So you have to make a library and select for it. So actually, what I would say in the end is you don't need immunization. So this becomes a immunization-free technology. Again, as I said, SCFVs are often cannot be used, so they have to be converted into mammalian expression system. So we optimized an XPCHO-based system to produce hundreds of milligrams of antibody per liter in laboratory setup. But antibodies will be good only if they bind tightly to the target. So from our library, we are able to get antibodies in single-digit nanomolar KDs, which is supposed to be good. And if you require better antibodies, they can be always improved by affinity maturation. Again, because we had the technology, a group led by Professor Kinney from National University of Singapore, they had a very peptide, which was thrombolytic peptide, and it was highly potent. So he wanted that whether we can have a blocking antibody, neutralizing antibody to neutralize its effect. So we were able to isolate from the same library these antibodies, and which are in nanomolar. And in fact, this paper came out in Nature Communication a few months ago. So there are at least, I mean, this is an older slide, about 2-3-year-old slide. And I compared the properties of the various libraries available in the from the various companies, academic, there may be some. There are two properties which are very important for library, whether it is how large it is. So most of the libraries are about 10 to 100 billion. But are they diverse? That means, do they have unique sequences? And then when we go for the selection, do they select the antibody which are repetitive, or do you find? So I will not go into detail to explain, but our library to us and to others is scored pretty good in that measure, that we have a large library and you can get antibodies to different targets without much difficulty. Without much difficulty means you have to have the expertise. Based on this background and also many other isolation related to snake venom antibodies and others, we were, as Sanjay said, we were asked by Dr. Renu Sarup that we should get involved in the COVID antibody while we were working on the snake venom antibody. And we did get involved, made a large program, and then we got handsomely funded by DBT and BIRAC. We isolated the antibody. This is an older slide. Eventually, because the virus was changing very often, we were able to get the antibody even to Omicron. And as I speak today, they have been tested at TSTI for their efficacy. Another very important target could be the anti-snake venom, because there you cannot get the convalescent horses. You cannot get immunized people. Okay. So here the naive library could be very useful. And again, I'll not show you the data. We have been successful in getting antibodies to neutralize Nasa venom. And then our antibodies are so good that we were, like, for example, the short toxin, 3-finger toxin, have 19 sub variants. With one antibody, we were able to pull down 4 of them. So we are hoping that with a mixture of 4 to 5 antibodies, such antibodies, we'll be able to completely neutralize Nasa venom. And so that will change the whole system of century-old technology. But time is essence. It will take time. We have the technology. So, I think last 2 slides. So our effort is that we have shown that our platform can be used for isolating antibodies for totally different targets, and they've been shown in efficacy. Again, it is missing. Can we use this technology or others which are available for getting antibodies against infectious organisms such as Nipah, Zika? So idea is that, not our idea, I mean, we can do only so much, that with such resources, can we tackle those new infections we are talking about? So immunization-free antibody technology offers avenues to discover antibodies against any target without to worry about whether patients are there or not. Okay, and then for this we have shown if we have a team of 5 experts, not hired for this job, they must know their job. In 100 days, antibodies will be available. But it is very important. But 100 days can be available, but not in India, because 100% of the reagents. Because yesterday there was talk, Professor Vijayaraghavan mentioned about 100 days is 100 days. Those 100 days gets postponed to about 200 days because the synthetic genes have to be imported. 100% of the media has to be imported, and that is the reason. So I end here. Thank you.
Thank you very much, Vijay. And again, so you are saying that your last sentence that in India it cannot be done.
No. I said 200 days because of import.
No, I think this point is coming up repeatedly since yesterday, that backward integration and getting the reagents right here is an important component of our preparation. We are open to, yes, sir.
My question is,
yes.
What about if we simply take, like B cells from convalescent patients and either stimulate with, say, EBV, or just fuse like with myeloma, like we make hybridomas?
Yeah.
So that may be a very fast source. So probably you may generate antibodies, say within 15 days, 50 days, isn't it, of 200 days?
So actually, what I believe, I'm not a human antibody expert, but what I believe those technologies are called, version 1, EBV transformation or other kind of fusion. Those who are expert in sorting the cells, the convalescent, Jayanta is maybe here, and others, they can isolate the B cells within probably 3 days. If they have the antigen, there are many if available, if the antigen has to be available of the right kind, probably 3 days, 4 days, 5 days, and clone the gene VL and VH. Again, the synthetic VL, VH is required. You go to Singapore or Germany, okay, they get it and put it in the vector. And then in 3, 4, 5 weeks, the antibodies are churning out. So those becomes EBV transformed. They have been all replaced by this cloning of the gene from the same convalescent patient. So it is those are very robust techniques, and that's how people were able to make COVID antibodies abroad in probably 2 to 3 months' time and publish a paper.
Any other? Yeah, yes, ma'am.
Yes, sir. Thank you for that very exciting talk. And this is more of a comment and a request that while working in public health, we keep on struggling with the competent pool in India who's available to make monoclonal antibodies. They actually know the process and the know-how. So would you be able to train some people in public health institutions, because sometimes they really have access to those critical samples which cannot be transported here and there. So having a trained mass of people who know how to raise monoclonals would be really critical for the country. I feel we really have a huge gap here.
Yeah. So I don't know. So probably you are talking about the mouse monoclonals. Okay. So, I mean, we made hundreds of hybridoma, but we don't make any more because it has become because of many other reasons. And then we have moved on to the human antibodies. So I think, at present, we may not be the right person to train people for mouse hybridoma. But in fact, during COVID in June 2020, or before that, we had written a very large program on anti-snake venom as a mission next generation treatment for snake bite, and DBT Apex Council or whatever was kind enough to approve the project for 56 crore. And it on the instance of Dr. Renu Sarup, when I was making the initial presentation, she set up a very special committee. He posed the same question that, Vijay, why you want to keep this technology to yourself? You and Amita and few others will know. Why not to spread this? And she insisted, and then she included a special vertical in the whole mission to start a facilitation center, not about the mouse monoclonal, but about the human monoclonal. And that is, but because we took up the challenge for the COVID, the whole plan has become flat, but we have not taken that initiative further. But you are right. And at some other time, I can tell that we do a lot of these training activities under the Center for Innovation in Infectious Disease Training. And we love to train younger people, and we do train almost 100 to 150 people in different technology, which are low end, medium end, and high end. But
I would request you to disseminate the information widely whenever the training.
So we'll send a link to our website to all supporting. Sure.
So it'll be nice if, for example, ICMR can chip in over here and arrange some workshop, like a small workshop.
Absolutely.
And there, many people from the country can come where you can train them.
Yes, yes. We love, actually, I love teaching.
Both since you are a very good teacher, both.
Amita and everybody, they have to take the torch forward.
Both humanized antibodies as well as human antibodies.
Absolutely. Yes. We'll definitely. Yes, sir.
Yes, Professor Ramarao.
Yes, sir.
I need a clarification. So are you using one standard library to screen against multiple targets?
Yes.
Are you making a library from each?
So we don't make library, but our technology, which is, why use word covered patent? Because to protect our national wealth, I know how to protect by filing. So this is a naive library of 10 billion independent clone. Okay, and it was made by using very special kind of B cell because Indian B cells were not working. So Sanjay spent some $50,000 to get those cells. So we made the library, and then truly it has got 7 billion independent antibody clones. So therefore, if this library is able to generate hundreds of clones for Genova's target, it is able to generate antibodies, same antibody library for the mycobacterial antigens, dozens to the COVID antigen and to the snake venom and to the peptide. So the same library is useful for everything. Actually.
So to generate these high affinity antibodies, you need the maturation of the antibody,
yes.
Right? How do you control for that?
So our library in practice, as I couldn't have explained the data. Most of the time it is generating antibodies, which not in our hand only, in others' hand, at Singapore and other, is about 1 nanomolar. So if you want to make an antibody for cancer therapeutics, so most of the COVID antibodies which were in the market, tested nicely as published, are between 3 to 5 nanomolar, okay, on a monomeric RBD. In our hand, all our antibodies are in the range of 1 to 5 nanomolar. So then if you want very high affinity antibody, you have to go through a process of affinity maturation, either through mammalian display or through phage display. So that becomes a longer process. But most of the antibodies which were used in COVID by different agencies, they were in the range of around 3 to 5 nanomolar, or maybe 1 nanomolar. But all the cancer antibodies which are used, which are like sub-nanomolar. So you are right, we cannot, with this library, and again, it will depend upon, suppose with our technology, if we start working on a new library of 100 billion clones, then you may get antibodies which are sub-nanomolar. But I think the better would be, as we understand, to get a 1 nanomolar antibody, which will do specificity and try to improve it.
I'm sorry, here.
Yeah.
I didn't quite understand what you said, what you meant when you
Yeah.
I didn't quite understand what you meant when you said the Indian cells didn't work. What does that
Indian?
mean? Cells didn't work?
Indian cells didn't.
The naive library.
Sorry. So actually, we struggled for almost 1.5 to 2 years to make the library. And we used to collect people from so-called healthy, like me or younger people, okay, with due ethics, Okay, but all of us have been unknowingly being exposed to viruses, or whatever, and whatever antibody library we produced. So what happens when we say naive means when we sequence the VL and VH of individual clones in our library, their sequences should be very close to the germline. They are all known. Okay, but what we found was the sequences of that library, which made us spend 2 years, they were having a lot of mutations, a lot of changes. That means they have already matured.
Right. So, but the question is, did you actually sort for naive B cells before you made the library?
No. So we got, we did not sort, because.
Well, that's why you got all these mutations, right?
Yeah.
Okay.
Thank you.
Okay. Thank you very much, sir.
From the last speaker of the session, Dr. Arun Sankaradas, for his presentation in dengue surveillance and nucleic acid vaccine development program. He is a research and scientific lead of the dengue vaccine program in Professor Sudhir Krishna's group at National Center for Biological Sciences, Bangalore. Over to you, Dr.
Is that the color mic or I need to use this one? It's okay, I can use this one. It's no problem. Yeah. Thank you to the chairs. And I would, first of all, I would like to thank the organizing team for this wonderful opportunity to present our work in this important vaccine science meeting. So myself, Dr. Arun, I'm working with Professor Sudhir Krishna of NCBS Bangalore. So I'm going to talk about our work on dengue surveillance and nucleic acid-based vaccine development for dengue viruses. Can I go ahead and explain straight to this one? In this talk, I'm going to cover 4 major things which we have done in our study. First, dengue surveillance, it's a dengue genomic surveillance, which we have done in India, and our vaccine design approach, and also our delivery system, which we used nucleic acid vaccine for our antigen delivery. And also we have done immune efficacy analysis to understand the immunogenicity and efficacy of the vaccine, which we have developed. And also in addition to our dengue vaccine program in India, we also develop our capacity in Africa. Yeah, I would like to start with the problem which we are facing with dengue vaccine development, because even 50 or 60 plus years of dengue threats in India, but we don't have an effective vaccine for dengue infection, because 2 major challenges have been associated with the dengue infection. One is dengue serotype and the genotype diversity, and then antibody dependent enhancement, because dengue consists of 4 viruses. Within the 4 serotype, there are multiple strains are there. Therefore, the name genotypes. Initially thought that infection with 1 serotype will provide lifelong protection against the same strains of the serotype. But that is not the case, because recent epidemiological studies challenge this assumption because repeated homotypic reinfection cases. Further, this was confirmed by the dengue vaccine, which was developed by Sanofi Foster. There's a leading dengue vaccine candidate so far, that's called Denvaxia, which induced high titers against dengue 1, dengue 3, and dengue 4, but performed very remarkably poor against dengue 2. Later sequence analysis confirmed that vaccine backbone, which they use, completely different from the strain which they are actually circulating in the clinical trial set area. Further, this is the first concern. Second is the classical one. Everyone is talking about antibody dependent enhancement, because infection with 1 serotype, instead of providing protection, it increases disease severity. This is a challenge. These 2, while we are thinking about dengue vaccine development, these 2 challenges need to be addressed. First, I will run through our dengue NGS-based surveillance program, which we have done in India. This study was conducted in 4 major hotspot regions in India.
Bangalore, Mumbai, Jodhpur, and Delhi. This is our pipeline which we followed. We have collected dengue NS1 positive samples, and then samples were transported to our laboratory, and then we gone for viral RNA isolation for dengue viral RNA isolation, and further we confirmed for nested PCR. Because dengue NS1 has high cross-reactivity with other flaviviruses. To confirm that, we have gone for nested PCR for further confirmation. Also, we serotyped patients based on IgG and IgM serology to confirm that whether they are from primary infection or the secondary infection. And we also classified them based on WHO criteria, which they fall in dengue severe or non-severe, with or without dengue cases. Further, we used Illumina MySeq library preparation for whole genome sequencing, and also we used our in-house build bioinformatic pipeline for data analysis. Around 1,000 dengue NS1 positive samples we have processed in our study, out of which 319 dengue high viral load samples were utilized for whole genome based sequencing approach, out of which we obtained 120 whole genome sequences and 270 near complete genome sequences. Yeah. Further, we investigated serotype diversity. Based on our genomics data, we investigated serotype diversity in India. Our data confirms that all 4 serotypes were circulating in India. However, 1 or 2 genotypes were dominating the outbreaks. In our study indicates that dengue 1, dengue 2, dengue 3, these are the 3 dominant serotypes which are circulating in India. And also we noticed that recent times, dengue 4 also started to emerge in our country. Also, we have noticed that the pink color bar indicates that co-infection. There are people with 1 or 2 infection or more than 2 infection. In the case of 1 and 2, 2 and 3, 3 and 4, these are the combination. Initially, we observed, we are very curious to know whether this co-infection is related to the leads to any severity of the disease. But in our data set, we haven't seen much difference in the co-infection, much difference as well as any correlation between co-infection and disease severity. Further, we investigated genotype diversity by phylogenetic based analysis. Our data indicates that even though there are multiple genotypes are circulating globally, for example, dengue 1, there are 5 different genotypes are circulating globally. Our Indian strains are belongs to genotype 3. And also we have noticed few strains were clustered with genotype 1, but these sequences are early 2000. Or late before 2000 sequences, but recent time there is no dengue 1 genotype 1 was circulating in India. Further, we investigated dengue 2 genotype diversity. We have seen our Indian strains are belonging to the cosmopolitan genotype. Dengue 3, our Indian strains belonging to genotype 3. Within the genotype 3, we have noticed 2 different clusters, clade A and clade B. Both the clusters were parallelly circulating in all 4 study sites. And dengue 4, our Indian strains are belonging to genotype 1. In conclusion, our genomics data concluded that all 4 serotypes were circulating in India. However, our Indian strains are belonging to single genotype of the serotypes. So based on this implication, we decided to target, given the concern of genotype diversity, we focused on the strains which were circulating in India rather than selling the strains which are not present in India for our vaccine design. Yeah, then I will run you through our vaccine design strategy and delivery system and an immune efficacy analysis which we have done. Yeah, this is a dengue genome. You must be aware of this. This is a single strand positive sense RNA genomes made up of 10 proteins, 7 non-structural and 3 structural protein. Most of the experimental dengue vaccines target whole organism or PRM and envelope protein of dengue viruses. Because envelope protein is the major target of immune response against dengue, and also made up of, it's a glycoprotein, dimer in size, dimer in nature. It's made up of 3 domains: domain 1, domain 2, domain 3. Domain 1 and domain 2, these are the discontinuous domains. Domain 3 is the continuous domain, and human makes high amount of antibody response against domain 1 and domain 2 of dengue envelope protein. However, the human makes only less than 5% or 5% of antibody response against ED3. However, the ED3 directed antibodies are highly neutralizing and also serotype specific. So we decided to target ED3 for our dengue vaccine development. Yes, because ED3 known to activate humoral immune responses by activating B cell mediated antibody responses. In addition to envelope, we decided to target other protein called non-structural protein, which known to activate T cell mediated immune responses. In our vaccine construct, we incorporate ED3 as well as NS1 in our vaccine construct. Yeah, this is the strain because we don't want to go with the we don't want to go with the strains which are not circulating in India, and we want to go with the strains which are specific to India. Even though India has a single genotypes or distinct genotypes, there are multiple mutations that are observed in ED3 of our envelope, ED3 of dengue genomes. High frequency mutations were spotted in highlighted in red, and low frequency mutations are highlighted in yellow. So this data indicates that there are several mutations in ED3 region. Even though dengue is a low mutating virus, because of the selection pressure, because ED3 is a receptor binding domain, the selection pressure associated with the ED3 leads to the high level of mutation. So it makes it complex which strain we need to choose for our vaccine backbone. That's the question we raised. And also we have spotted the mutations which we observed in important regions of B cell and T cell epitope binding regions. So we've come up with the approach of consensus-based vaccine approach. The consensus-based vaccine works on the basis of frequency of amino acid at the particular site. So by using this method, we could able to cover most of the high frequency mutation which were observed in our Indian isolates. And further, there are possible we could miss some of the low frequency mutation. We also use some computational algorithms to confirm that this low frequency mutations doesn't have much impact on protein structure and stability. This is our strategy. In addition to our studies, incorporated dengue sequences which was collected from various outbreaks in India. This is our vaccine design strategy. We expressed ED3 of all 4 serotypes, and then also incorporated dengue to NS1 because dengue 2 is the most dominant serotype in India, and also NS1 is a highly conserved protein across the flaviviruses, specifically dengue. So T cell response, we will get a broad against dengue infections. And this we also introduced some furin cleavage sites between the antigen to facilitate the expression, and also utilized IgG leader sequences to increase the expression of our antigen. And this expression construct was cloned in PVAX1 expression vector, which was driven by CMV promoter, and also we used kanamycin for selection marker. Yeah, this is why we have chosen DNA vaccine. DNA vaccine is could be the plug and play approach, and also it would be a good choice to start with an academic research exercise for vaccine development, and also it shows the promise of safety, and also it can activate both humoral and cellular mediated immune responses, and also it's a versatile, temperature stable, cost effective, which are essential features for delivering vaccines in a resource limited setting. And also there are several concerns still associated with DNA vaccines are low immunogenicity in larger animals and in humans, and also we need a special equipment like electrophoration or other devices for the injection. Still, these things need to be in consideration while we are moving forward with DNA vaccines. Yeah, these are the current status of DNA vaccine. Our India has approved the first DNA vaccine for human application. In addition to Zycovid, there are several DNA vaccine candidates are in the various stages of the pipeline. And also it's important to note that DNA vaccines could be ideal candidate for animal vaccine. And also these DNA vaccines are already successful in fish and animal vaccines. Yeah, this is our immunization. I'm going to show some of our slides, data slides here. This is our immunization schedule. This is a 3-dose regimen. It's animals were vaccinated 50 microgram of DNA vaccine in 2 weeks apart. Each time points, blood sera was separated and measured for dengue binding antibodies as well as neutralizing antibody titers. This data indicates that FNT data indicates that our dengue vaccine candidate induced neutralizing antibody titers against all 4 serotypes. And further, as per the WHO guidelines, dengue vaccine need to be tested against recent clinical isolates. In this one, we isolated dengue viruses from Indian patient and checks neutralizing antibody titers against Indian clinical isolates. This vaccine can construct induced pan neutralizing antibody titers against WHO as well as Indian clinical isolates as well. This study we have conducted in collaboration with Dr. Guru Prasad of THSTA. And then further, we measured T cell response induced by this vaccine candidate. Animals were vaccinated, as mentioned before. This is again two-dose regimen. After the final booster, animals were sacrificed, splenocytes were isolated and measured for IFN gamma expressing cells by ELISpot and CD8 specific IFN gamma cells and CD4 plus IFN gamma cells. This data encouraging us this vaccine induced strong antigen specific T cell responses in mice. Further, we investigated effector and memory T cell responses induced by this vaccine candidate by using CD62L and CD44 as a marker. This data clearly indicates that we have seen CD8 effector and memory T cell expansion after 2 weeks of the final booster, but we haven't seen much difference in CD4 T cells, but it might take some time to establish CD4 T cell, it seems. And further, we also measured protective efficacy against live viral infection. In this case, mouse sera generated in BALC passively transferred to EG129 mice, and we measured and then 2 hours after the passive transfer, animals were challenged with lethal dengue 2 strain, and survival, clinical signs, and weight loss, body weight loss. All of this parameter was measured. We have noticed that clinical manifestation was maintained in our passive transfer group, and also nearly 50% of animals were survived over the experimental period. With control groups and other group of animals, the 100% mortality was achieved in day 6. 50% animals were survived in our vaccine passive transfer group through the entire experimental period. Yeah, this is the overall summary. We have developed indigenous genotype specific vaccine candidate. The immune response was investigated in BALC and C57 mouse models. And this data confirms that this vaccine induced pan-neutralizing antibody responses and also antigen-specific T-cell responses. And this vaccine induced immune response protective against live viral dengue infections. And this is I like to tell you one thing here is we were focused on surveillance, bioinformatics data analysis, bioinformatics capacity, and antigen design strategy, and animal models and immune efficacy analysis. These are the important parameters. This study would offer a optimal and novel dengue vaccine candidate, and also offers a pipeline for pandemic preparedness and pandemic response. Even this kind of approach can be utilized not only for human vaccine, it can be applied for One Health as well. And this is our ongoing studies. In addition to DNA vaccine, we are also moving forward with mRNA-based vaccine candidates. And also we are utilizing nanoplasmid, because 2 challenges have been associated with the DNA vaccine. One is the antibiotic selection. Second one is the integration. To avoid that, we are using nanoplasmid. This is antibiotic-free selection method technique we are using now. In addition, we are also, in addition to our antigens, we are also expressing some genetic adjuvants along with our gene. So it might increase the immunogenicity. So there is no need for electroporation kind of devices for increase the in vivo transfection efficiency of our DNA construct. Yeah, these are our publication in recent years. In addition to our dengue surveillance and vaccine program India, we are also developing a capacity for sequencing in Africa. We are working closely with University of Nairobi, Kavi Center for Clinical Research. They have a very good clinical experience in conducting clinical trials, and also they are working on both communicable, non-communicable diseases. And our route is conducting workshops there, training people, establishing facility. We already established nanopore sequencing and computational workstation there. We have also sequenced Neisseria gonorrhea, 24 Neisseria gonorrhea isolates, one of our workshops. This study was recently published in Frontiers in Microbiology. Future direction, we are going to expand our study and develop vaccines for other viruses. And so we are going to continue genome surveillance of dengue and other flaviviruses. And also we are interested to building a course program around nucleic acid vaccines and vaccine science. And also we are in the discussion with developing a AI-based artificial intelligence-based infectious disease forecasting model for dengue and other infectious pathogens. This is our team. This is nearly 10 institutes that participated in this study, including THSTI, RGCB, 4 clinical center, IISc, NCBS. This is the consortia project. Yeah. So I would like to thank our funder, Mr. Narayan Murthy, for funding this effort. Thank you.
Thank you, Arun. Before I open up, have you, so you have developed prototype DNA vaccine with animal preclinical studies. Have you involved any industry in this whole process?
Yeah, we have been discussing with industry, but we haven't, so far we haven't, there is no research industry involvement in this so far, but now there are industry approaching us. We are in discussion with them. So yes.
Sanjay.
So you know that dengue vaccine has a problem of antibody enhancements of disease. So have you done any experiment like this, that first you infect your mouse model with the dengue, rescue it, and then give the vaccine, or once we have given the vaccine, and after that you have given infection into that, so you see the enhancements of disease. Have you worked on that?
At this moment, so the work that also we haven't done antibody dependent enhancement. I say if people usually do K562, there are 2 systems usually used for measuring antibody-dependent enhancements. One is where they use K562 related cells for in vitro, and in vivo people they use the way you explained, and also immune complex immunization method. So now we are working on immune complex immunization method where we can make 100% neutralization complex, and where we can make 30%, 50%, inject them into mice and infect with the virus and check for disease enhancement. We are currently working on it.
Thank you.
Arun, one additional question, because for DNA vaccines, generally the concern, and you also said about poor immune response, both antibodies and T cell. How does your prototype function vis-a-vis the other conventional recombinant vaccines?
Yeah, actually in our dengue vaccine, we've taken optimize our construct. Our construct was codon optimized, RNA optimized. So, and also we utilized IgE leader sequences. These are the parameters would increase in vitro in vivo antigen expression in terms of construct level. So in terms of DNA vaccine immunogenicity, we have used electrophoration. After injection, the site was electrophorated. That will obviously increase the immunogenicity. When it comes to humans, yes, we need to think about using the method, maybe Formajet or already other method already in use. For example, Zycovity, they have used other approach for human application. Something we need to explore.
Yes, Ramara.
It was hard to see that. It looked like you were getting stronger neutralization against serotype 3 and 4, but not against 1 and 2. Is that true?
Yes, we are getting lower titers, but not too low, because it's also more than 300 FNT 50 values. So yes, we are getting slightly low, but the thing is actually when we are getting slightly low against dengue 1 and dengue 2, we used dengue 2 infection model to prove protective efficacy. Whatever immune response which we are getting, it's protective against live infection in animal models. Yeah, so I think that even though if it's a imbalanced immunity, because these are ED3 antibodies, they are well established to not involve in AD of infection. That AD is not a concern in this case.
Okay.
Yes.
Good presentation. I was just reminded of the fact that in 2017, there was this chim vaccine, Dengvaxia in Philippines, which Sanofi had done. And when they finished that product, somehow the Philippines government didn't accept it, because that is when they told the government that it will cause harm to those who have, harm in the layman's terms, otherwise adverse events, for those who have not been infected before. So that is the reason it didn't come to market, but there was a lot of problem with the community, the people, and the way they looked at that. So have you taken care to see that those who are not infected are not in the same position?
Yes, actually, Denvax, I would like to tell you that is a whole virus-based vaccine, which was developed by Sanofi Foster, as you said. That is not a entire dengue genome-based vaccine. They used yellow fever vaccine backbone. They removed PRM and envelope protein of yellow fever virus. Instead of PRM envelope protein, they have replaced with dengue 1, dengue 2, dengue 3, dengue 4, 4 monovalent formulation, and they mixed together, they injected to people, made a tetravalent and injected to people. The thing is actually that's the genotype diversity which they have noticed. That's the one imbalanced protective efficacy poorly against dengue 2. That's the one concern. Second thing is the vaccine, they perform very poorly against seronegative people who are seronegative. They are protected at this stage of immunization. If the same people, if they exposed to natural infection, this vaccination was considered as a primary for your first episode, your vaccination would be a second episode. This due to the antibody dependent enhancement, during the second episode, this is gone worst, and most of the kids and school kids and people end up with a severe disease. Yeah, this majorly affected younger children's.
Yeah. So my question is related to the design, and you said that ED3 there is no potential of enhancement because ED3. Actually, if you look at the results, ED3 also has potential. It just has low potential because of the less occupancy requirement. If you need antibodies for envelope whole ectodomain, 60 antibody to neutralize one virus particle, with ED3 you may end up with only 5 or 7. It has been shown with dengue, it has been shown even with West Nile virus that the potency requirement is low for ED3. that means still you need high titer when you had, and you are using DNA vaccine. That's one limitation you should keep in mind, that even ED3 antibodies, like 3H5 for dengue 2 can enhance. There are in literature, a lot of reports, even ED3 antibody enhancing the infection at particular dilution. That is one comment. Second is regarding using NS1 as vaccine. Again, there are some data that anti-NS1, high titer anti-NS1 antibody plays some role with as autoantibodies, and they go and bind to the liver cells. And then there are efforts how to remove that. So have you looked at those, removing those epitopes from NS1 so you will not generate auto response?
Yeah, actually we made some modification, because that's why I'm very open to present here. Even IP was filed for this. We made some modification because there is a certain level. If NS1 goes high, it will leads to vascular leakage as well as auto antibodies later stuff. We have taken care of our design. That's under IP, so I could not. Yes, thank you.
Yeah, interesting talk. I was just wondering for your AD, are you planning to use a check for also Zika? Because we are getting cases now and we do see that they do have effect, at least in other countries.
Yes, definitely. Actually, because Zika is already well reported, Zika antibodies could enhance dengue infection, as vice versa. So definitely in our study, we are also including Zika challenge model to check for antibody dependent enhancement.
Yeah, so first of all, very good presentation that enabled a lot of these good questions, and thanks for all the answers. I think to me, this emphasizes clearly the innovation done by you and the group. So this is a great piece of work. But I think this is where, which was being called out yesterday as well. When should you try and partner with the industry manufacturing? I think this is just the right moment. The points which are being shared are actually very relevant. There's currently, vaccines in development. You mentioned dengue vaccine already licensed, the Takeda vaccine also already licensed, and there are other vaccines in development phase 3 onwards. The DNA as a platform is a limitation. We saw the Zydus vaccine, but you saw very limited uptake. So this may be an appropriate time for you to evaluate it. This could be switched to a different expression system as well. And hence, I think you should actively try and find a manufacturing partner to see the light of the day. I think that's, you will have to do a lot of work to make it manufacturing ready, and a lot of regulatory and the CMC pieces need to be pulled in. So this will be a great time to actually find a partner very proactively. I think that's where it should be prioritizing if you could, and obviously it's the organization should be helping you there. But that's clearly, I think, the next step, which will be an inflection point for taking this program from research to licensure. Thank you.
Thank you. Actually, now we are looking this program in two aspects in vaccine. One is construct, other one is a delivery system. We are trying to take our delivery system along with our construct as a one aspect, and also we are planning to use this construct which we have developed, express in another platform, just ramp up human application. We are looking at both the angles now. Yes, our institute also supporting. We are also, there are, as I said, industry discussions also currently ongoing. Soon there will be a progress on the next level. Yeah. Any more questions?
So, Arun, thank you. So clearly, what listening to you again makes another dimension, that a lot of work at laboratory level is taking place, and with all the capacity, with the energy, probably we can move much faster. And listening to Dr. Vijay Chaudhary also, that the alternate mechanism, RSV, actually monoclonal antibody may actually be used as immunization, primary immunization. So we look forward to these technologies. Robina ji would like to give her comments about regulatory issues before we conclude. Robina.
Okay. More questions? No. Okay. So understanding that we do not have more questions, and I would rather request sir also to finally conclude with me on summarizing, if something to add on. But I would like to just focus on some of the would have arisen in this matter. I think no, you can just.
Arun, thank you very much. I'm
Thank
so
you very
sorry.
much. Thank you very much.
I'm sorry.
So thank you for all the present and which we started from the WHO process development, the WHO's been the various platform, the starting from the candidate platform, how they have first they have gone into the different areas, and we do also coming to the next. I would like to answer on that. There were a lot of questions on the regulatory and the accelerated vaccine development, the next speaker. In this, he talked about a lot of limitations in the dynamism or in the various the accelerated approval process, or where he has spoken about the various platform, like when the WHO's platform, Dr. Madhur has spoken about the various platform which has come up, like the ACTI or the COVAX platform, where all the various partners actually talk together to ensure that yes, the vaccines, there is an equity of distribution, and how the regulators and also the other industry partners and in scientific institutions, they all spoke together, they all came together in those platforms to ensure that the vaccines, like wherever possible, get distributed to the various, the last supply chain. So here in this, as it was said, I felt that while we were listening to all of them, the regulatory pathway is, as I mentioned in the beginning itself, that there is no, we still think that the regulatory pathway is there, the predictability is there, as when I say from the side of the regulators, the predictability, transparency, the regulatory guidances, and the regulatory guidances when it's publicly available, the public relation office, the direct contact with the regulators in all the aspects was done. So as I would like to always mention about the various COVID-19 meetings, like now I think we should count it about, right, like more than 240 or more, where not only the vaccines was discussed, but all the COVID-19 products were discussed and the regulatory guidelines were given. And regarding the transparency and the predictability, would like to say that many countries at that point of time did not have the they did not have even the guidelines for a first track approval or a approval process where the accelerated approval. Even I do remember when I went with when I attended the various international platform network like the ICNA or the CEPIs. So I have understood that even countries like Singapore was also not having the accelerated, and they have to create the regulations for the accelerated pathway or first track approval process. So this was in place, and then the learning through this process, like the approval, I was seeing the timelines. So it takes a lot of discussions when we come to the to a new vaccines with a compressed clinical trial program, where we have to come back again when from the clinical stage. Sometimes it has to come back again to the some preclinical studies requirement, because it's a new science, and we call about this regulatory science. So I think regulators have responded. There are more, we need to more explore, and we have to really develop more. Also the resources, the constraints, the various constraints in the various manufacturers, also in the at that point of time during the pandemic was also there. And but tools, enablers are all right now available, and also along with this, coupled with this, while going into the collaboration with the various agencies like ICMR, while developing the COVID-19 testing kits. I remember the day-to-day interaction with the ICMR for all the kits, like the COVID kits while it was coming up. And with the validation of the kits, the day-to-day interaction with the DBT for development of the the additional capacity building of the additional sites, inclusion of additional sites to include the expand the capacity. So these are some of the things I like to highlight, that the pathways and the process, they were like, it was an immediate response, and the response was made, and the timelines which have taken probably has taken its own course of time in the scientific discussion, because it was very important at that point of time to also look into have to be very sound and scientific research. It was more that we need the scientific basis and evidence, and so sometimes it was more discussion happened when there was something which is coming up in a very compressed timeline. So that is one of the point. I would like to thank all the speakers, though I'm not going into each and individual, but it was a really enlightening sessions while learning from the research work and the various regarding the ethics part, then also the development of the vaccines, also regarding the various formulation strategy, and lastly the research. And I think there was a correct intervention on that the where we should take care that what we are doing at the research points really goes ahead. So where we should start interacting, where the researchers need to start interacting with the industries to see that yes, lot of research work has placed, and what we are developing will be actually translated. So the translation is important. I think I don't know, but whether I have covered all of the things, and it was a great session to hear about this, the library. And we hear when we feel that we really see that the dearth, when we go for some approval process for the such monoclonal antibodies and all such products, we find that we feel this challenge here. I know that the source, the strain, the characterization of the things which comes as a question each, and where is the standardization? Are we ready to accept the standardization which is being done by the researchers or the manufacturers? So those are a lot of challenges which come when because biosimilar or monoclonal, other than also the biosimilar that there are challenges on the standardization. And as sir was rightly mentioning, and the people were mentioning, the most important thing is why the research could not happen very urgently was the lack of dearth of raw materials. The resource was stopped. So as we are completely dependent on in most of the cases on the imported raw materials, all important raw materials. So at one point of time, there was even for a small change, because of the particular non-availability of one of the important component of a particular vaccine, the companies or the industries, the researchers have to wait for a long time to develop an alternate process, and or alternately when they try to make some additional changes in the post-approval changes. So these are various challenges which you have seen. I think I would now request sir to add something on this. Thank you, sir.
So thank you, Rumina ji, for that summary. I would just add one sentence, that what we heard from Madhur, Neeraj, Sanjay, and Veena ji, that COVID-19 has given us a lot of experience regarding regulatory processes. And obviously, it's a dynamic process. We need to constantly upgrade and improve ourselves. And I would suggest that a stakeholder consultation involving all the key persons, including CEPI and WHO, may be convened to look at the learnings from the COVID pandemic and how we can further build on that and improve the processes. Thank you very much for your patience. Thank you.
Thank you, ma'am. Thank you, sir, for chairing and summarizing the session. And can we request all the speakers to come up on the stage for the felicitation ceremony? We request Dr. N.K. Arora to felicitate Dr. Madhur Gupta. Dr. Neeraj Jain. Okay. Dr. Sanjay Singh. Dr. Veena Vaswani. Dr. Nivedita Gupta. Dr. Arun. Dr. Arun. Dr. Vijay Kumar Chaudhary.
Thank you all. We now request Professor Dr. K. Vijay Raghavan to kindly come up on the stage and felicitate our session chairs, Dr. N.K. Arora and Dr. Rubina Bose. Thank you, sir. Thank you, ma'am. We now request you all to kindly proceed towards THSTI reception for a group photograph. The outstation delegates are requested to confirm their departure flights or train arrangements at the registration desk. We'll break for lunch right now and we'll reconvene for the concluding session by 2 p.m. Thank you. Yes.
2 p.m. by 2 p.m. Dr. Soumya Swaminathan will give her keynote address. So everybody is requested to convene 5 to 2, please. Everybody should be here at 5 to 2. She'll deliver her keynote at 2 p.m.
I request everyone to please be seated.
Hello.
We also request everyone to please keep their cellular devices on silent mode.
So start. Welcome back, everyone. Now we have a keynote address by Dr. Soumya Swaminathan. Dr. Soumya Swaminathan is an Indian pediatrician and clinical scientist globally known for her work on TB and HIV. She is the former chief scientist and deputy director general of program at WHO. She has also served a tenure as the director general of ICMR. Also, she has also served as former director at the National Institute of Research in Tuberculosis. Ma'am, we request you to deliver the keynote address. Can we request Dr. Vijay to chair the session, please?
Good afternoon, everyone. Professor Vijay Raghavan, Dr. Pramod Garg, Dr. Shalja, and distinguished colleagues, both from, I know there's both government and private sector participation in this very unique workshop or conference. And the title is also quite a challenging one about India ready to meet the 100-day challenge. And I think you've all been addressing this question and sharing your views over the last 1.5 days. Maybe what I'd like to do over the next 15 minutes or so is maybe from my experience being at the WHO at a very critical period during the pandemic, when obviously for the first time, in in 100 years actually, we've had a real pandemic with a pathogen that was highly transmissible and which also caused significant mortality and morbidity. And I think we have not fully understood the spectrum of the pathology that this virus actually produces. And I think longer term follow-up studies will tell us that it's not just a simple respiratory virus. I think a lot of people I've heard recently talk about it as another cold, another flu. But if you really look at well-conducted cohort studies, we're seeing a higher incidence of cardiovascular disease, neurological disease, diabetes, etc., in people who've been followed up. And particularly the more severe the COVID illness, the higher the occurrence of longer-term sequelae. So it's a virus that does cause multi-system disease. All of this, of course, we have learned over the last 2 years and continue to learn. In terms of countermeasures, so I think while the focus perhaps is on vaccines here in this meeting, I think it's important not to forget diagnostics and therapeutics, because the next pathogen which is going to cause a major outbreak, whether or not it's a pandemic, we're not sure whether we'll be able to develop a safe and effective vaccine as well successfully as it was possible for SARS-CoV-2. In a way, we were very lucky that the SARS-CoV-2 spike protein turned out to be the immunogen, that it was that almost all the vaccine developers used that, and it paid off in terms of very highly safe and effective vaccines. Of course, we can talk about the fact that the vaccines today are not preventing infection, particularly from the newer variants of SARS-CoV-2. And so the protective efficacy against infection has certainly come down significantly. But the fact is that it protects very well against severe disease and death, and that's what is important. But we may not be lucky next time around to have a pathogen which is as easy to make a vaccine with. And also we have to think about the fact that if you look at emerging infections, there was an analysis done, I think, over a 40-50 year period, and some 300 odd emerging infections. About 50 of them were bacteria or rickettsia. So I think bacteria, rickettsia, and fungi, and of course, we all know about antimicrobial resistance also posing a problem. So this is why I think keeping a broader perspective on developing diagnostics early, because without diagnostics, obviously, you don't understand what the pathogen is and how it's spreading and its epidemiology, and also antivirals and newer classes of antimicrobials, including for AMR. For a 100-day challenge, let's take a step back, actually, because I think while we all want to be prepared for the next pandemic and be able to develop something in 100 days, this is not cannot be the only goal of either the government or industry, because when there will be another pandemic, when there will be the need for those products, no company obviously is going to be sustainable based on a future plan for something which may or may not happen. And therefore, we need to develop a plan, I think, for India, a roadmap for what are the products which are of high priority and which are there are gaps in terms of our public health need. So if we start out by doing this exercise to lay out, what are the major causes of the disease burden in India. Of course, there are non-communicable diseases and communicable diseases, and both are important, and I'll say why. And then you have diseases for which there are good vaccines already available, and it doesn't really make sense to invest in another vaccine. For example, the mRNA platform today is really, everyone is talking about, and it's a very attractive thing. It's new technology, new platform. But if you want to develop a measles vaccine or a diphtheria vaccine using an mRNA, one really has to think, is there a need? Is there a space? Will it be cheaper or more affordable than the existing ones? And what exactly will be the advantage, right? So you have the set of diseases for which we have existing good affordable vaccines. And many of them are made in India, of course, and supplied to the world. There are, however, gaps like the cholera vaccine today. There are major supply shortages globally. The WHO stockpile for cholera vaccine is just not enough to meet the needs of the cholera outbreaks. And I don't understand really the reason why there aren't more producers of cholera vaccines. And the existing producers are going out of it. So this is, and cholera is a killer. And because of climate change and other reasons, cholera outbreaks are increasing in many countries. Then you take something like IPV or the novel polio vaccines. We know the problems with existing ones and the need for innovation there. So again, even amongst diseases which have existing vaccines, there would be some where there is a need for more innovation and more investment. Then there are the infectious diseases for which we do not have good vaccines as yet. There's some amount of R&D that's going on. These are, of course, diseases like tuberculosis, HIV, there's dengue, there's malaria, and happy to see, I saw a news report that Serum Institute has just completed their phase 3 trials with the Oxford malaria vaccine. So, but we need more. We need for Vivax, we need for falciparum. And there could be other diseases in that list. And then, of course, there's the third set of infections for which the unknown pathogen. Now, I mentioned non-communicable diseases for a particular reason, and that is because in many conditions today, monoclonal antibodies are a therapeutic option, right? Both for communicable and non-communicable diseases. Take cancers, take autoimmune diseases, and for monoclonal antibody, there's a huge gap and a global need for affordable monoclonals. So in fact, one area where I think that there is an immediate need for us to invest in, to develop on the R&D side, but also on the biomanufacturing side, is in the space of monoclonal antibodies, particularly affordable technologies. And just like, we made generic medicines for the world, it should be possible to supply, I think, high quality but affordable monoclonals for a number of different, apart from meeting our own needs. Today, many cancer patients are not getting the treatment that they should be getting because these products are not available or not affordable. So I think that there is a very good, and not only that, the monoclonal technology will then also allow us to make it quickly for these new pathogens. And as we saw early in the pandemic, there was a lot of interest and a lot of investment and a lot of hope that monoclonals would be the answer and that they would be quick, they would be very effective, they would be, and it would be possible to give them as both therapy and prevention. And in fact, the studies did show that they were effective both at treating somebody who got infected, but also at preventing a vulnerable person. You give them a shot of monoclonal and they're protected for 6 months. And these are people who may not be able to mount an immune response to vaccines or where a vaccine is contraindicated. But then what happened, of course, is with the variants emerging, the variants of concern and the mutations in the virus, the monoclonals soon, one after the other were found to be not effective. And so that a lot of hope actually died away because of that. So again, we have to think about, broadly neutralizing antibodies, and, groups like IRV have been investing for a long time looking at that for HIV, but it might be worth looking, at other pathogens as well. Before I just move into the more technical side of the vaccine development, I just wanted to also say that it's important to have good data on epidemiology and demographics as well. And in fact, that's a big gap from many developing countries. If you look at, what is the burden of X, Y, or Z, we really don't have good data. We rely on modeling, or we rely on a few studies that have been done, some seroprevalence survey here or there, and then we try to extrapolate that. It's very important to develop the capacity and have multiple centers and institutions in the country actually with for good surveillance and cohort monitoring. And it's like the cohort that THSTI has developed of infected patients, adults and children, but also other work that THSTI is doing, cohorts of pregnant women and so on. So I think that ability, capacity to have these epidemiological studies ongoing, cohorts ongoing, pregnancy registries ongoing, because suddenly when we faced the same thing, even after the vaccines were approved and used, pregnant women were the last to access. And usually that happens with every new product, that pregnant women and children are the last. And that should not happen, because we found out that pregnant women were twice as likely to get severe disease and die if they had COVID, and yet they were the last in the line to get vaccines because there wasn't enough safety data. And so the default position has always been, for safety's sake, let us keep them out of it. But actually what happened is that more of them died because they didn't have access. And it turned out that these vaccines were very safe. So I think when we're thinking about the regulatory pathway, and it's good that, the regulators also here in these discussions, we must think about how do we prioritize also pregnant women and children in the studies that are being done. And if you have ongoing pregnancy registries and sites which are capable of doing these studies very quickly, then we can save a lot of time. Let's go back to what I was saying about the roadmap. And I want to give you the example of the WHO R&D blueprint for epidemics and what happened. After the West Africa Ebola outbreak, when it was found that there was a very uncoordinated research response where many different groups from around the world came to West Africa with their own plans, their own products, their own clinical trial protocols, and they tried to set up studies. And in the middle of a really bad outbreak in countries which had very little capacity in the health system or for research. And so it was all external groups coming in to do research. And the result of it was that even though it was a massive outbreak and more than 11,000 people died, there was no product that could be properly tested to the point where it could have gone for regulatory approval. So obviously there were many things that happened during that outbreak response from which we learned, but certainly the research response was suboptimal. So the WHO was asked to develop a R&D framework or a blueprint. This was developed, it was endorsed by the World Health Assembly in 2016, and essentially it's a very systematic approach to targeting high priority pathogens. And I'm sure many of you are familiar with it. You must have seen it on the website. You have 10 pathogens that were considered to be priority pathogens. For each pathogen, there is a research roadmap that's developed. And following that, actually that pattern, we did the same thing for COVID. So in February of 2020, we had a global forum on research and innovation. And out of that came a research roadmap for COVID with listing all the research priorities in different thematic areas. And obviously at that time, there was very little known about the virus. So from epidemiology, from transmission to animal reservoirs, to clinical features and management, and the development of vaccines, therapeutics, diagnostics, there were sections and expert groups formed around those thematic areas from all around the world. And these expert groups have continued to work over the last 3 years or so, advancing the knowledge and science. Of course, it was very intense in the first year, but it continued even later. And we learned a lot, and then we kept having these periodic fora to share whatever we had learned and then to reset the priorities. And there are still many gaps in our knowledge. So then what happens is that once you have that roadmap, you then do a deep dive into each area, diagnostics, vaccines, and therapeutics, the products that you want developed, and you go and you think about developing a target product profile. And this WHO has done successfully for many diseases, including malaria, including TB. And what it does is it gives developers a benchmark. So if you say we want a vaccine which is at least 50% efficacious, we define the confidence intervals around that efficacy estimate and The safety, of course, is a given, but you also define how that safety will be defined. And then you add other properties which are either optimal, that is, would be really good if that product had those properties, or like the bare minimum, the what will be minimally acceptable. So you can say a vaccine with optimal efficacy, let's say 75%, but minimum would be 50%. And similarly, storage conditions, there are a number of criteria that one uses to develop a target product profile. Obviously, it's different for how you would do for a diagnostic or a drug or a vaccine. But like I said, the goal here is both for developers and regulators and the policymakers, you are setting the benchmark from the beginning. And of course, you can modify it later. Like we started off with an efficacy goal of 50%. And the first two results that came out had 91 and 94% efficacy, the Pfizer and Moderna products. And so there was then the realization that you could have a vaccine for SARS-CoV-2 of a much higher efficacy than 50%. And so at some point you can revise that target product profile. But you have to start with something. And you have to be very careful about it, because you do not want suboptimal, vaccine. Again, you have to, for each disease, it's different. And so for TB, you may be quite happy with a vaccine with 50% efficacy. And maybe higher efficacy in some subgroups. So all those things can be defined, subgroups, the impact on elderly, on children, and things like that. So I think that's the initial brainstorming which is needed, and there's methodology on how to do that. WHO has done it for many, but not for all, obviously. So it would be good for India to have that incorporated into the roadmap. And then the next step would also be to design clinical trial protocols. Now, this is very important, particularly for epidemic or pandemic pathogens, because once you've started the outbreak or epidemic, then you cannot sit down and start designing a clinical trial protocol. It takes a lot of time, and if you have done it in advance, it's very good. So I remember for Nipah a couple of years ago, not for the, after the first Nipah outbreak in Kerala, I think ICMR, DBT all came together and said we have to be better prepared. And there were clinical trial protocols developed. I think it even went for ethical and regulatory approvals, and a stock of monoclonal antibodies which had to be imported were kept, so that if there was an outbreak, you implement from patient one, you should be able to implement the protocol. Because for rare diseases like Nipah, which will come in spurts and suddenly, unexpectedly, and then if there's good public health response like we saw in Kerala, you will not have a lot of infected patients, right? But you can accumulate data over time by doing this type of work in different settings. You can do the same protocol in Bangladesh, the same protocol in Malaysia, or any other country where Nipah might have an outbreak. This is so hemorrhagic fevers. So these are all diseases for which we need to develop products, but we're not going to have like hundreds or thousands of patients, hopefully, to test these on. And so we need to have these type of trial designs which are approved in advance. The product is available, having undergone, of course, the phase 1 studies, and then you can implement it under a protocol. And at some point, of course, consider it for emergency use listing. And I think the Ebola outbreak of 2018, that way was, I was there and I saw on the ground how well both the vaccine and the monoclonal antibody protocols were implemented and conclusively gave answers. So it was a ring vaccine design, a ring vaccination protocol, very well done, even though it was very challenging circumstances in the Republic of Congo, in the bush, but still there was very good follow-up. The teams were extremely well trained, and it established a very high efficacy for the Merck Ebola vaccine. And subsequently, the J&J vaccine also needed to be tested, but by that time the outbreak had reduced, and so that's being tested in a different way by giving it to healthcare workers and seeing whether they are protected over the longer term from Ebola. And now, of course, as you know, in Uganda, there's an Ebola outbreak and there isn't a good vaccine that's being tested. And so some vaccines have come off the shelf now and they will be tested in Uganda. So I think preparedness to put into place a trial as quickly as possible in as little time as possible is going to be very important. And therefore, I think the development of prototype vaccines, which is what CEPI is talking about now. And so one of the things that, again, I had started the process at WHO just before I left, is the prioritization of the viral families that can potentially cause human disease, that are already known to cause human disease or that can spill over and cause. So we have probably a list of about 25 or 26 viral families which have this potential. So there will be that prioritization and listing of those families, but most likely, we know like the flu viruses and the filoviruses, the coronaviruses, the pox viruses, and so on. But like that, there are so many families. And within each family, the goal is to pick one prototype virus. And the idea of picking a prototype virus is so that a vaccine can be developed against that prototype. And I think, again, it goes back to the fact that people had already started working on vaccines for SARS-1 and MERS. And the ChAdOx vaccine, for example, was something that was being developed for MERS. And as soon as the genetic sequence of SARS-CoV-2 was available, it was put onto the same platform. So I think now the advantage is we have many platforms. Of course, there are new platforms also being developed, and we should further encourage that. But there are many platforms today that can be used. So therefore, it's worth investing in developing these prototype vaccines for each of those, well, at least for the most common pathogens. But again, we shouldn't forget the other diseases and preparing for a pandemic that may or may not come. I think we have to also think about what our public health problems are today and how are we going to address those using the capacity that we have. Now, it is going to be critical for government to invest in this process, because this is, when you talk about preparedness, the problem always has been that the money for preparedness usually is not a good amount. And then it's too late. By the time we already have a problem on our hands, then any amount of money that's being pumped in is not going to have the same impact as putting it in beforehand. And the thing, of course, is that it's invisible. It's work that's done behind the scenes. Doesn't result in a big product that the public can see. But that's what will come in handy when the whole, mRNA technology was. I'm sure all of you have read the story of Catalin Carrico and all that, and the 30 years of work that went into developing the platform. And in fact, it was being used by companies for other diseases, but it came in handy. So I think that investment always comes in handy. So I think DBT, DST, ICMR should all really have a plan on how this can be funded, such an exercise. Working, of course, with the private sector and having very clear expectations of what the private sector should do and what is expected of them in return for this type of investment. And so I think the last point I want to mention is about, I talked about the protocols and also having field sites ready to get going very quickly. Again, we saw how important that was. You need sites which have high quality expertise in running clinical trials with protections. And I think community engagement, involvement, and education is also very important, because research, of course, now after the pandemic, I think the public has heard a lot more about research, even about clinical trials and complexities of vaccine development. So I think the general public, at least, is now familiar with these things. But I think we need to educate people more. We need to be also maybe running more community advisory boards for some of our institutions, so that the community feels like they're part of the process and they're not suspicious of the research process as something that is being done to them rather than something that they're part of developing. And we had many, I remember being quite amazed that there were so many volunteers for human challenge studies early in the pandemic, when we did not know enough about the virus. There was certainly no drugs. This was before dexamethasone even had been found to be effective in treating severe COVID. There were many young people who had formed a group. It was global, but it was very prominent in Europe. And they were saying, we are there to do human challenge studies. We are willing to volunteer, but for the greater good. So to that extent, people are aware of research, what does a human challenge study involve, etc. So I think for us to get to that point, we need to start those discussions early on so that there isn't the fear. And but at the same time, I think ethics committees have to be very strong, not only in approval of protocols, but also in the monitoring of the studies and take complaints seriously if there are any complaints that are coming up. And then on the last part, of course, is the equitable access, which we saw during the pandemic did not go well. Our plan at the WHO to protect the most vulnerable, we estimated was more than 20% of the population of any country. And if the vaccines available in 21 had been shared equitably and given to those 20%, the frontline workers, health workers, and the elderly, then it's estimated about 1 million lives could have been saved in that 1 year alone. But because of the delayed rollout of vaccine in many countries, there was a preventable loss of life. And so I think we also need to, and this is where the policymakers would come in on the implementation side, is that for each outbreak, it's going to be a bit different. The next outbreak may affect children more than the elderly, right? Or we could have an outbreak that affects a particular group of people. So we need to be very quick and agile in our approach to who we are going to target, who's going to be on that first priority list of people to receive the vaccine. And make sure that, as I said, if there's a government investment into some of these technologies and platforms, the private sector ensure that there is affordable access, which the government will have the first right, of purchasing. And of course, from India, people expect that we are also a global supplier, not just for our own population. So we're expected to have the capacity to scale up. And I'm sure that you've had discussions yesterday on the scaling up process. It's good to have these discussions, I think, in advance, including on the regulatory side. And I'm sure there have been good inputs that have been given. I think the regulators did really well, in fact, faced with this type of an emergency. And I know that globally also, it was a very well coordinated, the ICMRA, which is a consortium of the medical regulators, had regular meetings, and with WHO participating to agree on many things so that they fusion. And we found that there's very little of that one country saying one thing or another country saying another thing. Some of that happened, but not much. And also there was harmonization, but also there was what is it called? A collaborative registration procedure, which countries used. Countries that don't have strong regulatory systems, they use that based on the WHO emergency use listing or pre-qualification. They should use it for other products as well. So once there is a WHO PQ, there's no reason for the procedure to happen country after country. That really delays actually the licensing of products in countries. So there are many lessons learned from this pandemic on how to better do research, how to improve clinical trial quality. There was a lot of imbalance in where the clinical trials were done. Majority of them were done in high-income countries and very few in Africa. We learned lessons about regulatory harmonization, about ethics, and all the different questions that came up on ethics, not just around research, but other things as well, public health policies, for example. And then we learned about access, the problems in vaccination programs that many countries were facing. Of course, in India, I think the vaccination program went very well. The public health machinery really worked well, but in many countries, they got stuck, couldn't scale up. And then finally, the anti-vaccine and anti-science movement, which somehow has really taken off now, especially on social media, being driven by some groups. I don't know what the vested interest is, but this is going to have an impact on populations around the world. Wherever the anti-vax may start from one country, but today it spreads around the world very quickly through social media. And we're already seeing impact on childhood immunization in many countries now, where mothers are now getting suspicious of taking their children and this did not happen before. So I think this is again something to worry about for scientists in general, as to what is this sort of trend which has started and how do we, again, it's a lot of, I think, conversations we need to have with the public in order to educate them. So maybe I'll stop there, Dr. Vijay Raghavan, and of course can take more questions later. Thanks.
So if you want to take questions, you can sit here. You can take it from here. Thank you very, very much. That was a fascinating journey, starting from the kinds of needs we have for various diseases like the, dengue, tuberculosis, and so on, and how technology such as monoclonal antibodies, others could be useful. But broadly looking at the WHO R&D blueprint emerging from a epidemic context such as Ebola, and then being modified to current situations and including R&D in therapeutics as well as diagnostics, vaccines, providing platforms, clinical trial protocols, and also now going forward, looking at families of viruses and increasing preparedness by having vaccines for them. So it's a extraordinary range which Soumya has covered, and it's, you know, speaks of a extraordinary capability and experience recently in the midst of this fire which we've been having. So I'm sure all of you have lots of questions. So please, yes.
Dr. Soumya, thanks very much for that excellent.
Could you introduce yourself also when you speak?
Yes, yes. So thanks very much. My name is Ankur Mutreja. I'm from CEPI. I lead CEPI's low and middle income country engagement. My question is, you mentioned that next pandemic or next public health emergency that affects children more than the elderly or the adults. I wonder if you could throw a little bit more light on how WHO's thinking is in terms of managing a completely unknown challenge, and if it was to impact children more, considering all the human challenge model and everything will be out of bound largely. Not that COVID vaccines were developed on the challenge, but if you could share your knowledge on that, please. Thanks.
Yeah, no, that's a good question. I don't think we have a solution for that. I just mentioned it because of the fact that the way you allocate scarce resources would depend on the groups that are more affected, and it's not necessary that the next pathogen would behave the same way as a SARS-CoV-2 did. And as far as children developing vaccines for children are concerned, I mean, I think we'll have to use a traditional approach, you know, that we have used in the past. Obviously, we're not thinking about human challenge studies or anything but in general, you can always look at the efficacy, safety and efficacy in an adult first for any product, as you know, and then of course you have to do the dose finding, etc., for children and make sure that it still continues to have the same efficacy and doesn't have any unexpected safety considerations. But I think you're raising a good point. And this is why I mentioned pregnant women and children. We need to think about in advance in our research program, how are we going to address them? So thank you for the question.
Yes. John?
Since you have been with the WHO during this pandemic, so what are the preparedness this WHO has done for encountering any future pandemics, and especially in context with African countries?
In context with African countries.
Especially, I mean, for global situation, and especially since we were lucky that at least low income countries, they were not much affected. But in case if it happens, so how are they going to control that?
Africa was affected. In fact, if you look at serological data, most Africans became seropositive before the vaccines were available on the continent. So there was widespread infection. So it's not that Africa was not affected, but because the median age is so low in most African countries, I think we didn't see the kind of death that we saw in Europe and other countries where the population is of a much higher age. But even there, a lot of cases were missed, and we don't have data on excess mortality from any of those countries. I know some post-mortem studies showed a lot of positivity in people who had died. So anyway, so I think it's a myth that low-income countries were not affected. But coming to your main point, which is on what is the WHO doing to prepare for the next pandemic, I think the problems that we saw this time cannot be solved unless there is some international binding treaty on how countries must work together or behave during such a global crisis. It cannot be each one for themselves, which is what happened. And so this is why there is now an intergovernmental negotiating body that's been set up with representatives from different countries, and they are negotiating what will become a pandemic treaty or a pandemic accord. And then it will be, they've already agreed that there should be something legally binding. So that first hurdle has been crossed, but now the text is being negotiated. And this is, of course, we'll have to wait and see. There will be different forces pulling in different directions. There will be interest groups. There's, as you know, there's, many countries try to protect their industry as well. So our goal, of course, is to have equity at the heart of that process. Whatever the solution is, the treaty should lay out how during a global pandemic, it shouldn't be that only people who have resources have access to those products. That is really the crux of it. All countries agreed that this was a problem, and they need to find wording now that will solve it. But I think, so that will be when a pandemic breaks out. But at the same time, we're also now talking about strengthening the core capacities under the international health regulations, improving surveillance. In Africa, there were only 2 labs that could do PCR at the beginning of the pandemic, in South Africa and Senegal. Today, there are many more, a lot of countries now have at least some PCR, if not sequencing facility, and the sequencing capacity is also increasing quite rapidly, and there are efforts being made. Those will, of course, have good positive effects on other, health needs as well. So I think there are always good outcomes that come out of investing for one reason, but then you can use it for others. The workforce is extremely important as well to be trained. And I would think that even in our country, we should have an exercise really for testing at the state level. Which core capacities we are good at, which we are not, where are the strengths, where are the weaknesses, where do we need to invest, where do we need to train more manpower, etc. And that's the way to prepare. But again, there is a pandemic preparedness fund now that's been set up by the G20. It's supposed to have 15 billion or something. It has $1.4 billion. That fund will start giving money to countries, low-income countries now, to build capacity for pandemic preparedness. But a lot needs to be done. And as I said at the beginning, it's very difficult to get anyone to invest in preparedness activities.
Yeah.
Wonderful talk, Dr. Soumya. You were just talking about preparedness. Now, I guess the most obvious guess would be when we're talking about what could be the next agent for the next pandemic, would be some respiratory pathogens, because they have the capacity to spread. So would we be looking at prototype viruses of, say, coronaviridae, the flu viruses? Are we looking at, what are most likely to either jump out of the, crossover from an animal to human spillover? Are we making some, calculated guesses? Yes, that's what this committee is going to do actually. They're going to look at these viral families, and many of them are respiratory viruses. And you're right, the respiratory viruses are can spread much more quickly and cause this type of pandemic more efficiently. I forgot to mention that, there's also need to invest in mucosal vaccines, nasal particularly. And I know there's a lot of work going on in India as well. And for respiratory viruses in particular, that would be a very good approach to use.
So may I? Okay, sure.
Yeah, I am Dr. Syed from Nesia Biotics. Thank you for the excellent presentation.
My question is regarding global access, which you briefly touched upon. Now, global access was really obvious during the COVID pandemic. It was quite obvious from the way the vaccine distribution happened. But there are subtle examples of inequity in vaccine delivery and access available, which you touched upon briefly. Cholera. We are having, a 100-day challenge here for pandemics, which will obviously affect everybody, including the high-income countries. But if you talk about cholera, we are failing a 100-year challenge because even after 100 years, we don't have vaccines. As per one publication, half of global cholera burden is in India, and the only manufacturer who was making the cholera vaccine in India is now exiting. So for such diseases like this, what can we do? I think industry can play their part. We can invest and for vaccine which are for public health impact rather than commercial gains. But the entire vaccine community needs to do something, not just for cholera, for other diseases like this. So your thoughts on that? Thank you.
No, I agree with you. This is why I said I think we should have a plan where we are also, it should be an inclusive, just like we're doing here, private sector, public sector sitting together. But I think the policy formulation also has to be thought of in advance. We can't tell people now go ahead and develop all these vaccines and then we don't have a plan actually to use them. So this is why I think the health ministry and other ministries really need to say that for the next 25 years, looking ahead, these are the diseases for which we, if we had a vaccine, a good vaccine, we would use it. I mean, I think HPV is an example of where, there have been tremendous delays. Luckily now that, there is a plan and there's also an Indian manufacturer, which makes it easier for the country to scale up. So we need a good debate, and this is where you need the policymakers also, including for things like TB. If, let's say someone produces a TB vaccine with 40% efficacy, is the country going to use it? And for this, you need to do some very serious modeling work, etc., to say what would be the cost benefit. And then you could also say at such and such cost, it might be cost beneficial to use a vaccine even at a lower efficacy and so on. So it's a wonderful time to actually have that discussion and I think develop the roadmap. So hopefully the science ministries will take that forward.
So, Soumya, Dr. Soumya, thank you very much for nice talk. This is Prasenjit from Regional Center for Biotechnology. So, since we are discussing about preparedness, I have just one query. That that every diseases like respiratory diseases, so not only vaccine or anything, there are several other consequences, like we learned from this pandemic, that need of oxygen and supply and all these things. So does this WHO also make the countries aware about the preparedness about these consequences of any disease, like respiratory disease?
The WHO does a lot. So yeah, I think it became obvious, as you said, and we have been talking about health system investment in universal health coverage and primary health care. And all those gaps were quite obvious during the pandemic. Oxygen shortage actually has somehow never had the attention of anybody. Many, people need oxygen for many things, including pneumonia. And there are many newborns and young children who die because they don't get oxygen in time. But the pandemic just brought it out very clearly and everybody could see those gaps. So to that extent, yes, I think all health systems, all countries saw where their gaps are. Now we need to invest. And I think what we're trying to do is to say that let's also address the issue of climate change. And when we are trying to build something new, let's do it in a way that is green and that is not contributing further to the carbon emission. So for that matter, putting in place solar systems in hospitals to be able to run some of the basic facilities, including the oxygen concentrators.
Ma'am, this is Dr. Veena Vaswani, Director, Center for Ethics in Nepal. A very good presentation, ma'am, and very insightful. What I need to know is, like this time following pandemics, the females were disproportionately affected. School children, the ladies who bore the brunt of extra load of work and other things. At the same time, when it came to vaccination, they weren't first in the line for getting vaccinated. So is there a way where we can have a gender responsive allocation of resources, where that we're not decimated to the extent that many of them are still deprived of the fruits of the research and the vaccines?
Yeah, no, I think that's a great question. And I think the gender dimension must be built into all policies. You're absolutely right, that including vaccination, but also access to other things. And yes, they did bear a disproportionate brunt of the pandemic because of the lockdowns and the increase also in gender-based violence in many countries. But as far as access to vaccines, there are also practical issues, why women or from certain communities or the elderly could not access. So all those things will have to be thought about when one is launching a vaccination program. But I like your point that any policy should be gender inclusive.
Hello. My question is regarding, as you told, that it's a informatics also regarding the on social media and everything. Even the WHO own image was under threat as people were spreading meme about this. So are you also working on putting some guideline to the media and other social medias whenever such pandemic hits? So
Yes, so there was a term coined actually during the pandemic called infodemic. And infodemic is an over availability of information. And a lot of it is misinformation or disinformation. So how do you deal with an infodemic? So it's becoming a science of its own now. It's a multidisciplinary science. You need social scientists, behavioral scientists, not just medical or biomedical people to deal with it. And of course, the communication is very important. So it's not just during a pandemic. I think this has to happen regularly. Science communication to the public, I think we need to pick it up, because suddenly you start communicating something during a pandemic. I remember speaking to somebody from one African country who said to me that their vaccine uptake was less than 5%. And the reason she said, was that the population did not have any trust in the government. And they did not trust anyone who spoke on behalf of the government. That's a long-standing issue between the people and the. So the point is that you have to build trust, and that has to be done on the regular time, not during a pandemic.
Ma'am, one question from the side.
Last question. After that, Vivina, you have a question? No. Last two.
I'm Dr. Suma from TSTI. There was an issue of IP, which became the reason for equitable access for the vaccines. So is WHO work that aspect of device policy?
On what?
IP related issues for the vaccines, because that was also one of the reasons for inequitable access. So is WHO looking at that aspect also? Because you're saying that there's some international treaty which will be legally binding for the countries.
Yes, exactly. So it will be definitely debated, and that will be the most contentious aspect, I'm sure about it. Because as you know, it was debated at the WTO as well, and there were some very limited waivers that were granted. But it will be discussed. I don't know what will be the outcome of that, but it's really going to be an important issue.
Very quickly.
Yeah,
I've been partial to the backbenchers, and the front benchers are very upset with me.
So has there been an effort with respect to a COVID sequelae, or the long COVID that has been, now is the time to actually look into it more closely? And what are the efforts that has been put by WHO or by the Indian government towards looking into this more, more carefully?
Okay, so I don't want to get into a long answer on that, but on the Indian government side, I'm not sure how many cohorts are actually running for long COVID. I hope there are a few. But on the global side, what we've tried to do is to convene a lot of groups to discuss, particularly those people, they who are suffering from long COVID, they themselves have formed a lot of groups now. And in fact, many of them are doctors who suffered and they described their symptoms and all that. That's how it first became clear that there were these post-COVID syndromes. There are some excellent cohorts that have recently, there are many publications. In fact, there are 2 systematic reviews that have just come out now on the incidence of diabetes following COVID. So I think we're just looking at more data, but at the moment, there are no guidelines on treatment or management. I think we still don't understand really the pathogenesis.
Thank you. After that, Rubina, the last person. Go ahead. Shout if the mic doesn't come to you.
Question is just in 2 specific parts. We saw during the COVID that there were certain dilutants, excipients, media, reagents that were required, or material XYZ that was required for manufacturing, supporting COVID manufacturing, which was blocked at government level. Now this treaty that is being negotiated, is it going to have clauses regarding supply of such material, which will ensure that manufacturing continues in countries which has the manufacturing capacity, so that the kind of deaths we saw during the Delta wave are avoided? That's number 1. And number 2, under the pandemic fund, is WHO going to be an advisor to that fund and also advising about building capacities in countries for these kind of materials which the developed countries have put a stop to?
Okay, so 2 questions. Let me take the second one. The Pandemic Preparedness Fund is going to be, it's in the World Bank. It's sitting in the World Bank, but yes, the technical advisory group is chaired by WHO. So WHO will make the recommendations. I mean, there's a committee, but WHO is in the chair of that to whom those funds should be dispersed and for what purposes. I think most of it will go to low-income countries. It's not going to go for this setting up manufacturing of consumables, etc. I think that's something which we need to consider in India. Are we self-sufficient or not? And if not, then there needs to be some effort made, especially for products like for the mRNA or, things where even plastics, disposables, all those were in short supply. In terms of the trade blocks that were put in place, I think countries have their own rules and national laws, like, the Defense Protection Act of the US, which is what they use to stop the export of materials. Now, that needs to be discussed more at the WTO level. It's a trade issue. And also countries will have to then see how they make their own national laws in a way that if there's a pandemic, there should be some exception or something like this. So I don't think the WHO treaty will solve that problem, but the WTO is also discussing the similar issues.
Last question.
Thank you. It is always very great to hear you. So I have a question. Because I was also a part of all the such R&D blueprint discussions, and you were always chairing those sessions, I remember. So we had a discussion on the last part of it that they're going to, WHO is going to target for one like 12 valent, like 12 types of the MARS and the SARS group, and that would be like for addressing all the type of the diseases, whether there is any further progress, because we haven't here. After that, there was no such R&D blueprint discussions after that. So that was initiated and then it was not progressed later. So that was one of the question. And I just wanted to add on to the people who was asking about the medical oxygen, that WHO initially, they have put up a specific discussion and preparing the guidelines, which was already there to upgrade and put it into the medical oxygen guideline was put up by WHO, which is published as a WHO TRS for all the medical oxygen manufacturers. So that was one of the things. So that was the target product profile for a single vaccine, which would be covering all the
like a pan coronavirus.
Pan
vaccine.
coronavirus, yes.
I'm not sure where it is actually. You have to check
yes.
on that. Yeah.
Thank you very, very much, Soumya. It's been a very fantastic talk and really nice questions and answers. Thank you.
Thank you, ma'am. Now I request felicitate Dr. Soumya.
We now begin our concluding session for the event, developing roadmap for accelerating concept, design, delivery of vaccine development for future pandemics. We request Dr. Pramod Garg to kindly come up on the stage and moderate the session. The panelists for this session are Dr. Soumya Swaminathan, Dr. K. Vijay Raghavan, Dr. Sanjay Singh, Dr. Ankur Mutreja, Dr. Harshit Jain, Dr. Prabuddha Kundu, Dr. Priya Abraham, and Bernard Zayst will be joining us online. So,
Very good afternoon, ladies and gentlemen, and I'd like to thank the esteemed panel which is here to give us more insights into how we can better prepare ourselves for the next emerging pandemic. And we are also joined here by Professor Bernard Geest, who is online with us. Thank you very much, Professor Geest, for joining. And I think we know all of the panelists here. I think they have given talks yesterday and today. So I don't think we should do another round of introductions. I'm told that Professor Geest has some slides to share. Maybe we can begin with him and to have his views. Professor Geest, can you hear us? You're mute, you can unmute yourself.
Yes, I can hear you.
Yeah, okay. If you want to share your slides and give us some insights, that will be most welcome.
Okay. Sorry, I'm starting. That's what you're saying?
Yeah.
Okay. Well, first I would like to thank you for this meeting because I learned a lot during the last 2 days. And I had a talk in the beginning with a kind of shopping list with things to improve pandemic preparedness. And all these topics were discussed during the meeting. And even what more is that India as a country is equipped to tackle these topics. I have this list here. Let me see. Yeah, so India has production facilities, large quantities, have several platforms, high quality and flexibility. So it could also contribute to the equitable access, which has been discussed during the meeting. There's high quality academic research. And we have seen several companies working on new vaccine platforms, which is important. And there's the ability, as you heard today, to develop the messenger RNA platform. We also heard about the infrastructure, which is impressive, the diagnostic laboratory, the animal testing, the BSL-3 and 4 labs, genomic sequencing lab. The regulatory authority is strong, and we heard about clinical trial network. That's still improving, but that's also going very well. And of course, there's a strong international orientation. But I heard also from the discussions that not everybody is aware of all these possibilities. And so this may be improved. And also people asked for more alignments of the many ongoing activities. So this is something which I found very interesting, and maybe also something that could be taken up later on. When I come back to the question whether India is ready to meet the CEPI 100 days target, I think that the answer is yes, but there are a number of provisions, limitations. If we would have to start today, we would have to use a production platform which is proven to be safe in large populations, so not only a limited number of those platforms, and also a platform that earlier yielded a protective vaccine. If we want to stay in the 100 days, then we could not have phase 3 clinical trials because they would take too much time. And also, we could not do a human challenge study because the time is not ready for that yet. So we would have to approve based on correlates of protection. And that means that these correlates should be known by that time. So this is something I would like your opinion on, but that's the way it is according to me. And then, of course, if we see, okay, let me. Then, of course, the most important thing still comes, I would say, but because having a vaccine is not the whole story. The rolling out of the vaccine is very important, and that was, and there are several possibilities that already were discussed. One is stockpiling before licensing. This is obvious, of course, and it also happened. And the other is building spare production capacity. And the other one is shifting production capacity from regular production to pandemic production. And all these solutions require public-private partnership, and they cost money. And these are the challenges, because reserving production capacity costs money, and who pays for that? And then, of course, the danger exists that after a number of years without an outbreak, there's a lack of urgency, and the reserve capacity will disappear. So this is a very important problem that has to be solved. Finally, looking back at the meeting, I would like to stress the importance of closing the gap between academia and industry. And I heard things like different mindsets of GLP and GMP, and the lack of awareness of licensing rules. And I can just tell you that this is a worldwide problem. It's not something typical for India. And there are some solutions. I list them here. One is, of course, frequent communication between collaboration and public-private partnerships. And the other is training of academic researchers. And both aspects can be combined to make the best of both worlds in so-called bioscience parks. They promote contacts between academia and industry. And close to my own university in Leiden, we have the Leiden Bioscience Park, and which about 150 life science and health companies work together, and the science park also is responsible and active in organizing courses, many different courses for academia and to work together with industry. And I found out by looking on the internet that also India has some of these parks, and these could be strengthened maybe to solve the problem of the gap. Let me see. Then I go.
Professor Geest, are you going to share something or?
No, I stopped sharing. So now let me see how I can do that.
Okay, it's okay. Thank you very much for your excellent comments, and which we'll take into account. So the purpose of the entire meeting here, and particularly this panel discussion, would be to develop a kind of a document which we can share with the policymakers also, so as to understand what are the challenges that we have discussed over the last 2 days, and what are the things that can be done for future. So there are many aspects that we discussed when it comes to understanding the burden of the disease pathophysiology, and how do we prevent it in the best possible manner. For example, we are discussing vaccines here. You identify the best vaccine candidate, immunogen platform, and then take it forward for immunogenicity, efficacy, adverse events, and finally marketing. So there are many aspects there. So one of the things that yesterday I mentioned, and this is very important to my mind, we always used to have a kind of foreign tag when it comes to a new product, be it a vaccine or a pharmaceutical product. But this time there was, thanks to the Indian academia and industry both together, that there was a product coming from India. Bharat Biotech was the first vaccine, then mRNA, and then you are also coming up with new technology. So what is it that the industry would require from academic institution to say, we are very happy with your product, or we are very happy with your work, or what is it that you need, whether it is timeliness, speed, quality, reproducibility, what are the things that the industry would like? And I would like both Dr. Sanjay Singh and Dr. Kundu to respond to that, so that you're happy with the Indian academic institution, which is where I sit, and which is where they also always want the academic partner, the industry partner to come in.
So apologies because I was not able to attend yesterday's talk, but I have seen most of them online. What a vaccine constitutes? So there is one particular subject which can only come from academia, which is the disease biology knowledge. For example, if we have to develop the vaccine against dengue, which are 6 or 7 or 8 group in academics are working, whether they can sit together and they can build a platform around, and this is disease biology knowledge. Disease biology knowledge means epidemiology, pathophysiology, it's possibility of antigens, possibility of correlates of protection. This is one of the biggest expectation academia feel, because we cannot create disease biology knowledge group at industry. What we can do, we work on the platform technologies and how to pass the regulation to qualify the product. We are expert of that. So this is 2 part. Then the third part comes from the regulatory side. The regulatory side, as I said in the morning itself, we have to work with each other with trust, how to enable to take an innovative product forward. Because for me, when I hang the shoes, I want few more products from India to come first. We have to wait 10 years. HPV Gardasil 9-valent was launched in US in 2014. Still, we are very happy when we are talking about 4-valent, which is not in the market. So somehow this capability has to come from these 3: academia, industry, regulatory. But one more component is very, very important into it, where policymaker and governments comes. What government need to do? What government has to pump? So these are the couple of factors which I, as an industrialist, I see. I was an academician, I'm entrepreneur. This is the way I feel, and this can be expanded by Prabodha.
Oh, thank you very much. I always believe that it's a two-way relationship. So if we ask for something, we must be also willing to give something. It never is one way. You cannot clap with one hand. So if I were to open my, or so close my eyes and look, I totally agree with Sanjay. The mechanistics of the disease cannot perhaps come from industry. If we were to look at this model of bench to bedside and divide it into three parts, POC, POC to concept, and then concept to translation. Perhaps if I were to say the POC, if it can be taken up primarily in collaboration or in sort of a conversation with industry and performed at academia, then we have a common purpose and a common goal. And then the baton passes to perhaps industry or in another sort of process determined faculty. There are also a lot of other engineering institutes and process institutes that could help the intermediate phase. Then we would come to the phase where we have to now take that small prototype or a concept and scale it up in 2-3 different opportunities. One, can we mass produce it? B, if we were able to do it, how will we reach it to the people? And third, as Sanjay very rightly said, is the regulatory. Perhaps I will tackle the regulatory question slightly later, because regulatory also impinges, and I have very strong sort of opinion on how it can really enable innovation in India, because we have certain rules and regulations. If unshackled, we would be able to really accelerate this. So my possible response or request to academic would be that can we arrive at a common language of discovery and development? You do the discovery, we will do the development.
Right. Thank you. So I think as far as the disease biology and burden is concerned, I think there's no doubt about that. It comes from academia and it's in plentiful, it's there. The next step is, once you understand the disease biology and you think it's a preventable disease by, say, for example, a vaccine, the academia will also come up with a vaccine candidate and they take it through multiple steps. But I would request Dr. Mr. Harshit Jain to, tell us more about this, because we have partnership with Panacea. He represents Panacea here. That what is it that you want from us? Because I've gone through this journey in the last 1 year or so, because we came up at THSA with multiple candidates and then we discussed with multiple partners. And then finally we have very good relationship with Panacea now. What is it that you want from us when we give you 4 antigens, 4 vaccine candidates? They say, no go, no go, no go. This is the first time I heard these words, no go. So what is it that you want?
Thank you, sir. And I must first start with, thank you for giving us the opportunity to work with you. And it has been truly an enlightening experience working so close with Indian academia. Given that we have also worked with NIH in the US on our dengue vaccine collaboration for almost 15 years, and seeing this institute in action, the entire team at Panacea was very energized on the kind of good work and thinking, the clarity of thought that we saw at THSTI. Now, like Dr. Singh and Dr. Kundu mentioned, commercial sense has to come, and then good science. Commercial sense and good science, there is no substitute. And like Dr. Kundu mentioned, we must agree on POC. So what it means, a POC for an academic and what POC means for a commercial entity is quite different. And that is the prime focus area. Whenever we say no go and go, it is primarily based on, on our understanding of commercial sense, as well as the POC that you have reached. And in what way we can enable the development in a manner that POC from WHO, POC from Government of India, and from wider public is to be reached. So we are that bridge. So from academia, what do we want apart from the biology, the fundamental of
making any vaccine or drug, we want you to be more fast. We want you to be ready, not in terms of today there is a pandemic and then you start thinking about how to get a vaccine. No, I'm talking about much before then. We need ideation banks that are shared with industry at all times. We need deeper collaborations wherein you don't wait for publications to share your ideas and novelty with us, but you trust us with the information before, so that the time, the IPR, some people were asking, was IPR an hindrance to making COVID vaccines available? No, it was not, because that was too late. At that point in time, even if we license the technology, it will mean 3 to 4 years to increase access. So we want you to, have this ideation bank, get into long-term partnerships with industry, get us on your scientific advisory boards, have an open discussion within your university on what targets you will achieve within the next 1 year. And so the biggest of all is organizational stability and long-term vision. We must have succession planning. Ideas cannot be left, or products cannot be left just because the scientist has retired or moved on to another institute. The institute, you look at Fauci. Fauci was in the US public healthcare system for more than 50 years. And he was the NIH director until today, like I think December 31st, he's retiring after 16 years at the helm. So that consistency, if in our CEPI program, every year, the BD head or the scientist at THSTI changes, you will get nowhere. Every time, every year, it will be a rediscovery. So we need succession planning and we need long-term vision that you continuously get behind it. You keep on doing what you're doing until you reach success or failure. And finally, we need empowerment and dedicated BD personnel. Scientists are good at science, not at marketing. You need the empowered business development people who can go out there, strike these deals with industry on mutually beneficial terms and do it day and night, and market your technologies, not just through publications, but through interactions with industry, through seminars, conferences, posters. And I invite everyone else to add anything.
Yeah. Thank you. Thank you very much for these very nice comments. Ankur, coming to you, because CEPI actually is looking at this consortium between academia and industry, and you're fortunate to have been funded for that. What is it that you look at when you evaluate a consortium coming out with an idea or take it forward?
Yeah, no, I think much of that has already been answered, because what industry is looking for and what academia is looking for. So what we really do is we just marry those two. What we also do is we evaluate the strengths that an academic partner is bringing to a proposal and the strengths that an industry partner is bringing, and then how those two things could really come together to accelerate something that would otherwise not be possible by industry alone or by academia alone. The other thing that we do is, and this project that THSTI and Panacea are doing is really invest in early stage proposals and take those risks. I think pandemic preparedness is all about risk taking in peacetime. When we talk of beta coronavirus vaccines, we've sort of extended the remit of those even to Sarbeco viruses. All we have out there at the moment is SARS-CoV-2 and its variants. But we are already thinking of Sarbeco virus probably even broader. So it's really preparing for the next thing during peacetime and taking that risk, not in a crisis time when there is a lot of panic and there is a lot of chaos out there. Taking a risk with a proper mitigation plan and really encouraging that exciting science and the promising experience that is there from both academia and industry side to come together and really marry into something that would have promise.
Thank you. Very well said. Wars are won during peacetime. So, Dr. Swaminathan, coming to you, CEPI, for example, is a newborn. It's only about, I think, 5 years max. 2016, 17, Dr. Vijay Raghavan was at the helm of DBT when this was conceptualized. But over the last 2 years or so, CEPI has really come up with a lot of things, diagnostics and funding also. How do you see the role of such organizations vis-a-vis WHO in terms of planning and guidance and funding? So guidance from WHO is, I think, very important, but when it comes to funding also, that's a critical component for smaller entrepreneurs and universities. So your thoughts on that?
Yeah, that's a good question, Pramod. In fact, I used to sit on the board of CEPI as the WHO representative. I think CEPI depends on WHO for the normative work and the guidance, as you said. So, developing what are the priorities, what are the target product profiles, which diseases should be looked at, etc. They also look to WHO for the regulatory guidance and support. What CEPI does, obviously, is bringing the funding and taking the risks, as Ankur was saying, and investing in early stage development. And the board has been thinking about how far should CEPI investment go. Earlier, it was take it up to phase 1, but now they're thinking you should take it even a little bit further so that you have ready technologies which can quickly be taken up and operationalized immediately during a pandemic. So for the 100-day, obviously, then you need to go, much further. So I think they bring funding, they take the risk and invest. But other things that they can do is also what they call the Enabling Sciences Program. And I think THSTI is one of the centers around the world where the lab has been created, which provides a quality assured assays, because that's extremely important. I think that having quality assured assays for, especially when you're doing product development, so that you have some standards that all companies are using. Because one of the things we saw is that every vaccine developer initially, in the beginning, was using their own assays till we came up with the WHO international units, because then you can't really compare. It's like comparing apples and oranges. So I think it's really important to have the standardized assays, quality assured, have these labs spread around the world. And that gives opportunity for all developers also to use those labs, and also for academia. I think biorepositories is another thing we need to think about. I'm not sure that CEPI is really funding such a thing, but I think at the national level, it will be very useful for us to have few institutions, at least with biorepositories of, again, samples which have been well standardized and characterized for product development. So, and then you have Gavi, which is the third piece of the puzzle, which will be the procurement and distribution. You have Gavi and UNICEF to do the global procurement and distribution of vaccines. We don't have a similar mechanism for diagnostics and therapeutics, but there is a discussion going on now about how can a global alliance for diagnostics be created as well.
Thank you very much. Dr. Priya, I'm coming to you. From the academic institution point of view, what are your thoughts? How do you attract companies, industry to come to you and say, we are interested in your product? Why don't you come along? And what is it that you want from them? What are your expectations and wish list?
I'll just speak a little about our experience in the case of Covaxin, which was the ultimate product with BBIL. They came forward and we were ripe and ready to embark on that journey. And representatives are here. One comment I'd like to make is it was a continued journey. We didn't just hand them the Wuhan virus, but we've handed them all the subsequent variants, right up to BA.5. Every time we got a variant, we'd hand it over to them in case they wanted to rejigger their vaccine, include those variants into the latest formulations of Covaxin, or even to test their intranasal vaccine or compared to Covaxin. So that's a continued engagement with any one member of industry. Some of the other experiences which I feel we could learn from was that there were other members of industry who came to us. Like, for instance, members from eBiologicals probably are here. They came to us for NHP studies. But then this whole thing had to be referred to headquarters. And then we got caught into the terms of reference and engagement. And that took time. By which time time is money, as somebody had told us that, they want us to be quick. I think he said, we should be efficient with our time and move on. And, we got embroiled in that. And then they just moved on. Maybe they came to THSTI to get it done. And likewise, we lost, we as scientists in NIV were very much interested in the science and the challenge of, a new platform. but we lost the opportunity because it got caught in all those legalities, which should be ironed out, and maybe there should be a roadmap. When there is a need, there is a, an algorithm or a protocol that everybody follows, so we can expedite things.
Right. So there are two models of, engaging with industry. One is a fee for service model. You provide a
Yes.
service, you go along with it. Second is you actually partner with them.
Yes.
So which one model would you think that you should always go for?
It depends on the level of engagement. My colleagues and myself really didn't want to just hand over a virus
Yeah.
and then, that there's no science in that. But if we engage with them, like we did the preclinical trials, even when they were doing the clinical trials, all the PRNTs we did for them, and we did it for the variants that followed. So it would not be a fee for service, but rather based on the level of engagement. Maybe, I think scientists are not really interested if your parent organization is funding it. We're not interested in how much money that fetches. Most of us are interested in the science.
Right. I think, but we should be interested in money as
Yes,
well.
to some
Money
extent,
moves the wagon.
certainly.
So,
I
there's no
think
doubt about that. But what we have done here is we are in partnership with industry, but it's collaborative research. Plus, we are doing all the assays that are required. So
yes, almost like
obviously they are
equal
funding
partners.
that. So I think you can have, both the things go together.
Yes.
In terms of collaboration
Yes,
with them.
yes, certainly.
The other issue which often comes up is IP. I think if you have right in the beginning MOU and NDA in place, I think IP issue can also be sorted out. Would you all agree with that?
I think so,
It's not something
yes.
which is very contentious that I own this, I own that. I think when it comes to developing a new product, having an equal IP would be welcome to all of them. Okay. Dr. Vijayaraghavan, sir, you have been at the helm of affairs both in DBT as well as the government of India. What are your thoughts that how would you encourage the synergy between academic institutions and industry, looking at the government's point of view? Obviously, government basically wants a product, whether it is come from the industry or academic institution, it doesn't really matter. So what are your thoughts and guidance on this issue?
So I mentioned some of this in my talk, but I'll read it in a different way. I was talking last night with Amar Ramarao, and he does a lot of his work at the non-human primate facility at Emory. And I asked him how many faculty members there are in all in that facility, who use that facility. And my guess was 5 or 10 or something, and the answer is 100. So you look at that, and then you look at Emory University's clinical research facility and the kinds of investment which goes into that. Basically, it's more than what Indian clinical research is. So we're talking about one university's capacity compared with all of India in that situation. And there are dozens of such universities and there are dozens of such companies. So my point is, when you talk about academia industry interaction, you're talking about a very narrow sliver of academia in the country, which is very minuscule compared to global academia. And you're talking about it as if it encompasses academia in India. This set of elite institutions are relatively extremely well funded, but even they are poorly funded compared to the Emorys of it. The rest of the institutions are untouched. So what is the mechanism of academia industry partnership where industry can invest in our academia and be competitive with industry anywhere else in the world? Now there are ways by which it can be done. If you open up into the broader ecosystem, the university ecosystem, the medical colleges and so on, in truly innovative ways. The only way is to invert the current situation. The current situation is all talent in our top institutions are drawn from our broader ecosystem without there being any research in that broader ecosystem. Unless our research institutions become field stations for that broader ecosystem, public or private universities, state universities in particular, we are not going to be disruptive and not going to have the capacity for having interesting partnerships. Now, you might argue that these smaller state universities have even less capacity to partner with industry. And that is not the case, certainly. I'll give you the example in engineering. The smaller engineering colleges and the smaller polytechnics even bring great value to industry in their area. And they've been, doing extremely well. Now, you might argue that, high-tech biology or biotech or medical research is different. I mean, there are many components which are different, but there are many components which are not necessarily reliant on these kinds of facilities alone. So if these institutions partner with the broader academic ecosystem and build capacity as well as with industry, something is possible. Last very quick point, we have seen a dramatic change in the attitude of the global funders, the global charities, as well as global organizations. They have moved away about 6 or 7 years ago from parachute science, going into Africa or Latin America and solving problems and flying out, into actually trying to build capacity. When CEPI was formed, the purpose of CEPI was to fight against potential new epidemics and pandemics. And the best way and the safest way to do it is to fund big US multinationals, right? That way, if you give them unlimited money, they will, do anything for you. Second best way, along with the first best way, is to also fund big Indian companies who will provide volume for the work. The organizers, the founders of CEPI at that time said, no, of course you need to have these as your partners, but you need to have African countries on the table. You need to have Indian academia on the table. You need to have Indian smaller companies on the table. And that's why CEPI today turns out to be a much more inclusive capacity building organization rather than a problem solving organization alone. And similarly, Gavi has changed the way it approaches vaccines. The WHO is, also very keen on building capacity in multiple locations. The Gates Foundation and the Wellcome Trust are into capacity building. Capacity building was a bad word for these organizations, bad hyphenated word, because for them, solving a problem is something has to be done now. Capacity building is a long haul. Unless we solve capacity building on scale in India, we're not going to be competitive. And that's a solvable problem.
That's very important. And for that, you said even smaller universities are, have to come on board.
They don't only have to come on board. They are the solution. They are the ones who have the fire in the belly. Quite frankly, our top institutions are so overwhelmed by various other kinds of tasks there that they don't have the critical mass
Or the time.
all the time.
So institutions like BIRAC, which I think is doing an excellent job, should we have more of BIRACs and more funding to BIRACs to bring together smaller universities and industry together?
BIRAC is impressive, but BIRAC is impressive because it's an oasis. Every state should have 5 or 6 BIRACs.
That's
right. A country this size, and you need a BIRAC kind of structure in every discipline. And therefore, BIRAC is impressive because going from 0 to 1, you're infinitely better. But going from 1 to 10 to 100 is a tough task, and we need to have many more BIRACs.
That's very important. Thank you very much. So the other technological issue was, Dr. Kundu, you mentioned yesterday about platform. How do you define platform? And I think when we talk about future preparedness for a new emerging disease X, if you have a platform ready and you have the sequence, so the idea is you know the sequence and you have the platform, you're done with it. So your thoughts on that? What do you mean by platform in a different manner, and which platform is better, or it has to be disease or pathogen specific platform? So is it pathogen dependent or pathogen independent platform that you're talking about?
Thank you. It's an interesting question, and maybe I will request some of my other panelists to also jump in. There are two ways to sort of answer this question, sir. One is from a scientific point of view. Can I take a platform and go ahead and clone all sorts of antigens or sort of molecules that we need, and would it serve the purpose? The second is the definition of a platform from a regulatory point of view, which I had said. That how do you accelerate using a platform? Just the ease of cloning and purification perhaps is not the entire story. That is part of the story. The greater story lies in development and scale up, as well as getting regulatory approval, at least, which is pendency for our country. Would a regulator, just because you have used the same platform and you have demonstrated efficient safety for another disease, allow you to accelerate using that toxicological data and clinical data and give you an unbiased road to go ahead? Would that be possible? And what would it take? If we can solve those issues, as we said, while the temperature is cold and we are not fighting a pandemic, that would be fantastic, because then we are not going to select and let us spend money and time on deliberating. For example, E. coli for the E. coli expression for these sort of diseases, because there is over data says E. coli would perhaps be good for it and has safety data to support it. Yeast for this, which requires perhaps more complex hydrophobic membrane-oriented antigens. Those are the kind of things that you would perhaps tackle. And at the regulatory level, they would ask, okay, you used E. coli or you used yeast. This is the kind of microcontaminants or process-related contaminants and so on and so forth. They would look at it from that view. And then we would come to a model to say, this is the way perhaps a platform would be defined. We need our definition, sir, because without definition, industry would not be willing to go ahead and sort of do it. It's well as an academic exercise. I mean, we could publish that we've done 7 variants in yeast. So what? Would the regulator allow me to go ahead with each of them in a different way or in an abbreviated fashion? That is the sort of question that we would have to answer on a platform. And of course, platforms reduce and improve efficiencies. We've seen that, provided you are willing to go with one modality. Right now, I believe there are 16 steps, and Sanjay can correct me, of unit operations to make a mRNA vaccine, if I'm not mistaken, about 16 steps. We have to go in there and then do a process consideration is, how many steps can we reduce from there, provided we were all willing to align to a certain platform. Now, once we align to a certain platform, the owner of that platform should not then exploit the process, because then you can get bottlenecking in terms of that.
Right, thank you. I will go back to Professor Raghavan's thought process, and then I will come back on this. I think Emory must be having the funding from NIH around $3 billion. This is the days when I left. What's saying? NIH funding and industry funding? Yeah, so around roughly when last time I saw Rafi Ahmad, I was around $3 billion there. Now the question comes, one is, I agree with him at scale, because NIH budget is around $40 billion and we have to compare the DBT budget with that. So one is scale. The second is they have these kind of organization like BARDA. So at NIH, I was also serving on the BARDA. So BARDA just boom. So they have not gone one platform. They have given to Moderna, they have given to Pfizer, they have given to Novavax, they have given to AstraZeneca. So AstraZeneca is adenovirus, Novavax is recombinant protein, both Moderna and Pfizer. And they said, run who can make the fastest. They are not biased about it from where it is coming. But they will take only those which makes the real difference. So industry also have to compete for the best product to make. And then you are going to be accepted. This is the part one. But the part two is, should we wait for that $3 billion to come and work? No. In a given system, we have to evolve ourselves and we have to make ourselves competitive. I'm the biggest supporter of investing, because the Prime Minister aim is 5 trillion economy. And when I had one-to-one interaction with him, I said, we can do 20 trillion economy. He said, how? I said, Apple, Google, Microsoft, Amazon, Tesla, 5 companies, all are on the technology platform, science and technology. They have $12 billion market cap, and we 1.4 billion people cannot have $20 trillion economy, because somehow science and technology have been left. But this is the greatest opportunity, somehow opportunity in disguise we have got, where everybody realized the role of science, role of life science, saving the lives. No other science could have saved the life during this pandemic. We have to take it, we have to solve big problems of our. It not only solve, if I succeed in mRNA hub taking and succeeding over there, this is a huge opportunity. So somehow I will say I don't feel myself restricted because we have limited money. We have to evolve ways into limited money. The second part I want to answer, Professor Chaudhary is sitting there. His whole desire was to have a fast display library. And when I met him 10 years before, he said he wanted to make a human fast display library. I said, given the opportunity, we should do it. It was Nimitli grant given by Professor Bahan and Professor Samir Brahmachari to us. I have to return back, his was this, but roughly it costed us around 40-45 crores and Professor got 10 crores. But maintaining this relationship, only I can maintain with him. It is not easy because he has the knowledge and he is more intelligent than me. As an industry, I have to work and I have to work and I have to work and maintain it. Finally, we are so proud we both have the fast display library. Industry academic collaboration, if has to go, it has to go hand to hand, very respectfully, because somehow these are cultural problems we have. And I have to, if I have to meet Professor Raghavan, I have to meet with the professor. That attitude industry has to have it because we are not America, we are Indian culture. So I think these opportunities are coming, industry academic relationships are happening, but we have to work closely, and finally we have to evolve it. So these are my thoughts, though these are very crude one, but I just wanted to say it
Thank
because
you.
it was in my heart.
Very important. So Harshad, I'll come back to you. When you talk about, platform technologies, I realize that one of the things you always mention is regulatory hurdles. And you say you have to have tagless, you have to have, 5 liter and 50 liter and consistent batches, E. coli versus eukaryotic. So you tell us all the problems that you will face later when you go to regulator. So when it comes to, platform and processes, should there be more innovation or no, we have to go along the path that has been,
No, sir. Innovation is always there. Irrespective of which platform or which hurdle you're trying to solve, some kind of innovation will always be there. Platforms are more about plug and play. There are some things like histidine tags or there are other problems with the eukaryotic cells. We also have to look at scalability. So when we look at hist tags, the problem is they are known to induce immunogenicity. So you cannot, so regulators have now understood that hist tags are useless. They distort science. So you have to remove it. So wherever regulators have over the years ruled that these are the things that I will not allow, industry knows firsthand because we have burned our fingers. Anthrax vaccine getting rejected from the US FDA in 2008, filed by Panacea Biotech, a 50 crore NHP challenge study was 100% safe, 100% efficacy in NHP challenge model. And then US FDA in the phase 2 meeting saying you have a histidine tag. That's a blow. So like Dr. Singh mentioned, we have to learn from each other's experiences. We have to go hand in hand. So that's the first thing we told you, that, sir, we need to remove this tag. We had a candidate from JNU, a collaboration with JNU rejected by the US FDA in 2008 after a pre-IND, after a phase 2 meeting. Not the pre-IND meeting, the phase 2 meeting. They also took some time to realize, oh shit, there's a problem. The same thing with other delivery systems. LNP-based delivery systems are the most expensive. They're at minus 80 degrees. LNPs, there are only 2 or 3 manufacturers globally. When you look at eukaryotic systems, you have, I'm sorry, I'm giving you such examples just to try and communicate my point that we, over the years, being a vaccine manufacturer or a drug discovery company, have realized that some things are better, if possible, in E. coli and other systems where the scalability is very high. We were in the midst of a pandemic and people were thinking about, hey, India is banning exports. So we knew that when we go for a global pandemic situation, you want drug substance to be manufactured everywhere in the world. I know from personal experience, I've visited so many manufacturing facilities. They can't handle a mammalian cell culture. They cannot handle CHO cell lines. They don't have the kind of sterility, the technical understanding of scaling up such programs. It was Serum Institute of India who could have handled something like this, not any other Tom, Dick, and Harry anywhere in the world. So we have, over the years, we talk to each other. It's not just, we only talk to regulators. We also talk to industry. Amongst each other, we share insights. Hey, these are the best practices that please do this next time, don't do this. We talk to institutes like Wellcome Trust, we talk to Hillman, and we talk to so many other companies around the world that also guide us through this journey, including WHO.
Right, thank you. Dr. Swaminathan, we talk about platform here, and I understand that WHO is trying to establish mRNA hub. Also mentioning. So one is a whole, the virus or a bacteria, and then you have parts of it. But, you know, central dogma, you can have DNA, RNA, or a protein, or whatever you give. So do you think in times to come, mRNA or RNA technology is the one to go for? Should industry, academic institutions, government invest more in that, given the success of the mRNA vaccine this time?
I mean, it's still new, right? We've just seen the success of the mRNA platform for the first time. And yes, it has huge potential. But I wouldn't say that every industry should now go on mRNA and forget about other platforms. No, I think the other platforms also have a role. We have tried and tested platforms, tried and tested vaccines. There's no reason why those should be abandoned in favor of mRNA. mRNA, the ease of the rapidity with which one can create it, I think that's a huge advantage. But then there's the complexities, and hopefully, you know, the work that's being done in Genova and other companies will make it much easier also to use that technology. Already we know that we have can use this vaccine at 2 to 8 degrees, whereas the previous ones had to be used kept at -70. So over the next few years, I think we will see a lot of innovations coming using RNA and perhaps in DNA as well, though it has its own complexities. But I would definitely not say to give up the other platforms. And I think that's what CEPI is doing, is they are investing in building manufacturing capacity for a number of different platforms, not just mRNA. They have that, you know, diverse investment. For us, I mean, when we started our technology transfer program at WHO, we picked mRNA at the beginning because there was a huge interest and also potential. But the goal really was to expand the technology transfer program to other technologies, including diagnostic technologies. And then now let's hope, because I think there's a lot of interest from countries from the G20 as well in developing this kind of R&D network, particularly South-South collaborations. And the manufacturing element also needs to be worked on side by side, because you can, of course, have the R&D networks, but then you also need the distributed manufacturing network. And we know that on the whole continent of Africa, there's hardly any manufacturing capacity. So that will have to come up side by side, because if both of them happen, then I think we can not only produce vaccines much faster for the next pandemic, but also address the other gaps in diseases for which there are no good vaccines at the moment.
Thank you. I think this is important because, for example, we talk about hepatitis B virus. It's the surface antigen. You give surface antigen, excellent response. And I think this is one vaccine which has shown consistently over last about 40 years or so, drastic reduction in liver-related mortality and infections all over the world, including, of course, in India. And best data came from Africa and Taiwan and here. So I think the platform when you talk about has to be somewhat dependent on the pathogen and not really go for one technology. Professor Geest, you are there. You heard our conversation about platform technologies. Any thoughts on that from your side? You can unmute yourself if you want to respond to that. Yeah.
Okay, are you asking me?
Yes. Your thoughts
okay.
on... Yes.
So what was the question?
The question is about various platforms as we move forward for new vaccines.
Yeah. Well, we need more platforms, but at the moment we are just using platforms with proven safety. Safety is a very important point. And as we found out at the adenovirus platform, and there were very rare adverse effects, but still they influence the acceptance of the vaccine. So that is a problem when we need more platforms, but they should be tested in large populations to be sure that there are. So this is a catch-22, because once you have them, you should test them, but you can only test them when there's an outbreak.
Thank you. So one other issue that was discussed yesterday, I think touched upon today also, was the supply chain constraints that many of the industry people have faced during last 2 years or so in terms of scaling the capacity to produce more vaccines. Professor Agwan, if you could tell us, from the government's point of view, how could you solve their problem or facilitate, setting up of industry which can manufacture, innovate, and also supply whatever is needed for such ventures?
No, I think the supply chain issue is a critical one, which the entire globe faced. As a consequence of globalization, you had networks of supply chains with a very few extremely powerful nodes over there. When those nodes became inaccessible due to the pandemic for a variety of reasons, either nationalistic or other kinds of temporary blockages and so on and so forth, the supply chains collapse. And this is true on huge scale for a variety of products, but particularly in the pandemic, in the healthcare sector, many were affected. The semiconductor industry, for example, is a huge, has a huge impact on that. So that's that. Now, from an Indian perspective, if you look at the chemistry industry 3 decades ago, it was extremely powerful. And all these issues about gene synthesis or even the basic building blocks required for gene and protein synthesis, all of that were done, manufactured by Indian chemical industry in significant ways, small industries, very effectively. When the prices of raw material became much cheaper in China, for China, and the products which China could produce became much cheaper, then this basically dried up the chemical industry into purchasing finished products and then adding value to them and still becoming very powerful. So a different kind of niche was occupied. So it's not just the lab biotech industry, but the entire supply chains globally have to be relooked. I mean, the dragon in the room needs to be addressed, and I think that is addressable in terms of changing the supply chains within our laboratories, in our chemistry, and so on. We have the technology and the capabilities to do that. It's not just a scientific problem, it's also a scientific and import problem, having the right kind of non-tariff regulations which allow our industry to thrive. Because those who are able to give cheap products do it because the globe is the market. So we have the technology to manufacture many of these things, but we'll also need to be competitive to be able to market globally, because the national market is not significant enough right now. So all of this is feasible. I think there are very good government regulations which have come in, or new policies which have come in for active pharmaceutical ingredients, for example, and similarly for other areas. So this will change. It's changed in the solar energy sector substantially. That again was a point. So these are policy tweaks which the government needs to make, and they're making that. And I think we'll see the impact in another 5-year period, where you're going to see substantial increase in chemistry industry fundamentally, but also in other areas such as electronics.
So Harshad, coming to you, I mean, if you have a wish list for the supply chain issues to the government, what is it that you will ask for, or advise them, or suggest them, or request them to do for the industry? Whether it is increasing capacity in the country or supply chain from outside.
So when you look at supply chain problems, the problem is vaccines are not that lucrative for anyone. You make a vaccine for, I think government of India buys pentavalent vaccine for 30 cents. That's how much it costs just to save one life, that 30 cents. That's what the Indian vaccine industry has done, from $3 a dose in 2005 to 30 cents today. That's the innovation that we have done. Because of this 30 cents, how many cents will actually go to a raw material supplier? It's not viable business. So once we have more and more vaccines being introduced in the market, we'll have a bigger opportunity wherein there'll be more raw material suppliers wanting to be close to the vaccine manufacturers and make new products and offer their raw materials to us. Now, so this is just why raw material issues have plagued the COVID pandemic is because of this reason. Suddenly there was so much demand of billions of doses, no one was fill up their raw material capacity. Now, like sir mentioned, Government of India has production linked incentives for APIs, and in all of those programs, again, vaccines were left out, biological products were left out, raw materials of biological products were left out. But then you're right, what is our wish list? Media, filters, and we don't want large corporates making them. We want small startups making them, because we want universities getting into them, because not everyone is a good scientist. So why not use your capabilities to make something that is the best, and not at a scale of millions of liters a year. We just want 5,000-10,000 liters a year. That's all we need to make 300 million doses. We don't, in APIs, in pharmaceutical APIs, you need hundreds of tons a year for a diabetes product versus 1,000 liters for 30 million doses of a dengue vaccine.
Right.
So when you talk about this, we want, filters, everything, bags, everything to be made in India so that it's more cheaper. You can supply everywhere, but then through startups.
Right. Ankur, do you see any role of organizations like CEPI, not really CEPI, but in terms of supply chain issues, can they be facilitated?
Yeah, no, so I think an example of that is during COVID pandemic, CEPI had set up a marketplace where we connected suppliers of glass vials, for example, with rubber stoppers and with media, with those bags and pouches and syringes and whatever was needed. CEPI was able to play that central role in bringing all the key players together when people were really struggling to get these key components. The other thing we also did was we managed to at risk buy some of the critical, like adjuvants, the CPG adjuvant in large volume, and then use it in a way so that it went towards making vaccines that eventually would enable equitable access. So I think it doesn't just stop in, saying supply chain enables vaccine production, but the way supply chain can be used could also really enable equitable access down the line as well. So I think it's really an end-to-end process. We're also currently putting together a manufacturing facility network, and we are paying a lot of attention to how the supply chain network will work with our manufacturing facility network. So it's, as I said, it's all very connected.
That's very important. Yes.
I have another point. When you talk about wish list, we also need these ingredients and raw materials to be animal free. So today, majority of the vaccines that we use are in some way or the other made through animal source products. You have now, I think with the world moving towards environmental sustainability and other challenges with animal source materials, this is another innovation that academia can add by giving us. So if we use plant-based components versus animal components, our yields drop by more than half. So if academia can really, improve our technologies and increase sustainability through removal of animal-made products through the vaccine manufacturing process, that could also be a value add that India adds not just to Indian vaccine manufacturers, but increasing viability for the entire world.
Right. Thank you. The third dimension that I want to discuss is about the efficacy of vaccines. And to my mind, there are three things to discuss. One is clinical trial networks for conducting large phase 3 studies in India. And I think currently the environment is quite conducive for that. Second is, can we actually go for the controlled human infection models? And we touched upon that today. And third is that, can the regulators approve based on immune correlates of protection? So I think these are three things that we should briefly touch upon. Dr. Abraham, what are your thoughts on setting up large clinical trial networks in the country? Investment from the government. For example, I know BIRAC has actually established 5 fields clinical trial networks. ICMR is now in the process of setting up a large clinical trial network. So what are your thoughts that should industry also be involved and invest in developing this? Because, for example, a pharmaceutical company comes out with a product, they are the sponsor. They have a large network of institutions. A CRO will conduct a study for them. They invest millions of dollars into that. Why not have such a clinical trial network in the country with investment from industry and also to some extent from the government? Your thoughts on that?
I'm not averse to industry engaging with clinical trial networks on their own, as long as there's some supervision of quality from both parties, be it academia or from industry. It should just not be a cloning of clinical trial networks with compromise on high standards.
So they can't set on their own. I mean, it is only, say, for example, they will set up a trial
They will give it out,
network.
farm it out.
The one center is in CMC, one is in AIMS, and one is in GVPanth, one is in, Maulana Azad, one is in GV Nair, Bombay. So they are setting up there. They're not having on their own.
Yeah, but that's what they're doing anyway, isn't it?
So this is only for a product. They have a product, CRO engages with these people, whoever wants to come along is fine. But I'm saying as a permanent kind of a clinical trial network, you go to the coordinator, say, I have a product, I want to test it. There are 20 sites in this network, 10 may come along and say, I want to do it. So something like BIRAC has, I told you, just have done it. ICMR is doing that. So that's what I'm saying. So we should have capacity of doing this, because otherwise it takes much, much more money. Dr. Sanjay Singh on that.
So in this meeting, I think I met around 10 or 12 people to whom by face I was not knowing, but they were part of my clinical trial sites. Hire a CRO, give them job. They should be well validated by CEPI or Gates Foundation or WHO. They do a wonderful job. Only you have supervising. If like THSTI has to open a center for clinical trial, when possible, you have to ask the tough question, why you want to do it and what additional you are going to offer. There was a time, and because this is such an open discussion, there was a time Professor Bhan put me on the planning committee. And I was the youngest, so I have to do the whole, usually every elder person work. So when I was having IVRI, I was having DVT, I was having DST, I was having CSIR, I was having DVT, apologies. There are certain fancy projects which were in every place. And when I went back and I said that why everybody is doing these fancy words. So I think your idea to have a human challenge model, THSTI is the ideal place to do it. Ideal place, because nobody else can do it. I have tried to develop it 10 years before in the country for malaria. Somehow I was not able to fight that back because my fights were somewhere else. THSTI should not only go for human clinical trial center, but THSTI should go for human challenge trial center. Otherwise connect at VTU that NIH. Though for dengue vaccine, it will be required. For maybe some quick hour which are required like this, okay, we will require it too. For our own need, we have to innovate and we have to do it. So we have to ask the question why we want to, what is not available. I will say that I think it is finalized that PLI 2 is coming.
Yeah, I heard that.
So PLI 2 is coming. So PLI 1, we have worked so hard so we can compete for this pricing. But our manufacturer also have to stand up and we have to produce the best quality product in most affordable range. Otherwise, industry will go and buy from somewhere else if we have to produce. In PLI 2, biotechnology things are going to form. For example, we faced the biggest problem because nothing was established in mRNA. From same day we started backward integrating, we have made 90% things of our own. The professor knows it. He gave 5 crore to me for doing that. So basically what we want to do, T7 polymerase. We went back, we made it. And we made it, nobody now makes it like that. T7 polymerase was having some animal component, human serum albumin. We have removed, we have added something sucrose and we have lyophilized, we're doing absolutely
something like backward integration.
So we were having lipids. For making nanolipids and nano particle is lipid the most important. Cordian was in Switzerland. Pfizer gone and bought their whole supply for 3 years. They said, thank you very much. We cannot because our supplies are gone. We gone back to our partners. We said, make it lipid, went into the lab, put our scientists there. We made it. And now we made it 1,000 times cheaper, CGMP grade. And thanks to my regulatory authorities, thanks to Dr. Somani, gave permission in 1 day. He said, follow, gave permission to 1 day. So we have to choose which platform we have to do, why we should do it, what is cost primitive, and what is the demand. These things, scientists, we also have to do by ourselves. So if there is a whole chain, there are 3 enzymes are required. Only one thing we are not able to sort out. This is a cap, which is patented. But we are talking to them and we are saying that we will break the patent. Otherwise, you should give it. Otherwise, we will find out. So they can give us, they can take the royalty free of cost. We are going to manufacture here. They can get it. So I am saying that we have to go both ways. Yes, there are problems in the system, but we have to work for this and we have to figure out. For him, I am saying, I have gone to BIRAC. I have gone to the secretary, the past secretary, and I said, media is the critical thing which we don't make at all. I have holded hand of high media company, and if God is gracious, maybe in a year or 2, we'll have whole media manufacturer in the country. Bags, we face the problem. For bags, I have to go to White House with Jaisankarji. For bags, disposable bags, there are 3 facilities has been set in Pune, Chakad. They are going to make disposable bags. We have to work, we have to figure it out, we have to solve our problem. We cannot say that somebody else. And for that, we want to come back to you, people like you who are bigger, who has aimed to do it, and let's one by one solve it out. Because if we have to become powerful, have no other version other than Atmanirbhar Bharat. This I believe in our government vision. We have to become self-sufficient.
Right. I think, and the simpler things like mask and PPE were the best example for that. Okay. Professor Jees, you want to make a comment?
Yes, coming back to the colors of protection, I want to say that we need some more activity on T cells. We now have the easy neutralization assays with the pseudotypes, which can be done in large quantities. But we also know that protection against severe disease is mediated by T cells. And we had an excellent presentation yesterday about T cell assays, but what is still lacking is how to link these assays to protection. So that this is a challenge. And when that is solved, there are other challenges because we need less expensive assays because there are a lot of work and expensive. And we also need assays that we can carry out in large quantities, say to make to test a subset of the population to measure the protection rate. So this is something that should be on the agenda. It's not easy, but if you don't put it on the agenda, it won't happen.
Thank you. So, coming back to my issue of efficacy, and thank you for this, comments about the supply chains. So the clinical trial networks, the reason I'm saying is that, if you take any sites, it doesn't give you credibility. And also to the public. I know many of you might remember that about 7, 8 years ago, 10 years ago, Supreme Court actually banned all clinical trials in the country. And one of the reasons for that was that the pharmaceutical company came out with a product, they engaged a CRO, they hired, so to say, multiple sites, and the PIs had no experience of conducting research. And, they didn't even know about the disease itself. Many participants died in that. Supreme Court put a ban on that. It took us many, many years and advocacy, to come back to that. For example, DCGI said, even for a repurposed drug, you have to come to the regulator and take permission for academic institutions. For example, I am in AIIMS and I want to do a trial for a repurposed drug. You go back to the DCGI, take permission. And finally, with multiple meetings and, telling them this is not correct, for academic institution investigator-initiated trials, you actually allow that. They allowed that. So point is that when you talk about clinical trial networks, I'm talking about very, very established institutions. You have a network of 50 institutions across the country. One institution may be interested in oncology product, one is in, infectious disease, one is in diabetes, one is whatever. So you have, and they are accredited sites for clinical trial. For setting up a trial, you need to, train these sites for GCP, ethics, GLC, research methodology, ethics, and everything. And you have to have an infrastructure in place. So what I'm saying is not only the government, but also industry should think about it so that your credibility goes up. These are the academic institutions which are into clinical trials. That's the reason I was raising the issue. You have a point?
Sir, I just had one comment on this. When you talk about controlled human infection model, you talk about a ferret laboratory in India, NHP. Those are problems that need solving because capacity in India is limited or not available at all. When you talk about clinical trials, yes, in diabetes, in oncology, in all the other areas, majority of the sites anywhere in India, because of the huge population that we have, can do any clinical trial because they have some or the other population for that particular disease area or for that product and other things. In vaccines, that's not the case. It's a lot dependent upon epidemiology, incidents in that area, and many other scientific terms that I don't know, understand, and cannot say. But when you talk about a clinical trial network, this is a problem that does not require solving. Why? You have Jubilant, one of the leading research or CROs in India. Just like Dr. Singh mentioned, if you have a clinical trial, you hire a CRO and they get it done for you. You have GVK Biosciences, which has changed their name now. You have so many other companies, and they do for all pharmaceutical companies. If you go to Noida, Jubilant has a center there. You can look at their center and they're always ready to go for any pharmaceutical product. You tell them you have approval, within few days, they'll be ready to run your clinical trial for pharmaceutical products, not for vaccines. So where capacity is limited, not available in India, yes, I think that's a problem that needs solving. But you must also have the relevant business development people who can market and get you business to keep that running 24/7, to have a consistent pipeline of business activity.
So that is where I'm coming. When you talk about a business model of CRO and clinical trial sites, that's a different model. I'm saying an academic CRO. We have an academic CRO here, CDSA, right, at THSTI. We conduct many trials and we have many sites. When I say academic CRO, academic clinical trial network, this is much different from what you're saying. You engage a CRO, they will do it for you for a fee. But when I say academic institutions coming in, it brings in a lot of credibility and the costs are much, much less. I can tell you that. Having done some clinical trials on our own in multiple sites, not for a new drug, but we know that so many interventions, you have to do clinical trial at multiple sites. It is much simpler, much more ethical, much cheaper. Dr. Swaminathan and then Ankur.
So I agree with you, Pramod. I think that the industry model is fine, but it will only be for the products that industry is developing. And that may leave big gaps in what we actually need. So I think we need more academic clinical trial networks for repurposed drugs. As you mentioned, a good example is a national cancer grid. It started out of the Tata Memorial. I think it's been running for several years now. I really like that model because it's the National Cancer Grid is, I forget the number of institutions, but it's over 100 oncology centers around the world. And they do not only clinical trials, high quality, to reduce the cost of treatment and to have more Indian appropriate treatment regimens for cancer, but also things like, telepathology, teleradiology, capacity building, so that the smaller centers are benefiting from the expertise of places like Tata Memorial. And I think we need to study model like that. The National Cancer Grid, right, completely developed in India, but I think today it's showing results.
Very good results.
We should have such things for infectious diseases.
The ICMR TB trial is being done by CDSA,
Yes.
There's so many sites. We completed the, vaccination. They are being followed up now. So this is a model, I think, which is worth, pursuing. And also it brings
During-
quality people into it.
During the pandemic, I felt that there was a lack of enough clinical trials being done in India.
Exactly.
Yeah, they weren't good
And
enough,
public,
big enough.
public credit, public trust also increases once you have big institutions in place. Dr. Ankur and then Dr. Abraham and then.
I also totally agree that academia coming into clinical trial sites. I think there are examples. NICED in Calcutta, one of the ICMR institutes, has been absolutely critical in cholera vaccine licensure and actually the same data used for PQ as well later on, all the way from Dukoral several years ago now, and then UV-Col and others. So I think they've played a crucial role. And then, also I'd like to go back to the point I think you mentioned in your presentation yesterday, that the courses that you are setting up here, such as the Masters in Clinical Research, I think those kinds of initiatives are absolutely absolutely critical for these academic clinical trial capabilities to come up, because you have to really inculcate that from the root level. So well done on that as well.
Yes,
I just want to quickly echo that, yes, academic institutions must be in the driver's seat for this, because they are also able to train the PIs of the different
That's right.
sites. And there's commonality and, quality is maintained of a much higher order, rather than just grabbing sites that industries find convenient to work with. And we do not know whether there is
Right.
commonality and quality.
Yes.
Thank you.
You want to make a comment here? Yeah, please.
So yesterday, you and I spoke about offline as a private conversation about how to increase India's sort of legacy of new companies coming into science and research. If you see, venture capitalists for startups really like to get in when you have sort of de-risked some amount of the molecule. And one of the ways of de-risking is either a preclinical study or getting first in human. If you are able to have that kind of a network where young companies who do not have a lot of capital is able to access such a network, you would be doing a great service, because A, you will incentivize first in human studies in India. You will not only that, they will see now a roadmap. Somebody has to burn that road through the forest for others to follow. And if we can do that, then others would come in and you would see a great rush of products, or at least experimental drugs coming in, if not vaccines, where people would initially be able to do a 10-man study. Maybe let's select a disease like oncology, which is, greatly needed in India. And if you were able to do that, we would see a great amount of venture capital coming into these companies, and they themselves then funding and fueling this growth.
Right. Thank you. So the next issue about the efficacy is the human infection models, and Dr. Singh touched upon that. Do you think from a regulatory and ethics point of view, India ready for that? Or, one is that you create a facility, but I've seen the facilities firsthand myself, and I think the facilities outside are not really great. They're just like any other hospital. I've gone there and interacted with them. Facility is not a problem. And as you mentioned, THST is going to set up a facility like this, where we can do our observational cohort studies, we can do phase 1, phase 2 trials, and also at some stage, human infection models. We are building the infrastructure. But from a regulatory and ethics point of view, you think country is ready for that? We need more public engagement. Your thoughts, anybody, and maybe Dr. Singh?
Regulator honestly will not say anything. The regulator will not say anything. You have to put the example, you have to put your data, you have to have a pre-IND meeting with them, and based on that, they will come back. If I'm sitting at the place up, because sometimes I also have to put myself at the Rovinaji place and then say whether I'm asking the right thing or I'm asking the wrong thing. Country is ready, absolutely country is ready. The country should have done it 5 to 7, 10 years before, but country is ready for it and we should do it. But for clinical trial, at present, we have done 3 clinical trials in last, and continuously we keep on doing clinical trials. Yes, out of many people are sitting into that. We feel very happy if we have included 4 aims out of 22 sites. We feel comfortable, without a doubt.
That's right.
But here again, you have to become a businessman, and you have to say that THSTI is going to make its own network all over the country, bring ICMR, bring DBT site, put everything together, and you are going to offer better service than anybody else. And without a doubt, you can produce 10 times better service than anybody else. Cost is not a problem. For us, sustainability timelines, honesty, ethics are equally important, because we may get some advantage in the country, but we are not going to get advantage outside the country. So this is very, very important, and this can be done, but then you have to gang up and you have to put your things, and then you have all the infrastructure available. So my two answers: country is ready. I'm ready. If the Lithuania challenge model is here, tomorrow I will come to do it. Otherwise, I am going to do in England, if it has been ready. So we are ready for this, because these are the faster ways to get the product approved, without a doubt. We don't have to then do 30,000 phase 3 trials. If the 20 people human challenge data is there, then 5 to 7,000 is good enough. Statistically, we can demonstrate, and then we can keep on following, because there is no God rule is written anywhere. There is no God. These are the things which gives more safety, then people keep on doing it. So so I think country is fully ready for human challenge model.
Yeah.
Only you have to invest, you have to see the diseases which can bring the advantage to THSTI and to the nation.
Thank you. Dr. Vijay Raghavan.
Well, first of all, I think the human challenge system needs to be put in place speedily. There are many diseases for which it's not possible to do a broad trial. Good therapeutics are available, so a challenge is not risky. And new vaccines can be developed with speed in a challenge context. There are others which are more risky, but there will also be volunteers, depending on the context, that would be reasonable or not. But I would also view challenge models from a One Health perspective about livestock diseases and include that. Today, livestock vaccines are in a chaotic situation because of the absence of challenge studies facilities available in India for the kind of points which Sanjay said. If you want to test out something and see whether the vaccine is efficacious or not, you have to go to an expensive facility outside. That, if it's done for animals also, it'll have greater roles later on in pandemic protection. So both a human and a livestock challenge models are necessary.
Yeah, thank you. So the third issue is, could there be a case where you don't do a trial, phase 3 trial, and you have immune correlates of protection and say, this is good enough data for us to, approve it? Maybe Dr. Beena, would you like to comment on that? You have a disease where we have 3 vaccines available. They have shown very good protection. We know what is the immune response, antibody response, T cell response in them. Someone comes in, this is my vaccine. I show similar antibody, neutralizing antibody, T cell responses. There is no more pandemic, but I want to, get it approved. Your thoughts on that?
See, I have to go back to the regulatory requirements and always say that. So quoting the regulatory requirements, that there are provisions where the clinical trial waiver can be granted, other than where we consider like surrogate markers or even correlates of protection. So it depends on the emergency, unmet medical needs. So there are specific conditions under which the waiver can be granted. It is not that somebody probes the immune correlates of protection comparable with this, it is well established and the clinical trial waiver can be granted. Such grant is conditioned under specific, there are specific 3-4 conditions under which this is permitted, and this includes unmet medical need, and when there is a life-threatening situation, when there are specific disease of the prevalent in India, so the Indian scenario, so such kind of diseases where such waiver has been granted, and basically the permission has been given, but with a commitment of a very well set post-marketing surveillance, like a phase 4 clinical trial. So there are variable situations, and we cannot say that it can be done or it can be done.
So,
thank you. So there is a mechanism which exists.
I will add one now, because I wanted to hear the regulator view. So for biosimilar, phase 1, phase 2, phase 3 were supposed to do, but Professor Bhan built a committee and where industry and regulator sit down together. And as India, we were the first in 2014 or 2016, we wrote that if the PK and PD both are available, on phase 1, the approval should be given. We have not given approval, but US FDA gave approval to PEG GCSF based on PK and PD in phase 1. Now our regulatory authorities also, we wrote this. for the vaccines, if you have asked, so I am saying
Yeah,
to take your things. So we are working with the group. This group name is Puma S. Puma S. So if there is a vaccine, Shingrix, it is for herpes simplex virus, zoster. There, the correlates of protection are very well known. And if we are in the phase 1, we are connecting the correlates of protection, but they build the program and everything, all that 20 biomarkers, and based on that. So they have got some success in US. I think they have tried 10, all 10 they got success. Because they, by modeling, they say that this should come, and that has come in a limited model. So statistical whatsoever. So phase 1. So this is feasible based on clinical correlates of the thing. And if you want, I will connect to you with those people, how they do it and what they do it.
Sure.
That should be explored.
Thank you. Professor Jees, do you have a comment? You can unmute yourself.
Yeah, also in that respect, because it already happened recently, by both the FDA and the EMA approved the bivalent vaccine with Omicron BA.4/5 based on mouse studies. The original BA.1 bivalent vaccine was approved based on clinical studies, but the later on was completely on the basis of animal studies.
Thank you. So I would like to open the floor and take some comments and questions from the floor. Any one of you, please.
Just add to what was being discussed about the approval when we don't go for the clinical trial phase 3. So it's applicable in the biothreat scenario where the disease is very limited, say anthrax or plague. So, it's the youth environment, like Professor said that biothreats have been approved. The modern antibody for anthrax been in US. I thought there was similar one. And there is a 2-animal rule when the efficacy is 2 animals, and it you approve. So that should be looked into.
Yeah. Any other comments? Question? Yes, please. There. Yes, Madhur. Front row, please, and back there.
Thank you. And I think very, very interesting and pertinent points raised by Dr. Sanjay and also by yourself, the chair. THSTI has in fact deliberated on the issue of human challenge trials for last 3 to 4 years, along with multiple stakeholders, including the regulators and WHO as well. And I think there is a considerable progress in thinking on a consensus kind of a thinking on what are the requirements for moving ahead. The country indeed has the relevant required infrastructure in place, but there is a need, like the WHO has recently in the beginning of pandemic has brought out ethical guidelines on the human challenge studies, the principles for acceptability. So it would be a good way to proceed if ethics guidelines could be developed within India for the Indian context, adapted Indian context, with the partnership of all science agencies and with the partnership of the CDSCO. And that could set the ball rolling and set the pace for manufacturers actually following that, and then doing a human challenge study within India at some center, or potentially at THSTI center, and then providing that data to the regulator as an additional kind of measure, in addition to some clinical data or immune correlates of protection. And that is how I think a precedent would start. So to say.
Well, thank you. Thank you. Very nice point. Yes, back there first. No. 4th row, and then.
So if I may be allowed to add what Dr. Soumya Swaminathan was alluding to,
cholera. So because I'm from Nice, so I'm a cholera guy basically. But now what we have seen that cholera has come back in Haiti after 3 years, 2 years, and plenty of cases, and the age group has changed. And at this point of time, as Sanofi started withdrawing Shankal, the only BCG approved vaccine in India, so we do not have any vaccine right now. So there are some players have started working on that. That I was involved in those designs. So that design that what you are referring to, that it's a indirect, some kind of immune correlate of protection, and cholera do not have that kind of good correlate of protection. Vibrocydal is the only one, but we do not see the IgA. We don't only we look at the IgM or the vibrocydal. So in spite of that, BCGI approved that. And at the same time, what we plan to do is basically after doing the licensure, the surveillance is kind of phase 3B kind of thing and 4A, so as to see the and kind of observe a good designed observational study. So that can be a model that we can use. But definitely, as ma'am said, that cholera is definitely we are also sitting on some fire. Thank you, sir. Thank you. Mike in the front.
Very interesting discussion. I like to add one point which probably has missed, not necessarily it was part of the discussion, is the manpower. And I'm from the university, but I've interacted a lot with the industry since 1990, being advisor to Renbexy, many other companies. And my observation was that the people are hired. Maybe it has changed, but I've seen Geneva also. They are hired at a very early nascent age, right after MSc, and their training is not appropriate for industry programs. So what my suggestion is that not only one institution like TSTI, many other institutions should create programs, short courses, which are not for getting a certificate, but they empower MSCs or whosoever is to meet the need of the industry. Many of my students are in the, they are working on reasonable post. When I asked them, they said, sir, this is missing, we did under you. So idea is that, for example, to answer your histidine, and by sitting on the committees of the DBT, this histidine and vaccine, this antibiotic marker in the cholera vaccine, I've been listening for 20 years. So it is not because it was ignored, because people who were doing, they were ignorant. Because the histidine can never be used. And if somebody says the histidine is required to make a recombinant protein, that is good for an MSc project. When 7 protein in Sanjay's lab or other lab are made without any histidine tag, why can't an antigen be produced? So it is our capability. So what I'm saying, scientist capability, teachers, professor capability, maybe whatever, but we have to teach those who are going to do that question, that whether it is right or wrong, and then train them. So I think that manpower, it is a part of capacity building, real training, not that, just.
Thank you. Thank you. Very, very nice comment. Yes, Ramara. Yes, okay. And then I think these last two comments, and then we will kind of close it.
Yeah, I wanted to say more about using immune correlate as a way to not to do the phase 3 and go forward with the approval. It may not work for everything. For example, for HIV vaccine, after the RV144 study that they wanted to repeat the study, and they did this with a newer adjuvant that induced a better immune response. And then they went ahead and did the phase 3 study, and that failed. So there are a number of examples like that. So I would be very cautious in just using immune correlate as a way to approve. And the second thing about doing the clinical trials by the academia, are the centers that are the trial networks set up by the academia is, I think it's very important. I think what is equally important there is to have all these immune assays done by the standardized assays by labs in the academic. So that gives more credibility for the data. And then you can also compare data generated by different products.
Yeah, thank you. Thank you for these comments. Here, but at the back. Okay, I'll come back there, but we are running out of time. Yes, but sure.
My first suggestion is regarding, many things we learned during COVID and many things COVID taught us, including essays and then how to transport samples from one place to other place. So we should have SOPs intact. So there should be SOPs, whosoever worked on these problems, they should formulate SOPs. And these SOPs should be available at least with ICMR or DBT. Right, so it should be well documented, it should be accredited, so SOPs should be in place.
Right.
Second thing is that something like THISTI, which is here in Faridabad, at the same time, India is a big country. So such facilities should be generated at different zones in the country. So that, for example, some pandemic breaks out in Kerala, so they should, this pandemic should be controlled within that region. Right, from handling samples, treatment, developing vaccines. Same any part. So any part of the country, each zone should be independent. They should not rely. Kerala should not rely on THISTI, or
Right.
Bengal should not rely.
Again, capacity building. Yeah, thank you. Two comments from the back. Yes, lady at the back and then Jayanta.
This is for Dr. Sanjay Singh question. I just missed to ask you a question. Jayanta Bhattacharya from IIB. You touched upon mRNA on vaccine and you touched upon biologics. Do you have any thoughts on using mRNA platform to translate monoclonal antibodies? Do you have any thoughts? And if so, do you envisage any regulatory issues?
For me, yes, this concept is many people are trying to use mRNA to produce monoclonal antibodies continuously, because otherwise monoclonal antibodies are given in grams in the body to act. We have not started any program, but if you figure it out, there are 8 or 10 groups who are working on this. We are more interested in mRNA-based CAR-T technology, not mRNA-based antibody technology.
Last comment at the back.
Good afternoon, everyone. Very insightful discussions. I'm Nupur Mehrotra. I'm from Premas Biotech. I just wanted to add 2 points here. One, as Prabuddha and Sanjay both are sitting there as industry representatives, I think when we talk of preparedness for epidemics or pandemics, and we are bringing new technologies to the country, it's very important that while we are talking of how the development and research is happening, and we touched upon the regulatory part, could we have more openness about how the regulatory is looking at introducing these novel technologies into rolling it out quicker? I know it has happened with a few, but if we could have a more insightful discussion there. And the second point, which I thought panel could discuss, or if I could invite anybody to comment on, is data availability, authenticity, and the trends that we see at while we are preparing. How do you authentically look at the data? I know, Soumya ma'am, you're there, you would be looking at a lot of the trends, but how does it become available to the industry and the researchers on the regular basis, that we could all look at it and come together and say, okay, this is the way the trends to be prepared for are actually being formulated. Thank you.
So I think very important comments. Anybody like to respond to that? Or, I mean, data sharing, data availability, I think these are very, very important issues, and obviously brings about the issue of transparency also, whether it be with regulator or with trial network sites and other things. So I think the points are well taken. Since we're running out of time, I will just request each one of you to give one comment, which could be a wish list comment or a suggestion, and starting maybe from Harshit. I'll just note down if there's something that we need to incorporate in our policy document. One comment from each one of you, depending whatever, related to what we discussed or something else with regard to future preparedness in general. something that comes to your mind, or you want it to be done by the government or regulator or ethicist or whatever?
Uh, I think universities should have idea banks to identify novel targets and the fitment with which platform this works best, so that they're ready whenever something like that comes, you're ready, you know what to do.
Right. Ideation bank, I like that idea, so I've already written that.
So I would really say de-siloing the various pieces of the value chain so that the communication channel is ready when the crisis strikes.
Right, good.
I think a clear vision for the public-private partnership in product development and a commitment from both the government and the private sector on what their roles and responsibilities would be. And clearly a well-coordinated effort between the different science ministries. Each one has an important role to play and can be very complementary. But I think there needs to be, again, a frank and open discussion about who will do what and how will that all add up. And I think the data sharing point is mentioned is really also important.
Yeah, thank you.
Let's work together and solve one big problem. Like eradicate HPV, eradicate TB, something like this. So
Right.
10-year program, work together.
Very
I'm
good.
not interested in small things.
Thank you. Very good.
I think when facing a challenge with a possible pandemic, we all come to a common table, and different stakeholders, especially academic institutions, don't necessarily have to duplicate. So there are some core strengths that certain institutions have. They should be given the go signal to start with it. Others will take up somewhere else. In that rush, we shouldn't needlessly duplicate.
I thank you. Yes.
Three things, if I say short term, would love to have more such interaction where we are learning each other's language. So if THSTI or other institutions can please have more of this, where you bring us forcefully to have conversations, short term. Mid-term, capacity building. Long-term, influence youngsters to come into science. I think that's very important. If we could do that, perhaps we would have started off on solving some of this. And if I may end on a very positive note, sir, there was a salesman who came to Africa. Two of them were sent. One was a negative mindset, one was a positive mindset. Please, you can replace Africa with any other continent. I'm not biased. I've stayed in Africa for quite a while. And one person said they were shoe salesmen. He said, they don't wear shoes, so we don't have business. The other one said, they don't wear shoes. What a wonderful opportunity.
Right. Thank you. Professor Jees, before I come to Professor Raghavan. Professor Jees, do you have a final comment or suggestion?
Yes, I would try to get people together, like meetings like this, and that should culminate in a plan for the country where each party has its own responsibilities.
Thank you. Professor Raghavan, final comment from you.
Thank you. Collaboration has to increase across all sectors. Capacity building in deep tech and in deep science and deep thinking. If we have these two, I think then we're okay.
Thank you. With this, we'll end this panel discussion. I'd like to thank all the esteemed panelists here and give them a big round of applause. And thank you, Dr. Gupta, for organizing this wonderful two-day meeting, which has brought together all these stakeholders. And I think there'll be definite synergy, more collaboration and networking, working together. And I hope that we are better prepared for future. Thank you all very much.
Thank you, everyone. We now request Dr. Shailja Gupta, Chief Project Director.
So while you're taking pictures, I would also up to So thank you everyone for being here. I mean, it's not a meeting, arranging meetings are easy, but getting people to think together and work together is what makes meetings successful. So thank you all for being here and participating well.
Thank you, ma'am. With this, we come to an end to our two-day international meet on preparedness for future epidemics. Thank you for being a patient audience. It was a pleasure hosting you here at THSTI. I would request